and this is the 6th plenary session that we are having in this conference. And to chair this session, I would like to invite on stage Professor M.V. Nagabhushanam, who is currently the principal of Hindu College of Pharmacy, onto the dais. Professor M.V. Nagabhushanam has completed his M.Pharm from BITS Pilani and has PhD in pharmaceutics. He has a professional experience of 26 years and he is the fellow of Association of Biotechnology and Pharmacy and is a life member of APTI. He has a total of 120 research publications to his name, and he is the best. He achieved the Best Principal Award by Council of Skills and Competence for the year 2023 and 24. With this brief introduction, I once again welcome him and thank him for accepting our invitation to be the chairperson for this session. Speaker introduces.
Good morning to all. Good morning to all the respected dignitaries and dear students and all the innovators for the International Conference on Emerging Innovations in Pharmaceutical Sciences and Drug Discovery being organized by Chebrolu Hanumaiah Institute of Pharmaceutical Sciences. It is my pleasure and honor to be a part of this session, and I thank the organizers for making me a part of this wonderful event. And it is my pleasure to introduce and honor Dr. Sri Venkat Inkurti Garu, who is a citizen of US, renowned educationalist with more than 25 years of experience in data science and is working in the industry for the past 25 years at US and has completed his bachelor's degree in statistics from Hindu College, Guntur, and a master's in statistics from Acharya Nagarjuna University. And Sir is having his research degree in statistics from Acharya Nagarjuna University under the guidance of Professor V.L. Narasimham Garu. After completion of his PhD, he served as HOD of statistics in ASN Degree College, Tenali, Andhra Pradesh, and also worked as a statistics professor in well-known degree and PG colleges in Andhra Pradesh and Telangana. He left for USA in the year 1999 and started his career as analytics professional, and he has been serving in this IT industry for multinational clients such as Pfizer, BMS, Novartis, Amgen, and Bayer. He is based on advanced SAS certified professional from SAS Institute, USA. As a native of Guntur, he wants to contribute for the society in the field of education in India. With this intention, he started Chanakya Educational Hub, a single source for all the educational needs. Since 2005, Sir has been serving the students through this We Configure Analytics on data science and SAS programming. Sir has guided thousands of students across the globe on his specialization in data science and SAS. Among them, 90% of the students who has received guidance under his ample professional expertise are working in the industry at apex levels, having good positions. Sir is a corporate trainer for various MNCs like Novartis, CTS, Alembic, and many more other companies, and is working as a visiting professor for Southern Illinois University on data science and statistical programming, and conducted more than 300 seminars at various degree colleges, pharma institutions, engineering colleges, and management institutions, and on many universities across India. Sir is also a writer of Medidata Next and conducted in the in New York in the year 2019, attended many international conferences and also offered his services to these conferences as a chief guest. So from all his experience by conducting these seminars, he noticed a gap of, there is some more requirement for the students that what actually industry needs. And so he started his educational institution to educate the students through his expertise in the SAS and other statistical programming. With this few lines of introduction, it is my pleasure to invite Dr. Venkat Inkurti Garu, data science, a scientist, onto the dais.
Thank you, sir. Thanks very much for inviting me for this session. Thanks, Nagushan Garu. He's a good friend for me, and of course, Surya Vichadra Garu. We both are working together for last 20 years, I think. It's a pleasure. The topic is excellent. Emerging innovations. Innovation is very, very important, particularly in pharma and biotechnology industries. Pharmaceutical sciences and drug discovery. In this drug discovery, the concept is very simple. Why we are doing drugs? Why we are manufacturing drugs? We want to increase the lifespan of the patients. Always remember this word. We want to increase the lifespan of these patients. That's our intention. Remember this line. If a doctor is making a wrong diagnosis, patient may die. If a pharmacist is dispense a wrong medicine, same thing is going to be happen. That is the importance of pharmacist in the society. Continuously new technologies, new therapies, new methods comes into the picture. If you want to serve the society, always we should have this type of meetings. I really appreciate this management conducting this great seminars to reduce a gap between industry and education. Industry and education. I am in industry, you are in education. We want to reduce a gap between these two things. So, my topic is very simple and very interesting also. The topic is artificial intelligence in drug discovery and development in pharma and biotechnology. In this pharma and biotechnology, artificial intelligence. What is this artificial intelligence? Nowadays, everybody talks about AI, AI, AI. Every day you can see the newspapers, because of AI, so many layoffs are coming up. How it is going to affect our life? Our life means our careers, our jobs, our opportunities. Think about one thing. AI is going to replace 100% in some areas, 10% in some areas. So that means all industry is going to be effective with AI. It may be 100%, it may be 10%. Luckily, we are in 10% area. Because repetitive processes, all call center jobs is gone, AI. But doctors, pharmacists, discovery, Probably 10%. So we are safe, provided you must know a lot of skills. That is my intention here in this presentation. This is not like just like as out of your heads. No, we are going with very manner, down to earth. I'm going to ask a question. You can respond it. Absolutely, is fine. Okay. Now, this is our global company. We are at India, US, and even Dubai. We are planning to start even South Africa also soon. I am here. Basically, I am from Guntur. I studied a great college, Hindu college. I am a world student of Hindu college. So, I am here only because of education. Don't neglect your education. That's my sincere request to all of you. See, education is the best friend. Educated person is respected everywhere. Education beats beauty and the youth. Of course, this is my profile. We are the winners in New York metadata competition. We presented so many papers in international conferences. That conference, more than 10,000 people. This is my profile, little bit, that's fine. This is an interactive session item. These are all the numbers. If you have any questions, don't hesitate. You can send a WhatsApp message to these numbers. We will address these numbers. No? Next. Now, drug discovery. Always think about it. Drug discovery, suppose you are in pharmacy. Suppose a medicine comes to pharmacy, behind it has 15 years of hard work. 15 years. That means preclinical trials, clinical trials, preclinical trials it will take at least 2 to 3 years minimum. Clinical trials, phase 1, phase 2, phase 3, it took almost like, depending upon 10 years to 12 years, depending upon the nature of the drug. Totally 15 years of hard work, that medicine comes to pharmacy. You are going to dispense that medicine to the patients. FDA is asking 3 questions. Always remember this word, 3 questions. Where the first question is, whether the drug is really safe or not. Second is it really effective or not? Is it really effective or not? Third, there are so many medicines available in the market. I want to prove that my drug is safe, my drug is better, my drug has less side effects compared to existing drugs in the market. These 3 questions, we are going to give a perfect answer with authorization. Then FDA is giving a approval. FDA is giving the approval. Then the drug comes into the market. This is what exactly has happened. For that case, there are plenty of opportunities for professionals like you, provided you must have 5 skills. One is medical terminology, clinical trials terminology, regulatory information, biostatistics, and programming. It may be SAS or R. If you have the 5 skills, yes, you are the right person for the industries. Better to get this knowledge campus itself, in the college itself. See, it is a traditionally slow, expensive, and high risk. AI transforming every stage of pipeline. Pharmacy and biotechnology sectors are adopting AI to accelerate innovation. Previously, somebody make a phone calls. You won't believe that like every day 200 calls or 300 calls. Now they are making thousands of phone calls with AI agents, artificial intelligence agent. Suppose olden days, if I want to recruit a patient, I need to take a lot of information. Now, reverse engineering. Suppose in my, I have a smartwatch in my hand. This smartwatch is going to monitor my blood pressure every 2 minutes. My blood pressure is increases beyond the limit. Beyond the limit. Automatically, this phone is making a phone call to my emergency contacts and also 911 in US 911. And this data is stored in where? This data is stored in a database somewhere else. Suppose I have a pharmacy company. I need patient information with the age group of 45 to 55. Height is this much, weight is this much, and this blood pressure is this much levels. I don't need to recruit a single patient. System is going to provide you the data. Hope you understand this word. System is going to provide you the data. Then I am going to take the data and I am giving the medicines. I am doing my clinical trials. How you are going to do all these things? Using with the technology. Now it is so fast. Artificial intelligence is involving everywhere. Simulation. I don't want to recruit thousands of people. When you see the Bahubali of that movie, the simulation is there. So many wars are, probably 10 or 15 people are there in that war scenes. They are simulating same thing. So I am giving a treatment to 10 people using with AI simulation. I can get a results for thousands of people results. That is the situation right now. Why they want it? Corporates looking for, they want to reduce their cost. They want to reduce their cost. This is the way. And I am asking a very simple question. Quality. Who need quality? Customer or manufacturer? Customer. Everybody think about customer needs the quality. Customer needs the quality means I am using iPhone, my habit. She is using Samsung. Somebody using some other phone. Customer has so many preferences. Manufacturer, he don't have any preference. He must maintain the quality. Otherwise, he will be out of the market. Out of the market. For that case, AI, statistics. How to improve the quality without affecting cost considerations? I am giving a simple definition. I don't have any phone. I am not doing any chart GPT. Control chart is conceived and devised by the Shivot. It is a simple pictorial device for detecting unnatural patterns of variations of data. A control chart is conceived and devised by the Shivot. It is a simple pictorial device for detecting unnatural patterns of variations of data. Any one of you tell me one definition, I will give a surprise gift. Nagbhushangar is going to award that book. Of course, I am great book. I am going to give it here itself. Any one of you? You know any definition? Yes, yourself. No? Oh, you missed the opportunity. That's fine. Now going back. So, AI enables precision, prediction, automation, and personalization. The most important is personalization. These are all the things coming back to the reality. Olden days, whatever we are doing, everything is dream. Now it is reality. Drone is reality. Suppose you are talking something in your home, having with a smart TV. Next to YouTube, you will open it, you will see the advertisement for that. You will see the ads for it. Because your smart TV is listening your conversations. Olden days is different. Now everything is. Think about after 10 years. HMT watch, Bajaj scooters, Ambassador car, all are excellent products. They are all in museums. Why? They did not update their skills, update their knowledge. Simply yourself also. Update your knowledge. These type of sessions helps a lot to update your knowledge first. Okay. Most of the times when you are going to use with, clinical trials, reduces from years to months. Previously, we want to conduct this trial for so many months, so many years. Now it is going to reduce to months. That is the beauty here itself. And cut R&D cost by 40 to 60%. 40 to 60% cost is going to reduce it. Enhance prediction accuracy. Most important is accuracy. Drug target interactions. Enable rapid screening of millions of compounds. You know that compound and etc. everything. No. Support data-driven decision making. Data-driven decision making. Data is there. Too much of data is there. We want to make sure that data-driven decision making. Because using this smartwatch, all of my demographic information, all the information is stored in a database. Depending upon that, we are going to take a decision. Pharma companies are going to take a decision. Data-driven decision making. Olden days, we have data. We are collecting the data, something. No, everything is there itself. See, when you see this AI in drug discovery, When you see this time, traditional approach is 10 to 15 years. AI-driven approach is 2 to 6 years max. Similarly, cost more than $2 billion. It may be reduces to 40 to 60%. Screening, manual, very slow. Millions of screen in hours itself. When you are doing your visa transaction, that is the unauthorized transaction, you will get a call, you will get a message within a second. How many transactions in visa per second? Not per minute, per second. You have a visa in your pocket, how many transactions? At least 2 to 3 million transactions in a second across the globe. 1 million is 10 lakhs. It is going to do this. Same thing here also. Accuracy, moderate, high predictive accuracy. Failure rate, traditional approach is very high. AI is reduced due to early prediction. We are predicting something very early. That's the reason. These are all the things behind it is. So your drug discovery is badly affected with the AI concepts, AI approach. Better to learn those things now itself. See, current capabilities, future innovation. See, AI-driven drug design. Currently, we are using that one. In future, quantum AI drug discovery. Everybody talking about quantum. What is quantum? High accuracy servers. That is going to be happen very soon. Probably we are able to see the quantum university or something very soon in Amaravati, because the government is actively working on that side. I think you see the newspaper yesterday, the Honorable Chief Minister is at Davos. He asked IBM to start quantum some university in Amaravati. That will come reality for sure. So this one next, virtual screening and lead optimization. Lead optimization. You must know understanding about the words. Lead. What is lead? Just like as a customer, something like that. Autonomous molecule generation, prediction toxicity forecasting, AI design with multiple target therapies. Few days back, not few days, almost 12 years back, I signed a contract. That contract is in US. Women with more than 45 years of age, with menopause, with breast cancer. That is the study. We signed that contract. Unfortunately, we did not find a patient. That too, that study is sponsored by NIH. NIH means National Institute of Health, US federal government organization. But that rule is we want to conduct the study in US only, not outside of the country. In US, like mammogram tests, everything, because the cancer is there, they are going to identify very early stages. My condition is women with 45 years of age, with menopause, something like that. So that means, see, early prediction, that helps a lot, particularly here. See, similarly here also, oncology or whatever. See, real-time adaptive trials, AI-curated global trial registries, these are all the future innovations. You are supposed to involve in that future innovation part. Machine learning, if you want to enter into this side, probably it's beyond your head. Machine learning is very, very important. Your machine is reading all, but machine learning is very important, particularly for now deep learning, natural NLP, natural language processing, biostatistics, SAS programming, generative AI, molecule generation, reinforcement learning, computer, of course, vision, predictive analytics, and bioinformatics. If you have a knowledge in this area, that's really helpful for you to enter into innovative drug discovery area. See, when you see this, generative AI, what generative AI is going to do? Novel molecule creation, deep learning, this is going to do. Machine learning algorithms, prediction and clustering, finally data sources. This is the flow you are able to get of information. Analyze, target identification. How to identify the target? How to identify the target? See, genomic, proteomic and clinical data sets. Clinical data sets means adverse, like DEMOG, physical examination, a CONMED, medical history, etc., all these things. Just too much of data is available. We want to predict that part. And predicts druggable targets. There are so many technologies are available for this. Always about analyze geometric, genomic, proteomic, and clinical databases, all these things. See, finally, what we are going to use AlphaFold, DeepMind, IBM Watson, SAS programming, or project for drug discovery and development. Drug discovery and development. I am not like 100%. I'm a wrong person in this meeting, frankly speaking, because most of the speakers belongs to pharmacy background or bioinformatics background or biotechnology background. I am not in that area. I am with statistics background. Last 30 years, I am in clinical trials and drug development. Creating tables, figures, and listings is my expertise. What is the difference between table and listing? Table and listing means table contains statistics. Listing contains, no, just a dump from CRF. Case report form, just a dump. Lot of people, they don't know the difference of even table and listings also. Okay. Millions of compounds, within a second we are going to screen this, all these things. This is the manufacturing pipeline, raw materials, AI quality checks, automation production, real-time monitoring, predictive maintenance. There are two types of maintenance, like preventive maintenance and breakdown maintenance. You probably are all familiar with the word preventive maintenance and breakdown maintenance. Preventive maintenance means every 6 months they are going to do some maintenance. Even they are working, they want to do this. Breakdown maintenance means something has happened, they want to do this. That's the reason all these big companies, they want to do every 6 months maintenance part, because they don't want to go with the breakdowns. That's the reason it is. Structural modifications, generates novel molecules using generative models, optimize. Again, one more thing, even I am going to use ChatGPT or generative AI, etc., fine. Suppose tomorrow when you are going for interview, somebody is asking a question, they are allowing you to use your phone. Better to learn here. That's what I told you. One simple definition, you put it into next level. Next level. So you can use ChatGPT some other purposes, but whereas learning purpose, no. Once you got a job, then you can use it. It's fine. Toxicity prediction, pharmacokinetics modeling, animal studies simulation, early identification of failure risk, early identification. Because of the simulation, AI is doing lot of things. Okay. Now, area toxicity, PKPD, animal study optimization, risk prediction. What AI is going to help? AI is going to do this. Predict toxicity before animal studies. Simulates drug behavior in bodies. Simulation. Next one is reduce unnecessary experiments. Animal study optimization. We are conducting experiments. For example, what is experiment? A systematic study. Organized study is nothing but experiment. Principles of experimentation. You know what are the principles of experimentation? Replication, randomization, local control. Replication means repetition of treatments under investigation. Replication means repetition of treatments under investigation. Randomization means each and everything has equal chance. That is randomization. Local control. The pharmacy company is making a medicine. Doctor is making prescription. You are the pharmacy person is dispensing the medicine. Even though that is not good. That is not good for patient. Where is the problem? He is not follow your instructions. Not follow your instructions. I want to reduce that thought. That is nothing but local control. Local control. So any principles of experimentation, replication, randomization, local control. It's clear. So these are all the things is required. See, reduce unnecessary experiments. Identify some early failure signals. Early failure signals. That's more important is. Patient recruitment with electronic health records. That's what I told you, like everybody using smartphone. That means they are smartwatch. They are getting lot of information. So electronic health records in real world data, adaptive trial design, predictive modeling for outcomes, remote monitoring and digital biomarkers, reduce trial duration and cost. Probably you are all familiar with the word atorvastatin. Lipton, atorvastatin, you know that atorvastatin or pravastatin, etc. That sale, that's a drug from Pfizer. They invented a long time back. Patent is 10 years, for example. You know, 2010, 2011, that cost is 10 billion per year. $10 billion per year. Okay, make it 12 billions. Per month, 1 billion. Per day, 30 millions. Previously, recruitment takes like 3 years. Now I want to reduce it to 2 years, 1 year. 2 years is saving. That means $20 billion is company savings. Company is going to get it. So they want to speed up the process. If I want to recruit a diabetic patient in US, it's really tough. But here in India, if I want to recruit a diabetic patient, I will go to any hospital, I can get hundreds of diabetic patients within a day itself. Why all these companies are looking for India? Because unfortunately, India is a patient database. Cardiovascular, oncology, diabetic, everything, you can name it. Almost like 1/3 of the whole population in the universe is from India. So that's the reason they want to conduct experiments. So that's the reason they are looking into India side. That is the reason BMS, Pfizer, all these big companies, they started their offices in Hyderabad. We need to grab the opportunities. Future landscape, this is very important concept. Future, how the future looks like. AI-driven drug design, digital twins for patients, AI-powered clinical trials, and precision oncology, predictive maintenance, and quantum AI drug discovery. This is probably, it is going to happen in next 5 years. This is the concept. Think about at least 5 years. At least 5 years. Then you are able to get a success. Patient data, AI-based patient recruitment, adaptive trial design, predictive outcome modeling, remote monitoring and digital biomarkers, faster, cost-effective trials. Faster and cost-effective trials. The more important is. The next one is, I told you, real-time monitoring of production is always very important, and also predictive maintenance, automation of batch release, ensures regulatory compliance, FDA, European records, like all these. I probably are familiar with the word ICH guidelines, International Conference of Harmonization. Recently they changed the name with International Council of Harmonization. They have a lot of guidelines information, E3 guidance, E6 guidance, etc. Even though AI also must follow those guidelines. Must follow those guidelines. That's the important part. Next one is trials, treatment-based life science, etc. Everything, etc. Supporting. These are all the things behind. AI role is very important. Genomics, treatment selection, dosing and monitoring. This is important. Identifies mutations and biomarkers, and also predicts best therapy for patient. Predict best therapy. We have just like as Google. I want to go to this college, to this place. It is giving the best route. Same thing here also. Best therapy, best patient, best medicines for this. Precision dosing algorithms, AI-based digital biomarkers. Think about these things, after 10 years, what will happen? Everything is AI-based. This is like, Moderna and BioNTech, AI-driven vaccine design, reduced development time from years to weeks. You know that Corona time, we are getting a vaccine in months itself. That is the other part. Okay. AI-generated interface trials, DeepMind, AlphaMed, SolvePoint, etc. Everything. FDA's AI/ML based SAMed guidelines. Go to FDA site and you will see. Because US government is too fast, they are applying all these things there itself. Okay. What are the challenges? If you are using with AI everywhere, what are the challenges? The biggest challenges is like this: data quality and bias. Data quality and bias. That's the biggest challenge. Second one is lack of explainability. Somebody, AI taking something, somebody need to explain. That's the real problem. And the second one is integration with legacy systems. Regulatory uncertainty. Ethical concerns in genomics and patient data. Ethical concerns. Even though you are a pharmacy graduate, you are giving a pharmacy oath. Of course, you need to follow the oath. It's the same thing like this. There is the ethical concerns and genomics for patient data. These are all the challenges. And of course, data quality. Description is like this: incomplete or biased data sets, black box models, legacy systems, lack of clear AI guidelines, genomic privacy concerns. The more important is like this. The next one is, in future, everything is autonomous certain things. You know, AI-driven clinical trials, digital twins for patients, precision oncology, integration with quantum computing. If I want to calculate 10 patients, relationship between hypertension and blood pressure, I am going to use correlation. What is correlation? Relationship between two variables. What is the formula for correlation? Covariance of xy divided by standard deviation of x into standard deviation of y. What is covariance? 1 by n sigma xy minus x bar y bar, something like that. If I want to use it with 10, I can take a piece of paper and I can do this. If I want to go with 100, I can use calculator. If I want to go with millions of patients, is it possible in this case? No. I am going to use technology, software. If I want to go with billions of people, there quantum comes into the picture. Quantum comes into the picture. That is the reason government is always thinking about quantum technologies. Next. Finally, this is the important here. AI is augmenting, not replacing humans. AI is augmenting. But remember this one. The future will be a collaborative model. AI handles repetitive, large-scale data tasks, while humans ensure compliance, interpretability, and domain-specific accuracy. That means you need to think about these directions in this side. Pharmacy, these are all technologies you need to do this. This is different, this one. Any questions you can ask here itself. These are all the things is required: regulatory compliance awareness, FDA, documentation discipline, audit readiness, and pharmacovigilance fundamentals. Of course, programming side also required, etc. Everything, and also communication, soft skills. These are all important. These are all professional corporate behavior is important. Root cause of analysis, evidence-based decision making. Everything is required for enter into corporate side. Always think about this slide: learning to leading, like student to professional. This slide is very important. I like this slide very much because student mindset, professional mindset, apply knowledge to solve the problems. Take initiative and ownership, build real world project, seek impact and growth. Student mindset, professional mindset. Change your mindset to professional mindset. 4 days back I came here. Somebody is working here. After today I came here 3 days. The big conference, everything is excellent because of the professional mindset. You must understand the professional mindset, your principal. This whole credit goes to him. Same thing like this. I am going to stop. If somebody is coughing, means you need to stop. Again, I am telling you this slide. 1 to the power of 365 is 1. I am a mathematics guy. That's the reason. 1.01 to the power of 365 is 37.7. Coming to the college, learn something every day. Simply you can sit. I am asking a simple thing. I bought 2 good books, frankly speaking. That's here itself. I am asking a simple question. Somebody is getting the answer. I will ask your principal to gift. But nobody is turned up. Even though now I am giving the offer also. Give me one simple definition, your area. Whatever the one simple definition, please come here. Sir, this is the definition. Grab the book. Okay. So, doing nothing at all versus small consistent efforts. That gives lot of success. Same thing like this. These are all the important part. Ask is very important. Thank you. Thanks very much. Oh, that girl is cropping, right? Who is that girl? I think. Yeah, please come. I will give one good book to you. Okay. Yeah, please come. Make it fast. It is. Come upstairs. Sir, now please. Tell me one definition.
So I am telling about the chromatography definition. So the chromatography is the laboratory technique which is used to separate the mixture of compounds into individual compounds with the stationary phase and mobile phase.
Perfect. Excellent. Okay. Hey, principal, where are you? Ready, sir? Which book you like it? 7 Habits of Highly Effective People or Atomic Habits?
7 habits.
Okay. Sir, please come. Boys, one book for you. Sir, please come. Prepare a small presentation with the 7 habits and send it to me, send it to a principal and Hindu college principal also. We are going to share that information to all pharmacy students. Okay? Got it? Good. Thank you. Boys, last chance. No. Okay. Oh, you want to come? Yeah, please come. Take a mic. Give one definition.
Okay, I'd like to tell the definition of adverse drug reaction. Adverse drug reaction is an unwanted, noxious, unintended reaction occurs at a normal doses which is used for therapeutic, diagnostic and clinical purpose.
Perfect. Excellent. Sir, please come. The book you want. Oops. Masharan. Read that book. Okay? Oh, you want to take it? Oh. Thank you, sir. Thanks very much. Oh. Good, sir.
So while thanking Dr. Venkatikurthi Garu, a data scientist, for his wonderful session on importance of artificial intelligence in drug discovery and development in pharmacy and biotechnology, I request any questions. The session is open for the audience.
Somebody asking.
Yeah, please.
Please come.
Please come. It's wonderful.
Sir, Michael.
Good morning, sir. Good morning, sir. My name is Sai Rajesh. I am a faculty of pharmacy. In the first few slides, you have shown that the traditional drug discovery takes 12 to 15 years. AI-driven will take 2 to 6 years. Do we have any one molecule which came into the market with the help of AI, or it's just a prediction?
No. So many molecules are here already in the market. Because they are conducting the trials. Probably it is going to get approval, new drug application by end of this year.
So they are just into trials but not approved. No drug has crossed phase 3. It's just a prediction.
Yep, absolutely.
I'm not being cynic, but my concern is when I was doing my graduation, people were very much aware about computer-aided drug designing. But it has gone up, again it went down. No molecule came to the market because of that. So my AI is also, people are talking a lot about AI, but I'm really skeptical how far it is going to get a drug into the market.
Corporates, they want to reduce their cost. For that case, simulation. Previously, we are recruiting hundreds of patients. Now, using with the AI simulation, that is well proven. Monte Carlo methods, simulation methods is well proven. Results are 100% accurate. So, that saves a lot of their time. That's the reason finally I made it the word hybrid. Hybrid means AI is not going to replace humans. But humans, they need to change themselves. They want to adopt new technologies. They want to adopt new skills. Then only they are continuously working with AI technologies. That's called as word is hybrid. 100% hybrid is going to be effective in future, but still people think about AI is going to replace it. I am telling you the word call centers is gone. AI is going to take it. Doctors 20%, pharmacists probably 20%, lawyers 50%, teachers 30%. These jobs are taken by AI. AI. So that is the reason we need to change ourselves. Yes, absolutely, you're right. It's a great question, but yes, but timelines are going to reduce for sure. That's the reason the cost is going to be reduced. That's it. Thank you.
Any more questions?
Oh, good.
Good morning, sir. Does this AI affect employment, sir?
AI?
Affect employment?
AI is going to affect the employment. Almost like 100 million jobs are gone across the globe for sure. But at the same time, 190 million jobs are coming in new areas. So that is the reason some jobs are gone for sure. But you need to know about like a prompt engineering, statistical programming, statistics, molecule design, something like that area. There are plenty of jobs are there. 300 million jobs are gone. New 190 million jobs are coming up according to World Health Organization reports and all those reports. New jobs are coming in for next 5 years. Don't worry about it. AI is not going to replace humans.
Anyone?
Only boys? Girls? No questions? Okay.
Thank you so much, sir. With this, we ended this plenary session. Now I request our principal and our organizing secretary to please felicitate the chairperson and also the speaker. Firstly, I would request our speaker, Venkat sir, to please accept the felicitation.
Hello.
Next, I request Nagabhushnam sir to please accept the felicitation. And we once again thank him for accepting our invitation, for being the chairperson for this session. Thank you, sir. With this, we finished plenary session number 6, and within very short time, we'll be starting plenary session 7. And for plenary session number 7, I would like to call upon two chairpersons. At the outset, I would like to call upon Dr. P. Ravishankar, currently working as professor and head of the department at Vignan Pharmacy College. I request him to come onto the dais. Dr. P. Ravishankar is currently serving as professor and head of the department at Pharmaceutical Analysis and Chemistry at Vignan Pharmacy College. He has a total teaching experience of 32 years. He's completed his Masters of Pharmacy from VL College of Pharmacy, Gulbarga, Karnataka. And he is proudly the second place at the university during his master's degree. Dr. Ravishankar has an impressive publication record with 195 research papers published in both international and national journals. He has a H index of 19 and an I10 index of 35 and a total of 1670 citations to his name. Additionally, he has presented 30 posters at national and international conferences and attended more than 150 of them. Dr. Ravishankar has delivered 35 guest lectures at various colleges of South India and has book chapters read to his name. With this brief introduction, I welcome her. And once again, on behalf of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences, thank you for accepting our invitation, being the chairperson. Please see. Next, I would like to invite the second chairperson, Dr. P. Bhargava Bhushan Rao, who is currently working as professor and principal, A.M. Reddy College of Pharmacy, Narsapet. Sir has Master's of Pharmacy and PhD degree in Pharmaceutics with a total teaching experience of 18 years. He has a total of 75 research publications with a H10 index of 10. He has totally written 3 book chapters and also authored 3 textbooks. He has 7 patents published to his name and he has achieved more than 20 lakh grants from AICTE and also ICSR for carrying out different research projects. Sir, thank you for accepting our invitation and being the chairperson for this session. A small note to the delegates who are attending this plenary session. This is an online session where the speaker is presenting from Malaysia, and I respect our chairperson to please introduce the speaker to the audience.
Thank you. Thank you for giving me the opportunity to act as a chairperson. The management of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences and my teacher and the principal of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences, Dr. Vidyadhara Garu. So, today's speaker, Dr. Shajia Qasim Jamshed, is an associate professor, Department of Pharmacy Practice, School of Pharmacy, IMU, and formerly it was International Medical University, Malaysia. She earned her PhD on the contemporary issues surrounding generic medicine using among healthcare professionals and future practitioners. Prior, she worked in UNISA as associate professor, pharmacy practice, and assistant professor in IIUM, Malaysia. Dr. Shajia primary research interest is on generic medicine utilization, pharmacy education, and practice-based research. Over the past 10 years, she worked on the issues associated with care prospects in oral health, Alzheimer's disease, alternative therapies, self-medication practices, and antibiotic resistance. In terms of scholarship, Dr. Shajia has published extensively in peer-reviewed journals, periodicals, and newspapers. She is on the editorial board of many international journals like BMC Health Services Research, BMC Public Health, and Nature Scientific Reports. She shared her research work by participation in more than 20 conferences around the globe. She authored many book chapters and encyclopedia chapters of especially Elsevier and Springer. She won the travel grant for International Social Pharmacy Conference, Auckland 2008 and Portugal in the year 2010. She earned the prestigious Pharmaceutical Policy Analysis course, Travel Grant, Summer School in the year 2014. She has received more than 20 international research grants to conduct research in different areas of pharmacy education. So she supervised many PhD, masters, and undergraduate students. Thank you.
The presentation displaying?
Yes, ma'am.
Fine. Thank you very much. I would like to thank all of you to invite me for this conference as a plenary speaker. And I would like to thank the conference organizer, the Institute of Pharmaceutical Sciences, as well as my very dear colleague, Dr. Vasu, who connected for this conference. Thank you very much to all of you. So, coming to the plenary lecture today, I'm opening it like that. So, I think it's more clearer. Sorry. Is it more clearer now?
Yes, ma'am.
Okay, thank you. So, the topic of today's plenary is about the AI in pharmacy practice, changing landscape for medication management and patient care. And when we are talking about the today's presentation agenda, so it is on the introduction to AI and healthcare and vision of WHO perspective, whereas historical context of AI and pharmacy automation also, as well as the current AI applications which we are using in medication management, how we are using AI in patient care and adherence, and there are different AI tools and platforms which we are applying in pharmacy practice, and we are going to have a brief outline on the impact on the pharmacist's role and decision making also. And the last part of the slides will be about the ethical and the privacy perspective and the regulatory issues from the WHO guidance, and a couple of the, you can say, the case studies and the real world examples from telepharmacy or robotics, and benefits and limitations, as well as future directions and recommendations and takeaway home points. Now, when we are coming to this slide, what is AI in healthcare? When we are talking about the AI in healthcare, what is AI in healthcare? We come across this terminology many times. So, artificial intelligence or AI is recently emerged as a cornerstone of digital transformation in the healthcare system all around the world. And it's going to have a large range of computational techniques, and which is going to help us to utilize the machines to mimic the human cognitive functions of performances, could be learning, could be perception, could be decision making also. And if we can see in recent years, AI has changed or transitioned itself from experimental research to real world healthcare applications, like we are using it in diagnostics, disease prediction, sometimes in the drug discovery also, and definitely the clinical decision support also. When we talk about the perspectives of the World Health Organization, we need to remember one thing, that from the growing burden of chronic diseases which is happening, and the aging population which is also we are like struggling with, the polypharmacy, the healthcare workforce shortages, these are all the components which intensify the need for the intelligence system, and that can augment our human expertise also. In terms of that, the World Healthcare Organization recognizes AI itself as a critical or fundamental enabler of universal health coverage. But it should be developed, it should be deployed ethically, transparently, and equitably. That is one of the important core. So we can say that AI offers the ability to estimate the large chunk of volumes of structural and unstructured health data. We are supporting clinicians in making the timely and accurate decisions. Now coming to the point of why AI is important in pharmacy practice, or what is the role of pharmacy in AI, or how data explosion and AI is supporting the data explosion also. Now, if we can see the strategic importance of AI in pharmacy practice, so pharmacy practice always occupies a unique position at the section of medication safety or patient education, or either it is clinical decision making. So as a pharmacist, we any not from pharmacy, we could be from pharmacy like technology also, we could be clinical pharmacy also, we could be from pharmaceutical chemistry also. So as pharmacists, we are considered responsible for analyzing the prescriptions, for ensuring the appropriate drug selection also, as well as we need to monitor the therapeutic outcomes. So we can prevent adverse drug reactions, and we definitely need to promote medication adherence in our patients also. So these responsibilities, all these responsibilities invite the continuous processing of large amount of patient-specific and drug-related data. So that's why we are sometimes struggling with the medication errors also, and which is one of the leading causes of preventable patient harm throughout the world also. If you go to the literature for the medication errors, it frequently arises from the complex regimens, or either incomplete information, or either from the prescribing cascade. Either it's related to time pressure, or it's sometimes related to our cognitive overload also. So AI definitely has the potential, has the ability to systematically or strategically address all these challenges, because it's going to provide the consistent, the continuous, the data-driven decision support system. So many times when I go to the conferences also, and when I present to the conferences, people say to us, and like recently graduated pharmacists, they are sometimes also scared of that do AI replace pharmacists? No. AI is not going to replace us. It's going to strengthen our ability, enhance our ability to deliver, to give safe, effective, and patient-centered care by reducing the routine workload of ours, and definitely it's going to support our clinical judgment by giving high-level information or high-level importance also. So we can say that inarguably, artificial intelligence is definitely revolutionized our healthcare, our pharmacy practice, and by that way we becomes more effective, more efficient, and as a pharmacist, we are not feeling left out. So always remember, as a pharmacist, we need to acclimatize ourselves with the AI tools. So we are not going to be kicked out by AI, but we must become a part of the AI, or AI could be incorporated into our everyday professional activities. If you see the like previous literature, so we can see that there is a growing interest in the uses of AI technology, and it has been identified for many years, and is going to analyze and interpret some important fields of pharmacy also, not only pharmacy practice, drug discovery also. You can see it in dosage form designing also, polypharmacy also, and hospital pharmacy also. Now coming to the next slide, if we have just a brief outline about the historical context of AI in pharmacy. When we are talking about the historical evolution, or either the key milestones, so you can see from the foundation, from 1950s to 1970s, the concept of AI was introduced, and during that time, the early work was focused on rule-based systems or symbolic systems. But what happens in the early 1970s, mycin was developed. and which was considered as a pioneer for clinical decision support system. And it was helping that time to diagnose the infection and recommending the antibiotics. MySyn, for your information, it was the pioneer artificial intelligence program, and it was developed at one of the US universities. I remember it's Stanford University in the early mid 70s, and it's going to help the physician that time how to diagnose, how to recommend the treatment for infectious diseases and for the specific cases of bacteremia, as far as I remember. So we can say that it is the granddaddy of expert system by AI pioneer, that is Ellen Newell at that time, who represented MySyn as a significant leap forward in the clinical decision support system. In the 1980s and 90s, we have expert systems data. 1980 saw the emergence of expert system, which can copy or which can mimic the human decision making. And by 1990s, the digitalization of all the healthcare records started to begin, and which allows us the first major concentration of data for analysis. From 2000 to 2010 era, it's about the machine learning and the genomics one. So we can say that the completion of Human Genomic Project in 2003, it's going to speed up the use of AI in personalizing the medicine also. And that is why we saw significant improvement in 2010 in machine learning and deep learning, which can strengthen or enhance the imaging analysis and the drug discovery also. Till 2009 or till the present time, we consider that 2009 is always a defining year for AI in pharmacy. And what will happen with the continuous rise of AI-driven risk stratification, or you can say the robotic dispensing through automation, or either the predictive analytics in daily workflows. We incorporated all these from 2019 onwards. And if we remember the COVID-19 pandemic, so it's augmented or accelerated the adoption of AI-driven telepharmacy and other modalities, you can say also. Now, coming to the next slide, when we are talking about the AI in the medication management. So here we talk about the machine learning, the deep learning. So just a bit of information about the machine learning. We know we have algorithms that learn from the historical data, and by that way we identify the patterns and we make the predictions. So in pharmacy practice, machine learning is still used to predict the medication non-adherence. And by machine learning, we identify the patients which are at the high risk of hospitalization also, or you can say we can detect the potential drug-drug interactions also. And sometimes we can predict the medication demand also. For deep learning, which is already abbreviated sometimes as DL, so it uses artificial neural networks. And this neural network has multiple layers. So it can process complex data set also. And this DL or deep learning is always used in pharmacogenomics, or we can say image-based pill identification. We can also use deep learning for that purpose. And sometimes when we want to analyze the high-dimensional biological data, we can use that deep learning also. For the natural language processing, which is NLP, we always have computers which needs to understand, which makes us understand and interpret the human languages. In pharmacy practice, NLP is always used to extract clinically vital information, relevant information from different electronic health records or EHRs, prescription notes also. Sometimes we can use discharge summaries also to extract clinically relevant information, and sometimes we use NLP for our patient communication also. So definitely because of this, like you can say, the capability allows AI system to process unstructured data, which was sometimes inaccessible previously to the automated analysis. And one more thing which we come across in pharmacy practice for computer vision also, that is computer vision. It always helps us to interpret the visual inputs. Like if you can see in the pharmacy practice, it's like a verification of medication labels, detection of dispensing errors, or automated inspection of compounding preparations also. So there is a vast historical trajectory of technology adoption in pharmacy, which begins from initial digitalization, automation era, or either the clinical integration with the intelligent system era also. So we can say this progression demonstrates always that pharmacy's adaptive capacity and readiness is always available to integrate advanced technologies in the practice also. Here we are talking about one more thing. If you can see here, the optimized prescribing. So when we are talking about the clinical decision support system or the optimized prescribing, so AI also powered our clinical decision support system, as I said to you, and it assists our pharmacists, it assists our doctors by synthesizing the patient data and by helping through clinical guidelines and the different drug databases also. So we can say these systems can identify inappropriate medication and suggest the dosage adjustment also. AI we all can also use in dispensing also, compounding also, and also in the supply chain management. For example, automated sterile compounding systems, we use AI algorithms to ensure that we have doing precise dosing, aseptic technique, and by that way, we have evidences from different hospital systems which reflects that substantial reduction in dispensing errors, and there is improved operational efficiency being encountered when we adopt the AI-enabled robotics also. Now, if we can come here to the AI for the patient care and the medication adherence also. So, medication therapy management, as I already told you just now, it's a critical service which is aimed for optimizing the therapeutic outcomes, and it strengthens the medication therapy management by stratifying the patients, which is based on the risk, identifying the polypharmacy also, and also predict the adverse outcome. AI we are also using in pharmacovigilance and adverse drug reaction also, and also the traditional pharmacovigilance system rely on spontaneous reporting. Now, at that moment, what will happen when there is no AI? It is limited by underreporting and delays. So AI-based systems always continuously estimate the EHRs and prescription data also. So when we are talking about patient counseling also or engagement and adherence, so we can say the AI-powered digital assistants like chatbots we have, they provide medication education, they provide adherence reminders, and they provided our lifestyle advice also. So in a way, these tools or these chatbots are for improving the patient engagement, especially in the chronic disease management. The results are and the outcome are very fruitful. So we need to ensure ourselves that as a pharmacist, we should involve ourselves in complex clinical decisions also. We have smart devices also, wearable technologies also, which generate adherence data, and that AI system also can analyze to identify the patient at the risk of non-compliance also. Now, if you can see here, next slide could be on AI tools and platforms in pharmacy. When we are talking about AI tools and platforms in pharmacy, so I just don't want to go into that much detail. I already told you about the large natural language models also, but artificial intelligence is transforming the pharmacy industry also, because it's going to incorporate the automating routine tasks and not only enhancing the clinical decision making, but definitely improve the patient outcome through personalized care. So wherever when we are talking about AI applications and tools in pharmacy, we are going to talk about the CDSS, that is Clinical Decision Support System, which as a clinical pharmacist, we are being helped by that support system because we can review the prescriptions and analyze the data through EHRs. When it comes to automated dispensing and robotics, so robotics are always used to pick, robotics are always selected to package, always selected to dispense or medications also with high accuracy, and is going to reduce the errors almost to zero in some of the hospitals also. In terms of inventory management and medication adherence and drug discovery also, we cannot deny that we have AI-powered software in inventory management like Blue Yonder. If you go and Google, you can find out. So it can forecast the demand for medications with up to 95% accuracy. So by that way, stock outs are reduced and overstocking is also minimized. In terms of medication adherence, as I told you earlier in the slide, we have smart technologies also like wearable sensors, like AI-enabled apps also. So we can monitor our patient compliance, and we can also provide as healthcare provider real-time feedback also. We cannot deny its use in drug discovery and development also. So AI like generative adversarial networks like GANs. So they use always to identify the new drug targets and predict the molecular activity and expedite the clinical trials also. We have a lot of prominent AI tools and platforms like Drug GPT also, which helps healthcare professionals to audit the prescription. We have ACEPA also. It automates the clinical and the administrative workflows in pharmacy. We have MedAssist also, which can use as a tool to get evidence-based answers to clinical questions also. And many other like Atomwise, in silico medicine, you can find generative AI also. We can, in the generative AI we have ChatGPT, we have Claude, we have Gemini also, and in the pharmacy management system also like McKesson and Liberty. So these are the systems which are adding AI for sometimes better inventory management and clinical decision support system also. Now, when we are talking about the next slide, so we are talking about the impact on the pharmacy roles. Okay, I understand that we are transformed, and AI is transforming pharmacy from a product-focused and manually and administrative-heavy field to a patient-centric data-driven data, highly collaborative clinical practice. But again, I am emphasizing, AI does not replace pharmacists, but it is going to serve as our helper, as our digital advisor. So it can automate routine tasks. It can provide us valuable insights. So as a professionals in our practice, we are at the top of our licenses. So AI is driving a shift from technical, repetitive tasks to high-value clinical activities. We are going to talk about the automation of routine tasks. For if we have the robotic system, if we have the AI algorithms, so we can handle inventory management, we can handle dispensing and prescription verification with high precision also. When it comes to workflow optimization, so we reduce the administrative burden like prior authorizations also, and we allow the pharmacist more time for direct patient engagement. So when we are shifting to clinical focus, so we are transitioning ourselves from dispensing a medication to managing the patient outcomes. And as we talked about medication therapy management, especially in chronic diseases also. And sometimes we are providing proactive care also, because AI algorithms analyze and estimate EHRs. So they can predict the patient needs also, and they can predict the medication non-adherence before it can happen. Clinical emphasis and new specialties are like pharmacogenomics integration, predictive analysis and safety also, and new specialties like emerging roles like clinical pharmacy, like can be now termed as AI specialist also, or AI trainers also, and digital therapeutics and telepharmacy, because AI enables the remote follow-up of our through supporting digital tools like smart pill bottles also, and we can monitor our patient compliance also. So in a way, we are enhancing the collaboration. In a way, AI is acting as a bridge system between the various stakeholders. It could be either the pharmacist-physician collaboration, it could be the integrated care house, it could be the improved communication also, it could be anything. But definitely, I'm not denying the fact there are challenges lied to it. There are ethical considerations like data privacy and security, algorithm bias, and definitely we are in need of new skills also. Now coming to the next slide, AI-driven decision support system. When we are talking about the AI-driven decision support system, so we are revolutionizing, we are changing, and we are transitioning that how organizations manage data and how they detect errors. That is very important, how they detect the errors and how they are revolutionizing the data also. And we need to understand this phenomena also, that in order to predict the risk, we are transitioning from reactive manual processes to proactive automated or intelligent networks also. And when we are talking about the risk prediction here, so we use predictive analytics to forecast the risk, like predictive maintenance. In manufacturing, AI analyzes the sensor data to predict the equipment failures. Also in financial risk modeling, if you can ask me, machine learning models we have that, like random forest models that can predict the market volatility also, how market is volatile or vulnerable itself also. Apart from that, we have project management also, which identifies the early indicators of budget overruns also. We have decision support and mitigation also, where we have real-time dashboards, where AI interactive interfaces visualize the risk levels and enables the immediate intervention also. So in that also we have key challenges and considerations like data quality, because sometimes we have garbage in, garbage out. That is one of the problems also. So algorithm biases also, because AI sometimes can perpetuate the existing biases also. Now coming to the ethical considerations. We are talking about the ethical considerations so far, but in the detail, ethical considerations of AI always embark on, or always focuses on, or reflects on ensuring our patient safety, or you can say equitable care, while we are navigating from algorithm bias to black box transparency also, or you can say the shift in the pharmacist accountability is also now going to be a challenge for one of us problem. So key ethical areas in this is data privacy, handling vast sensitive patient data set, transparency and black box risks, that is the opacity. The AI algorithm is not transparent, it is opaque. So the opacity of AI algorithms can make it sometimes cumbersome or difficult to handle, to understand or explain or trust the justification behind AI generated recommendations. So these are some of the ethical concerns also. When we are talking about algorithm bias and fairness also, so AI trained on non-representative data. So sometimes it may produce inaccurate results also. So this sometimes lead to unequal treatment outcome or the discrimination also we can see. So adopting always AI requires a framework, and it's going to prioritize the human oversight. It's going to prioritize the patient privacy and fair access to technology-driven care also. When we are coming to the regulatory and the governance issues, so we cannot deny this fact. There are key issues which can include like managing the black box, that is AI opacity also, how to prevent the algorithm bias, and how to validate the continuous learning models, and how to navigate the sometimes fragmented regulations which are there globally. So effective governance or strategic planning of the governance always requires strong oversight and clear accountability. And definitely, as I talked earlier, ethical frameworks for AI-driven clinical decisions also. Now, there are many challenges. When we are talking about challenges, there are many challenges. AI models can evolve. AI models can learn from new data, and it can defy the traditional validation methods also. So it requires new approaches also for continuous monitoring. When it comes to data integrity and privacy also, so we need to ensure data quality and compliance with regulations, and that is vital, that is fundamental, that is important, especially in terms of patient privacy and security and ethical data handling also. In terms of explainability and transparency, AI algorithms always operate as black boxes. So it is difficult to understand their how they reach to the clinical decision, which sometimes undermines our trust and accountability also. So governance and strategic responses, establish the oversight committees, which I can say and which I can recommend. Organizations are doing well, creating well dedicated committees, because they need to monitor how AI is performing, and they need to ensure about the coherence, about the alignment of the safety and the ethical standards. We need to have explainable AI techniques also, that is, we need to implement the methods which needs to make the AI decision making transparent. It should not be opaque, because it is essential for the clinical adoption also. harmonized standards. Also, we need to think of ethical frameworks. As I told you, we need to embed the principles, the regulations, like beneficence when acting in the best interest of the patient, or non-maleficence when doing no harm into the AI development. Pharmacist training, which I come out in later in the slides also. Pharmacist training is also very important because we need to address the need for proper training of pharmacy healthcare professionals, how they understand, how they have to use, and how they have to critically evaluate the AI tools. These challenges sometimes look very prompting, and it requires a shift from passive compliance to proactive compliance, ethical compliance, and continuously managed AI governance also. Now, coming to the case studies, very short information about the case studies. So we have one case study that is about AI enhanced telepharmacy Shanghai. And here they talked about during the COVID-19 pandemic, what will happen that in one of the Shanghai grade hospital, Shanghai grade 3A, they come termed as 3A hospital. It was a specialized hospital, and they implemented an AI-based internet hospital pharmacy because they want to strategize and manage the increased demand, and they utilize AI for the prescription review and intelligent dispensing, you can say, on robotic delivery also. And if you go to the paper, you can find out that the system achieved more than 80% qualified rate for the AI prescription reviews, and more than 80% of the patients opting for the QR code based system and contactless offline self pickup of the medicines also. So they said that they recommend, and they reported that this model improved efficiency and patient convenience during the lockdown also. Another case study about the robotics when improves the safety, and it is about from the Ballad Health I'm talking about. And when we are talking about the Ballad Health, what is a Ballad Health? Actually, Ballad Health is offering a wide range of surgical options to help making the choice for those patients who need surgery. So expert surgical teams are available. It is in the US, and they are providing the services to the rural areas of US also. So they have expert surgical teams and providers, and those will work for the patient and the family to identify the best option, and which is definitely fit into the unique care plan. And Ballad Health, if you can say, is a 21 hospital system adopted advanced robotics because it's going to enhance the patient safety through operational efficiency, and it can easily address the staff shortages also. And the whole system employs robots in various areas like surgical procedures and pharmaceutical services. And they partnered with the one company, Omnicell. So they install robotic technology at their central fill pharmacies. I think it is in Tennessee as far as I remember. And their focus is to increase the medication safety and efficacy. So once come to the robotics improve safety, Ballad Health, I already talked about this. And when we are talking about this Ballad Health, I would like to highlight here the initiative features like the dispensing robots name. They have initiating names were Dolly, Johnny, and June. And they installed this in early 2024. And after that, they later installed robots like Hank, Willie, and Reba for IV compounding, and it happens very lately. That is in, I think, in September 2025. And definitely, this can help the repetitive task easily for the pharmacist, and it fosters patient benefit also. Now, coming to the AI in research and personalized medicine case study. It's a very simple case study which talks about the University of Florida. And in fact, the University of Florida is considered a leader in terms of academics. It's considered a leader in applying artificial intelligence and machine learning for research purposes and for personalized medications. Or it's going to be largely revolved around its partnership with different other companies which have supercomputers. So University of Florida Health, its university's academic health center, they in fact implemented AI to improve the diagnostics, and they can give treatment plans, or you can say tailor treatment modalities, and accelerate the patient discovery also, drug discovery also. And they use the machine learning models to predict this and identify those patients and the high risk of hospitalization. And as I already emphasized earlier, it is a University of Florida initiative also. But we have several studies also which can use unsupervised machine learning to correlate the complex, high dimensioned or unlabeled data also, like unsupervised machine learning models were used to identify the correlation between the low dose statin and the favorable outcome in cerebral hemorrhage patients also. Unsupervised machine learnings, like we can say, once we are talking about the word unsupervised machine learning, so it's always say as a clustering. So clustering is always used to correlate genomic similarity also. When we are talking about the clinical data pathway, so unsupervised learning is used to cluster the patient based on the metabolomic signatures, and it can map them to clinical characteristics also. Now coming to the benefits of AI in pharmacy. When we are talking about the benefits of AI in pharmacy, so there is improved patient and safety care, because I already told you it analyzes patient records and laboratory data, patient history also, because it helps us to predict the adverse outcomes also, and sometimes it puts a red flag on the drug infections also, help us in optimizing the dosage regimens also. Whereas robotic dispensing, when it comes for operational efficiency and automation, it talks about automated labeling and AI-driven chatbots also, which can streamline our workflows also. So by that way, our manual tasks are reduced and the pharmacists are free to focus more on patient care. Personalized medicines also, we have AI which leverages the genetic information of the individual patient data, so it can tailor the personalized treatment plan, which improves the efficacy in that particular individual also. We have predictive inventory management also, because AI algorithms forecast drug demands. So it can prevent shortages, it can reduce the waste also, and it can enhance the supply chain efficiency also. And as I already told you, drug discovery and development, it accelerates machine learning, accelerates the identification of new drug candidates and repurpose the existing medications. But coming to the next slide, I need to highlight one thing. Despite all these benefits, nothing is free of limitations. Nothing is free of bias. Nothing is free of challenges. So we have different challenges also, which we come across here in terms of this slide, limitation and challenges. So we say most of the pharmaceutical companies, I can tell you one thing. I came across one article which was published, I think, in September 2025. And in that, they highlighted that most of the pharmaceutical companies do not like disclosing that their approach is towards using AI. They keep a tight lid on their integration, but several big names in the industry explained how they are using AI. For example, AstraZeneca, they already incorporated in 2021 OncoShot to find suitable candidates for their clinical trial also. So, okay, I'll conclude within a short period of time, no problem. And a Bayer also has partnered to experiment with artificial intelligence. Eli Lilly also announced many AI projects in June 2003 also. Pfizer also is using supercomputers in AI since 2020. Sanofi is also developed AI platform with the help of Eli Labs also. But in some cases, collecting data for training AI models creates biased outcomes. So we have, even with accurate data, representative data, there can be problems, and it reflects inadequacies as well as underlying healthcare system bias also. Now, just one or two slides going, future directions to fully realize AI benefits in pharmacy practice. We need a comprehensive approach. We need a multifaceted approach. And there must be deliberate efforts in policy development where AI continues to advance rapidly. We need to understand there is no comprehensive, up-to-date legislation in place to regulate its use in healthcare. So we need to establish clear guidelines and regulatory frameworks to govern the use of AI in healthcare, which is a essentiality. Now, coming to the next slide, when we are talking about the training and education of pharmacists, core components of AI education for pharmacists will be digital literacy and foundational AI, ethical and regulatory training also, clinical application and interpretation also, analytical skills also, that is the development of skills to interpret the data also, and implementation strategies in our pharmacy curriculum through simulation-based learning, through interdisciplinary training, or through tailored learning paths also. And coming to the last slide, policy and collaboration. So FIP releases a document on the responsible use of AI, and I feel very enlightened to read that policy document. And they predicted that with artificial intelligence, we are predicting to reshape the future of pharmacy. But the responsible use of AI in pharmacy is a must. Now, when we are talking about the key takeaways here, so we have only a few points in that, and we need to understand that there are few things like on the basis of recent analysis, the integration of AI into the pharmaceutical sector is shifting from experimental pilots to core strategic implementation. And with AI-driven value creation projects reaching about more than 400 billion annually, successful adoption requires moving beyond technology to focus on infrastructure also, regulation also, and human-centric integration also. These are the recommendations for the AI use also, and future recommendations. Also, you have an idea, you have to adopt the AI tool with the oversight also, and we need to adopt, automate routine tasks, enhance our clinical decision support system also. And thank you very much for that. Conclusion is about the just one line about the conclusion, that's it's our reshaping our pharmacy practice by enhancing the medication management and patient care. And when you implemented it responsibility, it can improve the safety, efficiency, and personalization, and we are remain central to this transformation. Thank you very much. These are my references. Thank you very much. Thank you very much. And I apologize because my video didn't open. Thank you very much for that.
Okay. Hello, everyone. Thank you so much for all being here for an incredibly warm welcoming. I wholeheartedly thank you from the bottom of my heart to all wonderful students, staff who are here in this very big open seminar hall, right? Okay. Ma'am, we have listened a very insightful information. Dr. Shajia Jamshad ma'am, that's nice, awesome, welcome. That's incredible, right? Shajia has clearly explained about AI in healthcare and AI in pharmacy practice, role of pharmacy, and data explosion. She also emphasized, shared on views on historical content in pharmacy, and she elaborated medication management, training and education for pharmacists. Okay, 1 minute. And she also explained lots of things about case studies and limitation and challenges, benefits of AI, so many things, right? Thank you, ma'am. Thanks a lot for that. I would like to say one thing to you. If you want to find the next drug and discover the next breakthrough and create the next robo and the code the next robo, to improve the, something like healths of, lives of millions of people, you will have to work on for long days, for long days. For that, assiduous research is required, right? And I want to say one more thing. Students, researchers, innovators, leaders, and entrepreneurs and dreamers who never give up, never abandon your aspirations or inclinations, whatever it may be. Yeah, you will have to work on that. You will have to strive for the better days tomorrow, right? And eventually, I want to say one thing. If you attend such a sort of international conferences, you can gain two sorts of things. One, you can uplift your educational knowledge, and also the one of the biggest gift of such a type of conferences is you can make friends, you can make friendships, not only your college, from the peers from the other colleges. That's why I have been doing these things from a long time. My brother Vidyadhara, we are roommates, right? Something like that. So, in fact, this is the place where you can interact or intermingle with each other, and it is the beauty of these conferences. Alone you can say, together we can discuss, alone we can smile. Okay, 1 minute. Together we can discuss, and alone we can smile, and together we can love. Alone we can, enjoy. Together we can celebrate. This is the real celebration of you. Thank you. Thanks a lot.
Next, we are proceeding to plenary session number 8. And to do the chairperson, I would like to invite on stage Professor K. Tejomurthy, who is currently working as Professor cum Principal at University College of Pharmaceutical Sciences, Acharya Nagarjuna University. Sir has 23 years of experience in industry and academia and has total 95 publications to his name. He authored 5 textbook chapters and 2 textbooks, and he has total 10 grants published under his name. So with this short introduction, I welcome you, sir, and once again thank you on behalf of our institute for accepting our invitation and being the chairperson for today's session. And I request you to please introduce the speaker of this session.
Good morning, everyone. It gives me immense pleasure to be a part of this wonderful program, a conference. I would like to thank legendary principal, Professor Dr. Vidyadhar sir, who has given an opportunity to be a chairperson in this session. It gives me an immense pleasure to read out the biodata of Associate Professor Dr. Ui Ying Hong, Head of the Department, Pharmaceutical Chemistry, School of Pharmacy, IMU University, Malaysia. He is an Associate Professor. Dr. Ui Ying Hong is the Head of the Department, Pharmaceutical Chemistry at the School of Pharmacy, IMU University. He obtained his BSc Honours in Science and MSc from the University of Malaya and his PhD in Polymer Science from the University of Akron, OA, USA in 1998. He is a member of the Royal Society of Chemistry, UK, and the American Chemical Society. Dr. Wu's research expertise lies in the design and development of biocompatible and biodegradable polymeric systems for biomedical and pharmaceutical applications, with particular emphasis on advanced drug delivery technologies. His work spans nanotechnology-based delivery systems and sustained and controlled release formulations. In recent years, his research has focused on neosomal drug delivery systems, including neosomes encapsulating polyphenolic molecules. He has successfully secured research funding under the Malaysian Fundamental Research Grant Scheme as well as grants from the Ministry of Science, Technology and Innovation through the eScience Fund. He has authored and co-authored a total of 62 publications and is the inventor of one US patent. Thank you very much, sir, for being here. The stage is yours. Please go ahead, sir. You are welcome, sir.
Thank you, Professor, for your kind introduction. Good afternoon to all distinguished members of the audience. I'm Dr. Ng from IMU University, Kuala Lumpur, Malaysia. Sorry, am I audible to all? Okay. Okay, first and foremost, I would like to express my sincere appreciation to the event organizer for inviting me to deliver this talk. I'm truly honored to have the opportunity to share my knowledge with such an engaged and enthusiastic audience. In this plenary lecture, I will discuss the fundamentals of neosome, highlight recent advances in their formulation and application, and finally, explore future perspectives and remaining challenges in translating neosomal system from a laboratory to the clinic. As I understand that a significant proportion of the audience comprises undergraduate and graduate students and also PhD students, it is my hope that by the end of this talk, the following learning objectives will have been achieved. So, in understanding the concept of neosomes, learning their formulation and characterization, explore the pharmaceutical application, and discuss recent advances and future scopes. Now, conventional drug delivery systems are often limited by poor bioavailability, whereby only a small fraction of the administered dose reaches the systemic circulation or intended site of action. As a result, frequent dosing is required to maintain therapeutically effective drug concentration in the body, which can compromise patient adherence and increase the risk of dosing-related complication. In addition, non-specific drug distribution commonly leads to systemic side effects, as the medication affects not only the target tissue, but also healthy organs throughout the body. Furthermore, the lack of site specificity means that drugs are not precisely directed to a single target or location, often resulting in inconsistent therapeutic outcomes, variable efficacy, and a disconnect between drug and administration and its intended site of action. So over the years, a variety of carrier systems have been explored to address this limitation of conventional drug dosage form, such as polymeric nanoparticles, lipid nanocarrier, inorganic nanocarrier, nanoemulsion, and vesicular nanocarrier. So among these, the vesicular drug delivery system have attracted sustained interest due to their versatility, stability, and cost effectiveness. Now we need a vesicular drug delivery system because they can protect drugs from degradation, especially those drugs that are sensitive to environmental conditions such as body pH, temperature, presence of enzyme, etc. The delivery system can also improve solubility of poorly soluble drugs by being small in size, having high surface area, and come in amorphous state. And also very importantly, the vesicular drug delivery system enable controlled and targeted delivery, and thus reducing the need for frequent dosing. Now, what are neosomes? Neosomes are basically microscopic bilayer vesicles made from non-ionic surfactants and cholesterol that form spherical structure, acting as a nanocarrier for drug delivery, similar to liposome, but using cheaper, safer ingredients. So this biodegradable nanoparticle can encapsulate both water-soluble or hydrophilic drug and fat-soluble or lipophilic drugs, improving their stability, bioavailability, and targeted release, making them useful in pharmaceutical, cosmetic, and agriculture. Shown here is the schematic representative structure of a neosome. And type of non-ionic surfactant used in neosome generation include span, or also known as sorbitol ester, or even twins, also known as polysorbitol. Often, charge inducers are also added into the formulation in order to prevent particle aggregation by way of electronic repulsion, thus enhancing particle stability. These include negatively charged compounds like diacetyl phosphate and phosphatidic acid, and positively charged inducer like stearoamines and stearopyridine chloride. You can see the structure here, spherical in shape, and we have aqueous domain in the core, and this is the bilayer structure over here, whereby the lipophilic drug will be embedded in the bilayer structure, and the hydrophilic drug will be in the aqueous domain here. So the commonly used non-ionic surfactant, for example, under the name of the span, is also known as sorbitol ester, produced by reaction between the sorbitol and fatty acid, span 20, span 40, span 60, and so on. For twin, the polysorbitons such as twin 20, 40, 60, and bridge the polyoxyethylene alkyl ethers. So incorporated in the neosome formulation, this cholesterol is acting as a membrane stabilizer by interacting with non-ionic surfactants to improve the structure integrity of the bilayer. And this is achieved through hydrogen bonding. Okay, hydrogen bonding between the cholesterol hydroxyl group and the ester functional oxygen atoms of the surfactant. These hydrogen bonds increases membrane rigidity, okay, and cohesion, which protect the vesicle from degradation and thus reduces the leakages of an encapsulated drug. Now, various types of neosome can be formed based on their size, structure, and preparation method employed. Sonication will produce a small unilamellar vesicle with size range of 10 to 100 nanometer, and this is good for systemic delivery. The ether injection method will produce large unilamellar vesicle with size range of 100 to 300 nanometers, which can carry more drug to the internal volume. And thin film hydration method result in multi-plane lamellar vesicle having wide size ranges from 0.5 up to 10 micron. What are the advantages of neosome? Now, comparing with liposome, which are made of phosphate lipid, neosome composed of non-ionic surfactants, which are more robust than the phospholipids and more chemically stable and cheaper. They are inherently biocompatible and do not trigger an immune response from body's immune system, ensuring enhanced drug delivery efficiency and safety. And neosome are suitable for various routes of administration, such as oral, topical, transdermal, parenteral, ocular, nasal, and pulmonary. Their design can be tailored to the specific application, making them a flexible, versatile drug delivery system. However, there is a limitation in neosome itself. So, firstly, neosome in suspension can aggregate and eventually fuse into larger particles, especially if they lack sufficient repulsive forces, for example, electrostatic charge or steric injury. This changes the size, distribution, and morphology of the formulation. So changes in the size and morphology of neosome directly affect their stability, drug encapsulation efficiency, release kinetic, and therapeutic efficacy. So secondly, due to particle aggregation, the encapsulated drug can leak out of the vesicle during storage due to changes in the membrane permeability or structural integrity over time, which reduces the efficacy of the formulation. And thirdly, it's difficult to adapt for large-scale production because resulting product often has inconsistent physical and chemical properties. Example, size, stability, and drug loading. So, method of preparation of neosome. One of the most frequently used method is a thin film hydration method. So, in these protocols, the first step involves the preparation of solution of surfactant plus cholesterol, usually in organic solvents such as methanol or sometimes mixed solvent methanol with chloroform. And these solutions will then be subjected to evaporation, solvent evaporation using rotary evaporator and at elevated temperature. And if span 60 is used, the temperature usually is set at around 55 degrees C, which is slightly higher than the gel to liquid phase transition temperature. So by carefully reducing the pressure together with heating in the evaporation, so a thin film will be formed at the bottom of the round flask, which is then followed by hydration with aqueous drug solution and followed by sonication to break down the neosome that is formed into smaller size. And this method is useful for encapsulation of hydrophilic and hydrophobic drugs. And high EE is often obtained through this method. There are many other neosome preparation methods. Ether injection In this method, it involves injecting a surfactant organic solvent mixture through a needle into a warm aqueous phase containing drug to be encapsulated to create small, generally uniform vesicle. The aqueous phase is maintained at a temperature higher than the boiling point of the ether, typically between 55 to 65 degrees C. Large unilamellar vesicle with size range of between 3 to 1,000 nanometers are produced with good encapsulation efficiency. The general step in reverse phase evaporation involves dissolving liquid in an organic solvent like chloroform ether, adding the aqueous drug phase, sonicating to form inverted micelle, and rotary evaporating the solvent to create a gel, and finally agitating the gel to form the neosome, and resulting in high encapsulation efficiency for larger unilaminar molecule. The microfluidization protocol involves loading a sample of emulsion into a high pressure system which processes through tiny microchannels between 75 to 100 micron at extreme velocity, about up to 500 meters per second, using an intensifier pump to produce uniform size small unilamellar vesicle with size less than 200 meters. Now, why preparation method matters? The preparation methods for vesicle drug delivery system is critical because it directly dictates the final physical and functional characteristic of the carrier. Different methods yield varying results regarding the size, lamellarity, number of bilayers, and robustness of the vesicle, which in turn dictate how effectively they hold, protect, and deliver a drug. Now, neosome are vesicular novel drug delivery system, which may be used for the sustained control as well as targeted delivery of medication. So, to ensure their quality, stability, and performance, there's some critical parameters are evaluated. So, these will include vesicle size, zeta potential, entrapment efficiency, and morphology. So, neosome with a smaller size, less than 100 nanometer, are generally better at penetrating tissue, particularly for tumor targeting, as they can navigate through smaller capillaries and get. Larger vesicles are often more suited for localized or ocular delivery to prolong the residence time, while smaller vesicles are better for sustained systemic circulation. In addition, larger multiple lamellar vesicle may be more prone to drug leakage, and reducing their size via sonication can, in some cases, negatively affect encapsulation efficiency, making optimization of the size necessary. A zeta potential value exceeding 30 millivolt, either positive or negative value, is generally required for a stable colloidal system. High surface charge creates strong electrostatic repulsion between adjacent vesicles, preventing aggregation, flocculation, and fusion. Size optimization is essential to balance the encapsulation efficiency with stability. Lastly, the morphology of a neosome is essential, as the TM or SEM, both are electronic microscope, provide direct and high resolution visualization of their surface, morphology, shape, and structure, which are essential for confirming successful formulation, optimizing drug delivery, and ensuring stability. Now, talking about the release mechanism, neosome release drug by few mechanisms. First, through diffusion control release. Through bilayer, drug partition into the hydrophobic core or hydrophilic shell of the neosome and slowly diffused out through the surfactants layer over time. And through aqueous core, hydrophilic drugs in the aqueous center can leak out as the vesicle interact with the body. Now, neosome, which are non-ionic surfactant-based vesicular carrier, disrupt the tightly packed, rigid, and highly ordered liquid bilayer structure of stratum corneum, primarily through their surfactant component, which act as penetration enhancer. They reduce the barrier function of the skin by fluidizing the intercellular lipids and hydrating the skin, allowing drug molecule to pass through the more easily. And control and sustained delivery mechanism involve manipulating the vesicle structural component, primarily non-ionic surfactant and cholesterol, to create a rigid, stable barrier that slows down drug leakage while allowing for gradual, sustained release over an extended period. Now, rules of administration. So, neosome drugs can be administered through multiple routes, including oral, parenteral, such as injection like intravenous, intramuscular, or subcutaneous, topical or transdermal, ocular, and respiratory, pulmonary by inhalation, offering flexibility to overcome drug instability or improve targeting for various therapies from cosmetic to cancer treatment. Now, here are a few examples of a drug investigated, which involves the use of neosome. For example, this anti-cancer agent, doxorubicin, it was found to have improved cytotoxicity in a breast cancer model. And the natural product, curcumins, show greater anti-tumor activity in colon cancer. Another anti-cancer drug, Felicitation, together with oxaliplatin, demonstrated effective delivery for ovarian and colorectal cancer. And also, letrozole, metformin, celecoxib show potential for hormone-dependent breast cancer treatment, and artemisinin and derivatives show enhanced anti-cancer effect in lung cancer, liver cancer, etc. So these are just a few examples to show how the neosomes are useful and for cancer cell, disease treatment. Recent advances show that more have been done to have targeting capability of the neosome to specific disease or cancer cell. For example, using targeted neosome as advanced drug delivery system using non-ionic surfactant vesicle to encapsulate drug, guiding them specifically to disease site like tumors for enhanced efficacy and reduced side effect. And this is achieved by attaching targeting ligands like folic acid or higher hyaluronic acid to their surface or it can also involve incorporation of magnetic materials for external guidance. Now, stimuli responsive neosomes are smart nano carrier that change their property, like releasing drug in response to specific internal condition, pH, enzyme, temperature, or external involving light, magnetic field, ultrasound triggers, enabling precise on-demand drug delivery to targeted area, improving treatment efficacy and reducing side effects, especially in cancer therapy. The essential size range of neosome for effective cancer cell targeting and induction of pulses is generally between 100 to 300 nanometer, with many optimized formulations focusing on a smaller 100 to 200 nanometer range. This size range is critical because it enables accumulation in tumor tissue via the enhanced permeability and retention EPR effect, while avoiding rapid renal clearance or excessive uptake by the mononuclear phagocytic system. So this is the diagram to show how neosomes formulation that have folic acid as the ligand is delivered into the cancer cell, okay, through endocytosis and coupled with the release of the drug in the cell, and this the cancer cell is undergoing apoptosis. However, there are challenges in clinical translation. Okay, neosomes are non-ionic surfactant-based vesicles offer significant advantage for drug delivery, such as higher stability, lower cost, and lower toxicity compared to liposome. However, their clinical translation is hindered by several key challenges, ranging from manufacturing complexity to regulatory hurdles. Many laboratory scale techniques are labor intensive, time consuming, and hard to scale up to commercial production level. Neosomes are susceptible to aggregation, fusion, and leakage of the encapsulated drug during storage. So the stability during storage is crucial, affected by formulation, by cholesterol, charged molecule improve it, and storage condition. Usually, refrigeration at 4 degrees C is generally best, but freezing or lyophilization can also work. There's a lack of a standardized testing protocol for evaluating neosome quality, safety, and long-term stability. So, demonstrating consistent quality and safety to a regulatory bodies require extensive data and adherence to guidelines, which are still evolving for new modalities. Now, a future perspective. In the area of personalized medicine, often referred to as a precision medicine, is the current focus of study because it allows healthcare provider to tailor treatment to a patient's unique genetic makeup, environment, and lifestyle, moving away from the effective one-size-fits-all approaches. Advanced nano carrier like neosome have become prime candidates for personalized medicine due to their high structural versatility, enabling them to be customized to meet individual patients' needs, specifically in cancer therapy, gene delivery, and dermatology. Their ability to encapsulate both hydrophilic and lipophilic compound, coupled with their superior stability and lower cost compared to liposome, allow for the development of targeted patient-specific therapy that minimize side effects and improve therapeutic outcome. To conclude, three key points emerge from today's discussion. Neosomes are considered a highly versatile drug delivery system because recent advances in formulation strategies, surface modification, and targeting approaches have greatly expanded their functional capability and therapeutic application. The niasomal formulation work demonstrate a strong link to pharmaceutical principle that is firmly grounded in core pharmaceutical knowledge that students are expected to learn and apply, rather than being broadly related to pharmacy in general. In addition, it shows significant clinical and research potential as the findings go beyond statistical relevance to provide practical and actionable insight that may translate into improved health outcome and enhanced quality of life. While Neosomes show great promise, challenges related to large-scale production, reproducibility, regulatory acceptance, and clinical translation must still be addressed. So this concludes my presentations. Thank you for your attention, and I'll be happy to take any question.
Any questions from the audience?
Thank you so much. Thank you so much for the session, sir. And I would like to ask you a small question. Am I audible? Yep. One thing that I would like to know is the formulations of neosomes. Of course, the topic of neosomes is very naive to me, but what I would like to know is, in case of drugs that have this property of showing systemic toxicity, either due to narrow therapeutic index or due to some renal or hepatic failure, so can these drugs which show systemic toxicity can be formulated via neosomes in order to reduce the toxicity? Is there anything like that?
The answer for this question is yes. So the systemic toxicity is often related to instability of the neosome, any nanoparticles. So if we can improve the stability of the formulation, the nanoparticles, so we can avoid the systemic toxicity. And in neosome formulations, we can attach a ligand for targeted delivery. In addition, this cholesterol play a very important role in improving its stability. So improving the neosome's rigidity, which can prevent drug from leaking out. So that will minimize the systemic so-called toxicity.
Does the presence of cholesterol in the formulation affect any absorption parameters of these neosomes across the gastrointestinal tract?
Sorry?
The cholesterol lining that we are using in the manufacturing of neosomes or the formulation of neosomes, does it alter the absorption property of the drug across the GIT?
The cholesterol molecules is actually embedded in the bilayers of the neosome. So it would not affect the crossing of or absorption of these neosome particles.
Okay, sir. Okay. Thank you. Thank you for the information.
Thank you.
Thank you very much, sir. Dr. Roy. Sir was explained about few things which are very essential in the drug delivery systems, neosomes, plays a pivotal role in drug delivery system, and they also explained about the recent advances of neosomes and its challenges in the clinical translation. Thank you very much, sir, for your wonderful presentation. Keep going, sir. Thank you.
Thank you. Bye-bye.
I request you, sir, to please stay online.
Okay.
I request you to please stay online. We would like to felicitate you for your participation today here.
Thank you.
Now I request our principal and organizing secretary to please come on to the dais. And I also request Dr. Vasudeva Rao to please come on to the dais and accept the felicitation on behalf of the speaker. Sir, can you please turn on your video? We would like to have you during this session. Now I request Vasudeva Rao sir to please come forward and accept the felicitation on behalf of our speaker.
Sir, good afternoon, sir. Good afternoon, sir.
I request our speaker to please turn on his video. If the speaker is still available with us, I request him to please turn on the video. So thank you for the session, sir. And Dr. Vasudeva Rao is accepting your memento on behalf of you.
Thank you very much.
Now I request Vasudeva Rao sir to please join our principal and organizing secretary as we felicitate the speaker of this session, chairperson of this session, Tejomurthy sir. I request our principal and organizing secretary to please stay on the dais. Due to short time, we did not felicitate the chairpersons of the previous session, so we would like to do that. And I please request Professor P. Ravishankar and Dr. Bhargav Bhushan Rao to please come on to the dais. We are sorry that we could not felicitate you in the lapse of time. Now I request Dr. P. Ravishankar to accept the felicitation. Now I request Dr. Bhargav Bhushan Rao to please come forward and accept the felicitation. Now we are about to start the last plenary session before lunch, plenary session 9 of this two-day international conference on drug discovery. And to act as chairperson for this plenary session 9, I would like to invite the chairpersons onto the dais. The first chairperson is Dr. Sheikh Abdul Rahman, who is currently working as professor at Vignan deemed to be University, Guntur. I request him onto the dais. Sir has 23 years of teaching experience and research experience. He had more than 130 articles published under his name, for which he had a total of 1245 citations with an I-10 index of 36 and a H index of 17. He authored 7 books and wrote 27 book chapters. He has 26 patents published under his name, and he received multiple awards, which include Best Researcher Award given by APTA Andhra Pradesh, Best Teacher Award, Global Researcher Award, and Academic Excellence Award, etc. With this brief introduction, I welcome you, sir, and thank you for accepting our invitation and being the chairperson for this session. Next, I would like to invite on stage Dr. C.H. Jitendra, who is currently working as head of the department at Pharmaceutical Sciences, Vignan University, onto the dais. Sir has a PhD in neuropharmacology with a total teaching experience of 20 years. He is an associate fellow of AP Academy of Sciences and has received Best Researcher Award from Vignan University. He has a total publications of 52, and he has 3 book chapters written to his name and has 8 patents published in his name. I also welcome him and thank him for accepting our invitation for being the chairperson for this session. Now I request Abdul Rahman sir to please introduce the speaker to the audience.
So very good afternoon to all. So today is 9th session, scientific session in the second day of international conference organizing by Chebrolu Hanumaiah Institute of Pharmaceutical Sciences, Guntur, Andhra Pradesh. I am very much delighted to introduce the today's 9th session speaker, Professor Dr. Lee Choi Sin, Associate Dean Research and Consultancy, School of Pharmacy, IMU University, Malaysia. She is an accomplished academician and researcher in the field of green chemistry, biomaterials, and oleochemistry with a notable track record in research and innovation. She is currently serving as Associate Professor in the Department of Pharmaceutical Chemistry and Associate Dean Research Consultancy for the School of Pharmacy at IMU University, Malaysia. Her research areas focused on the development of biocompatibility and biodegradable smart polymers from renewable resources, particularly palm oil for pharmaceuticals, biomedical and industrial applications. Dr. Lee has secured a total of 1.8 million research and consultancy grants from various government and non-government agencies. Right? Then her scientific contributions and innovations have resulted in 3 patent filings in Malaysia and across Asian countries. Then she has secured gold medals in the year 2024-25, silver medals 17, 2019 and 23 at International Invention, Innovation and Technology Exhibition. She has supervised 25 postgraduate students. She has also published high-impact research and review articles in various indexed journals. She has invited as a speaker to deliver the sessions on sustainable polymer chemistry, green drug delivery systems, oils and fats innovation. Right? So she is the recipient of Young Scientist Award for outstanding work in oils and fats research. She is also serving as the council member of MOSTA and chief editor of the Malaysian Oil Science and Technology. With this brief introduction, I would like to welcome Professor Lee Choi Sin, Associate Professor and Associate Dean Research from IMU University. So now I am Madam to deliver the scientific session on this today's session number 9. Welcome you, ma'am.
Thank you very much for the chairpersons for very kind introductions. And also thank you to the organizer for the opportunity for me to share some of our works on the bio-based polymeric excipients that we have produced from renewable feedstocks for drug delivery as well as medical and biomedical applications. So as what the chairperson said, he has very kindly to introduce that you can see that a lot of my work is related to oil and fats and as well as our Malaysia golden crop, palm oil. Okay, so just a little bit about our universities, IMU universities. So IMU universities is the first private medical universities in Malaysia and established since 1992. So thank you so much for the organizers to have us here for me virtually, and also I have some colleagues, and also Dr. Wasu, be able to actually present to your conference. So this is our universities, some of the histories as well as our new branding in 2024, okay, whereby we used to known as International Medical Universities, and in 2024 we have new branding and logo, and we are now known as IMU University. Okay, so now let me straight to the topics for today's on the medical polymers. So the demand of the polymers, the macromolecules, is enormous, especially in healthcare. So as you can see from the Grandview research, the medical polymer market is expected to grow with a CAGR of 8% to reach USD 37 billion by 2030. And this growth is largely driven by the pharmaceutical and healthcare sector. And within the medical polymer markets, you can see that the most demanded segment will be on the fiber and resin, such as using it as sutures, surgical meshes, followed by the medical elastomer and medical packaging, as well as the biodegradable polymer. So on the other hand, there's also growing demand in the pharmaceutical excipients, and the global markets of the pharma excipients is estimated to be reaching USD 10 in 2023, already reached 2023 USD 10, and then in 2028 it will be reaching 14 billion. So for the Asia Pacific pharmaceutical excipient markets, it's worth USD almost 2 billion in 2021, and then the growth is going up, whereby all this is due to the prevalence of the chronic disease, geriatric populations, and the demand of the generic drugs. As a matter of fact, the global polymer solubility enhancer excipients, there's a huge demand there, okay, due to the poorly water-soluble pharmaceuticals that represent 40% of the top 200 marketed oral drugs, and as well as 75% of the compounds under development, they are all poorly water-solubles, and a lot of new chemical entities, they are poorly water-soluble. And what are the applications of the polymers in the medical and biomedical applications? So the polymers, they are heavily involved in disposable supplies such as surgical gloves, syringes, injection pipes, and catheters. So these actually all contributed by the polymers. And it's also very much important in medical implants. So as you can see over here, so polymers, the materials that can fulfill the mechanicals, chemicals, as well as the biocompatibilities material will be used as an implant, and as well as medical devices, diagnostic biosensors, wound healing dressing, and tissue engineering. So all this we need to cater by using biocompatible as well as the polymers that is having sufficient mechanical properties and do not release out toxic material. So for drug delivery, polymers also play a very important roles in drug delivery, such as in various dosage form, for example, tablet dosage form, as carrier, diluent, filler, binder, disintegrant. And for capsule, it's being used as the material for the capsule shells. Suspension is being also, we use a lot of different excipient in the suspension. Transdermal patches. Transdermal patches, there's a lot of products for ADHD, depressive disorder, Parkinson, and dementias. They're already in the markets whereby we make use of the polymers to deliver the drugs, and as well as for some of the medical devices, for example, the IUD for hormone releasing devices, it's also made of the polymer coatings as well as the material, and also for the polymeric nanoparticles to use the polymers as the carrier to deliver the drug. So a little bit about the polymers. Polymers, they are classified into natural polymers as well as the synthetic polymers. So for the synthetic polymers, mainly they are produced by petrochemicals. And from these petrochemicals, they classify into thermoplastics as well as thermosets. So thermoplastics is the one that able to be molded and then and prepare it into different form, and you can melt it again and to prepare it into a different form again. For thermosets, you will not be able to recycle the polymers. And in terms of natural polymer referring to the macromolecules where it's originated from living organisms, such as natural resources from the wood, leather, and plants, such as rubber, silk, wool, cellulose, and proteins. And just now when I mentioned about synthetic polymer, so I mentioned about the petrochemicals that produced from the fossil fuel. And now we're also looking into the synthetic polymers that can be produced from renewable feedstock, such as the fermentation product. Example is polylactic acid, PLA, and as well as the polymers that can produce from plant oil. And my research interest is always on vegetable oil, our Malaysia golden crop palm oil, to produce different chemicals. And these chemicals is known as the oleochemicals. And by modifying the chemical structures of the oleochemicals, we're able to produce it into different polymers, such as the polyolefins, polyester, polyethers, and polyamides. Okay, majority of the synthetic polymers, as mentioned, they are derived from petrochemicals, and they are known for all these advantages. Their cost effectiveness, because can be produced abundantly, and reproducibility with the desired physical chemical properties, desirable mechanical properties, desirable thermal properties, and they are known to be stable. And on the other hand, for the natural products, natural polymers, polymer that produce from plant oils or fermented products, they are, because of the origin, so they are more biocompatible and they are more biodegradable. But the natural polymers always having a limitations in terms of its mechanical properties, physical chemical properties, is not as good as the synthetic polymer. And our research interest, and we think of like, can we leverage the synthetic polymers to have all these advantages, and at the same time we want it to be biocompatible and biodegradable for the environment as well as the health and safety concern. So thus we think of to bridge the advantage of both synthetic polymer that used to be produced from petrochemicals, as well as to have the advantage of the natural polymer to have the biocompatibility and biodegradability. So we work on producing synthetic polymer from the renewable feedstock and making use of the plant oil, vegetable oil, for example, palm oil. Be able to actually produce the polymers that we can modify its chemical structures, improve the mechanical properties, desirable thermal properties, physical chemical properties, and because of its feedstock origin is from the plants, so it's usually less toxic and also biodegradable. So in our research team, we focus on two main SDG, on the SDG 3, good health and well-being, as well as the SDG 12 in promoting responsible productions. So we try to achieve this by developing polymers and biomaterials for pharmaceuticals and also medical application using renewable feedstocks instead of petrochemicals. So in our chemical processes, we also prioritize the green chemistry principle to guide us in minimizing environmental impact and maximizing the sustainability. For instance, we will try to apply all these approaches, green chemistry principle, 2, atom economy, 3, less hazardous synthesis, 5, benign solvents, and then design for enhanced energy efficiency, use of renewable feedstock, incorporations of catalysts and also to produce the polymer as well as the material that meant for biodegradable. Okay, so so let me also introduce you a little bit about different type of angle from our vegetable oil. So as you know that Malaysia is the second largest producers of the palm oil. Indonesia is number 1, then number 2. So there are different range of the palm oil products. So in terms of palm oil, of course, we use it a lot for food. Okay, so it actually guaranteed us for food safety. Okay, and as well as in terms of the downstream products, we have the oil chemicals whereby it's can be produced into a wide range of applications. And also we look into a lot. So one of the things that we are looking into is the used cooking oil. So in Malaysia, mostly the cooking oil being used is palm oil. And of course, the eateries, they are using the palm oil. So actually, we have a national used cooking oil whereby this exercise it's being regulated by the Malaysian Palm Oil Board. Okay, and this year the Malaysian Palm Oil Board, they are launching a reference price in Q1 this year to ensure transparency of fair trading. So actually the eateries as well as the consumers as well as households like us, the used cooking oil that you use for cooking, for frying, instead of to dump it into the to the sink. Okay, so you can actually collect it back in Malaysia, and there will be someone to come over to collect all this used cooking oil. And current selling price in the cooking oil in Malaysia is around like ringgit Malaysia 150 to 250 kilogram. So the Malaysian Palm Oil Board will be now monitoring the price for this used cooking oil to ensure that everyone also be able to get the benefits. And of course, this is we are doing it for the green growth as well as for sustainability of the environment, and at the same time also to generate some incomes for the countries. Usually in Malaysia, the used cooking oil, part of it will be used as the biofuel, the biodiesels. And yeah, so this actually bring me I want to actually bring up our one of our projects, okay, back in actually 2011, whereby it's funded by Japan Toray Foundations, whereby actually I collected the used frying oil, the used cooking oil from the fast food chain, and this is the oil that I have collected from the chain, from the fast food chain, one of the local fast food chain, and to do some pre-treatment, I'll be able to actually remove the all these residues. And actually you can see the middle one is the used cooking oil that after we have done some pre-treatment. So what actually I wanted to highlight over here, so as you can see before pre-treatment, you can see that actually our used cooking oil in Malaysia is actually the used palm oil. So the used palm oil actually preserve a lot of the C18-1 and also C18-2. So in the oil and fats, so we look into a lot of the functional groups, and the main functional groups is actually the unsaturations. So this actually shows you that even the unsaturations of like the oil that collected from the fast food chain that's like becoming so dark, but it's not degraded much, whereby we are able to preserve the double bonds, the unsaturations of about 46%. So we actually remove all those black residues and be able to, you know, the oil, you can see that is totally different, and it's actually like the fresh oil, and the without actually disturbing the C18-1 is the oleic acid and linoleic acids, the fatty acids of the unsaturations being able to retain even after the pre-treatment. So actually with this unsaturations, we actually convert it into the epoxides. So we measure the oxirane oxygen content, the epoxides, and then we're able to actually produce this used cooking oil that have been recycled. So we produce epoxidized cooking oil. So this is the epoxidized oil. And the percentages of oxirane oxygen content is actually comparable with the fresh palm oil. Usually a fresh palm oil will be able to produce epoxide palm oil with the oxirane oxygen contents about 3.2 to 3.5. So this actually shows you that instead of to just discard the used oil, so there's a lot of things that we can do with the used oil. And what we actually we have done with this used cooking oil, we produce the epoxidized palm oil, okay, epoxidized oil, and then we further convert into a polyesteramide. So later on, I also have some examples of the polyesteramide to show you. And this polyesteramide from this used oil, we actually incorporated some metal, and we use it for some of the, we prove it for antimicrobial properties. So another class of the polymers that we work a lot on is the polyglycerol ester. So this polyglycerol ester is being produced from glycerol. As you know, the glycerol in Malaysia, it's actually a byproduct from the biodiesel process. So when the biodiesel process, they produce a lot of this glycerol, as well as Malaysia one of the productions of the oil chemicals is the soap. So in the saponification, they also remove a lot of glycerol. So in view of the glycerol, okay, that is abundantly produced by the biodiesels industry. So we actually make use of the glycerol to produce a polyglycerol ester. So this polyglycerol ester is biodegradable, and then a lot of ester linkages. It is enzymatic degradable by the lipase, and its application is actually for surgical material and drug delivery agent, and etc. So this is the work that we have done. Sorry, it's jumping. Okay, by using our polyglycerol ester that we have produced. So we produce it by making it into a hybrid branching. So there's a lot of work producing polyglycerol ester, but they are not hybrid branching. So our innovations here is actually the hybrid branching, and because of the novelty, we have able to actually file a patent on this metal incorporated hybrid branching polyglycerol ester. So whereby we make use of the glycerol and also some of different range of dibasic acids, and then we produce a hybrid branching of polyglycerol ester. So by making use of the OH group, and then we further actually synthesize copper incorporated polyglycerol ester. So in all our synthesis methods, we're actually trying to apply the green chemistry principle. So we use solvent-free in these reactions, and then we use renewable feedstock, and then the material that is being produced, make sure that it's biodegradable. And this is the next step, okay, how we actually apply the what is the application of the copper incorporated polyglycerol ester. So we further react it with this isocyanate IPDA. So this is isophorone by isocyanate. It's an aliphatic isocyanate, whereby we actually make use of the copper incorporated polyglycerol ester that we have synthesized, and further react it with this IPDA, and to produce a PU polyurethane coating. And this coating actually be proven for its antimicrobial properties. So in these reactions, we make sure that we use less hazardous synthesis. Why? Because in the industry, mainly the isocyanate that the industry is using is the aromatic. So they're using the aromatics. For example, they are using the different type of TDI, toluene diisocyanate, in order to produce any polyurethane material. So for us, we're using aliphatic. So aliphatic is less reactive, but however, okay, it is also less toxic compared to the aromatic diisocyanate. And this is the findings whereby the when we produce the polyurethanes by using the IPDI and also the metal incorporated polyglycerol ester. So you can see that we conduct the diffusion test and then using different microbes. So what we can see is that this is the standard control drug, and this is the polyurethane that without the copper incorporated polyglycerol ester, and this is the one that with the copper incorporated polyglycerol esters. So you can see that but it's able to incorporate into the polyurethane, but at the same time also it's exhibiting the antimicrobial properties. So another work that we have done is actually to produce palm oil based polyester polyol. So this is another work that we have worked on it, and it's actually funded by the Malaysian Ministry of Higher Education exploratory ERGS grant. So this is what we produce. It's a new palm oil base, produce it from palm oil, and it's a polyester polyol. So it's a different approach and also different chemical structures of the polyester polyol. So what we actually make use of this to react with IPDI. We produce a palm oil based polyurethane and use it for soft tissue engineering scaffold. So this is the synthesis method whereby we make use of the epoxy ring. So again, this epoxy is the epoxide group is being created from the palm oil. So palm oil will have a lot of double bonds, or not a lot, okay, but we have a sufficient number of unsaturations, and whereby we be able to convert it into epoxide, and making use of epoxide, we be able to react it with the dicarboxylic acid and to produce the polyhydroxyl compound it is known as the polyester polyol. And this polyester polyol, okay, how we actually synthesize it, the approaches is again solvent-free, one-pot synthesis methods, use of renewable feedstocks, as well as it's biodegradable. So the production's next step is actually we want to make a material out of it. So for soft tissue engineering, so we actually make use of isophorone diisocyanate as mentioned, that is less reactive and less toxic. And we also do not use those solvent. We actually use a water blown. We use water as a solvent in these reactions. And the good finding is that it be able to produce a material whereby it has high water uptake, and then the mechanical properties of it is suitable for soft tissue engineering and very high porosity. And this is the images that we show the cell attachment on our newly produced the polyurethane scaffold. As you can see, the development of the philopodia was observed on the polyurethane foam that we produced by the palm oil based polyester polyol. So another work that we have done is on this polyester amide. So just now I think I mentioned about that we produce polyester amide from the used cooking oil. And so another work we actually make use of the palm oil as well, palm styrene, because the excipient that we wanted to use is to use it in the solid dosage form. So okay, and then what we actually have done is we are producing this polyester amide and use it as the excipient for a solid dispersion, and we try to incorporate with BCS class 2 drug mefenamic acids and to produce it to prepare it into a solid dispersions pre-formulations. So how we actually did that is that the palm oil, okay, the palm stearine, we mix proportionally in order for us to achieve the desired physical chemical properties, because we wanted to use it as a carrier for a solid dosage form. So we react it with diethanol amine and then produce it with diethanol amide. So by making use of the OH group from the diethanol amide, we choose choose a suitable dicarboxylic acid, and we produce it into a polyesteramide with the desired properties. So of course we need to do a lot of trial and error, and then optimizations, and then be able to actually get a whole series of the polyesteramide with different molecular weight. So we have different grades. We have 4,000 up to 17,000 of the molecular weight of this polyesteramide. And we also make use of different compositions. So with all these different compositions, we actually just choose one of the active ingredients. We actually wanted to try to see whether be able to improve the dissolution as well as solubility of the mefenamic acids. So the results is quite promising. So as you can see over here, all these are the solid dispersion that prepared by the polyesteramide with different grades, different molecular weights that we have produced. And this is the mefenamic acid pure drug. So you can see that it's a huge improvement in terms of the drug release. And we also compare it with the solid dispersions that we have prepared, compare with the phenogesic. So this phenogesic is one of the mefenamic acid formulation that we obtain from the local pharmacy outlet, and we make a comparison with it. So we found that our formulations, the pre-formulation short dispersion, not even that added with other filler, and then we did not do that. Okay, so we did not add other filler and also other excipient. We just use a solid dispersion, compare with the finished marketed phenogesics marketed formulations. So we can see that it's actually improved the pure drug, and then comparable and even better. Okay, so and also we also found that even the physical mixture. So we use the polyesteramide that we have produced, just mix with the pure drug, and we can see that it's actually better than the pure drug. So this means that the polyesteramide that we have prepared is actually having the surface lowering effect, some surfactant effect. We just mix it, it's also better. And when we use a little bit of the techniques like solid dispersion techniques, is so much improved and is better than the marketed drug. And this is another work that we have done on the polyglycerol ester amides as the drug carrier for solid dispersion. So you can see that we are trying to actually diversify our work all by using the oil chemicals from the palm oil. So some of the work we actually use glycerol. Okay, so the glycerol in the earlier part I have actually shared that we produce a polyglycerol ester. So later on I think of how about, because the polyglycerol ester that we be able to produce is hyperbranching. So I think of how about we make use of this hyperbranching of polyglycerol ester to make it into an amide. So I produce this hyperbranch polyglycerol ester amides. And this work is novel because of its hyperbranching, okay? And because all the esteramide that's being produced in the market is linear, and what we are able to produce is hyperbranching. So that's why it's actually enable us to file another patent on this hyperbranching polyglycerol ester amide. And we actually make use of the stearic acids and then and also diethanol amine to produce the diethanol amides. And then after that, we have another step of we are making use of the glycerol and also the dicarboxylic acid to produce the hyperbranching of polyglycerol ester. So subsequently, the hyperbranching polyglycerol ester, we make use of the stearic acid-based diethanol amide. You may wonder why stearic acid-based, because of we wanted to have the solids, we wanted to use it for solid dosage form. So we need to have some solid excipient. So that's why we will need to choose stearic acid. So in the pump-based oleochemicals, there's a very huge range of properties that you can actually explore. So that's why by understanding its chemicals, structures, as well its properties, to be able to produce you wonderful products. And this is the hyperbranching polyglycerol ester amide that we have produced. And again, we try to apply as much as possible the green chemistry principles into our reactions. And this is the degree of branching that we actually be able to determine by using carbon-13 NMR based on its dendritic as well as the linearity branches. So when we do our calculations, we found that the hyperbranching of the polyglycerol ester amide that we have produced having quite a promising degree of branching range of 57 to 62%. This is considering a very high percentages of high hyperbranching. And in terms of its in vitro dissolution performance, so by making use of the hyperbranching polyglycerol ester amide as the drug carrier for the solid dispersion, so we actually incorporate with PCS class 2 model drug, we use lovastatin. And then what we can see is that again, like we're able to produce like different grade, different molecular weights and chain length of the polyglycerol ester amide range from 5,000 to 12,000, and we incorporate with the lovastatin as a solid dispersion, and we also carry out dissolution study as well as a solubility study. So we can see that is this ST7 making use of the hyperbranch polyglycerol ester amide 12,000 be able to release 88% of the lovastatin. So compared to upper lovastatin is the commercial formulation that we obtain locally. So it's only 52%. And the pure lovastatin is only 43% of the release. So this shows that the effectiveness of the hyperbranching polyglycerol ester amide that we have produced. And in terms of solubility, so this is the one that we have selected it, ST7 with the prepared by PGEA, the hyperbranching polyglycerol ester amide 12,000 is actually fourfold better than the pure drug. And then this is upper lovastatin, only 17. So it's only 17 microgram per ml. In terms of solubility, it's so much better, our solid dispersion. So we also wanted to know, like actually, when we make use of our hyperbranching polyglycerol ester amide, is there any changes on the physical properties? So what actually we did is actually using the SEM. We can see that the pure lovastatin at first is like sharp edges. So after we mix it into making it into a solid dispersions along with our PGEA, so we can see that they turn into the amorphous form with all the rounded edges. We can't find all these crystalline drugs anymore. So this actually shows the conversions of actually we have disrupted the crystallinity of the lovastatin into amorphous form. That's why the release will be improved. Because the drug is no longer crystal. And this is the SEM images of the solid dispersion of the lovastatin before and after storage. So we have also done some other characterizations whereby we also characterize to look into its dissolution, solubility, SEM. We also look at the chemical properties. So we found that there's actually no difference after storage. So this means that the amorphous solid dispersion prepared by using the polyglycerol ester amide is stable. And this is what actually we have published, that we have explained that what is the interaction between the hyperbranching of the polyglycerol ester amide with the model drug lovastatin. So it's actually a lot of the hydrogen bonding. And when we actually we design the structures of this hyperbranch polyglycerol ester amide, is because of its enable a lot of the site for the drugs to be forming a hydrogen bonding to incorporate them so that they are in good interactions, as well as that's why it is stable. So because if you do not have enough sites for it to do the binding, after storage, after some time, it will be leaching out, and then the drugs will be becoming crystal again. So yeah, coming to the last, I think, 2 slides. Okay, so yeah, just to share some other works that our group that we are doing. Okay, so because of the time, I'm unable to actually share all of our work. So some of our ongoing work is actually we are working on multi-responsive polymer. So we actually are working on smart polymer. And one of the interests that we are actually looking into, the research interest that we are looking into, is the non-isocyanate routes. So I have actually presented that in my work, we try to use less hazardous chemicals, whereby from the aromatics, I used to be using aromatic diisocyanates, okay, in my postgraduate work. Then later on, I moved on to using aliphatics, because aliphatic is less toxic, and we also carry out some in vitro work to show that the aliphatic isocyanate is less toxic when you use it to produce the polyurethane. And these recent years, actually, I have able to come up with a new path, a new route, chemical route, whereby we are offering like non-isocyanate route. So we produce polyurethane as well, but without using any isocyanates. And we actually be able to actually produce this material, polyurethanes, whereby it's proven for its pH responsiveness, light as well as thermal responsiveness. And we have actually published some work. Okay, if you're interested, you can also you can also download the articles to know a bit more. And this pH responsive, light as well as thermal responsive polymers, we use it for different applications. So you can use it as a plasticizers, as well as you can use it for drug delivery because of the pH responsiveness. And of course, another and also other work that we are doing is also we are working also in metal organic frameworks. And this metal organic frameworks is produce the ligand part. Okay, the linkers, we actually produce it by using oleochemicals, and the oleochemicals is the novel oleochemicals that we have modified. And then we react it with the metal complexes, and we use it for drug delivery as well. And all this work, we try our ever best to apply the approaches, green chemistry approaches, atom economy in our non-isocyanate routes, less hazardous synthesis, and renewable feedstock, and so on. So yeah, just to share with you, so this the non-isocyanate polyurethanes routes that we have actually managed to file patents, and also this pre-polymer, okay, we name it as oleonippon because it's from oleochemical. Nippon means non-iso polyurethane acrylate. So we actually also making it into a polyurethane acrylate, and currently we have some proof of concept, and then we have proved it TRL4. We hope that we are now slowly working into improving the technology readiness. So this is the exhibitions that we have exhibited our oleonippon and some of the products. So just last but not least, just to share some of the challenges, and also we think some of the strategies that can improve the sustainable bio-based polymers. Of course, when we work on all this from scratch, always we have encountering scale-up limitations, and we think that maybe we can use like DOE methodologies to improve the scale-up parameters. And of course, all the researchers, we have a dream, wanted to commercialize our brainchild. So one of the things that we also, there's a lot of challenges when you come to productions. There will be regulatory hurdles. So I think we will need to actually work very hard on providing comprehensive toxicological and also biocompatible data, animal work, preclinical work, etc. And also when we are because we work on bio-based. So bio-based, one of the things that of course is sustainable. So when sustainable, we also need to balance with the performance. So a lot of time, if you are opting for the renewable feedstocks. So one of the limitation, of course, is you will need to actually work very hard in order to make sure that the performance is on par with the petrochemical based product, and of course cost competitiveness. So we try to need to actually optimize the green synthesis process. So as like the Olonipua, so we successfully produce it, but of course it involves more steps. So when more steps is involved, then there will be costs, incurred more costs. So we need to actually balance these two things. And of course, the market acceptance, because the market usually they will have already have been using the petrochemical based product. So even like when you talk to the manufacturer, why they actually wanted to use your oleochemical base. So market acceptance is also one of the challenges, and that's why a lot of time that we try not to replace everything, but we replace proportionally in order to increase the market acceptance, and of course provide more clinical and preclinical evidence on the safety as well as the efficacy and the performance is on par with the marketed product. So last but not least, I'd like to thank my university, IMU universities, for providing me all the support, as well as the research collaborators, all my research students, all funding bodies, MOHE, Malaysian Palm Oil Board, Japan Toray, MOSTI, and many others. And also would like to take this opportunity to give a special appreciation and thanks to Chebrolu, Hanumaiah Institute of Pharmaceutical Sciences. Thank you for the opportunity to have me to do a bit of a sharing over here. So this is our research team, some of research students, some of the collaborators, and thank you very much. Terima kasih. And thank you for for your attention. And we welcome you to visit us in Kuala Lumpur. So our campus is in Bukit Jalil, and this is our very well-known KLCC Tower, KL Convention Center. So with that, I'll thank everyone for the opportunity. Thank you.
So I request the audience to come up with questions.
Am I audible, ma'am? Am I audible, ma'am? My question is, hello, ma'am, am I audible?
Yes, I can hear you.
Yes. So here you said that there is a metal conjugations with these oils, right? You said cobalt. Cobalt is the metal you have selected for the conjugation, right? So other than cobalt, any other metals are adjustable to have the complex? You have conjugated. Yes. Tell me.
I can't hear you very clearly. A little bit of breaking. Can you repeat the question?
So
Am I audible, ma'am?
Yes.
The question from our speaker is, at some point of your presentation, you mentioned that cobalt is used as a compound for the preparation of the polymer. Apart from cobalt, did you find any other metal having that property similar to that of cobalt?
Oh, okay. All right. Thank you so much for your question. So actually we have tried like several different type of metal, and then the metal that we choose is actually the less toxic. So apart from cobalt, we also have tried zinc. So zinc also be able to to give us quite a good antimicrobial properties. Yeah. So and it's also not that toxic. Yeah. So we have actually tried cobalt, and then we tried zinc, and aluminum. Unfortunately, we are unable to produce a desired properties of the polymer. Yep.
Okay, ma'am. Thank you. Can you please show me the slide where you kept the microbiological report of where you performed this disk diffusion method, where you used antibiotic combined with the polymer?
Sorry, dissolution?
You showed us a slide where antibiotics, when along with the polymer, had better inhibitory zone in the microbiology report.
Okay. Yeah. So what is your question? May I know?
I would like to know what was the reference taken for the drug. I think it was ampicillin.
Correct.
There were A, B, and C. 3 points were taken.
Correct.
I think the C one is the one in which the antibiotic is given along with the polymer, which had better inhibitory zone.
Oh, okay. So I think you are referring to the one of our work whereby we make use of the cobalt. Yes, cobalt incorporated. Cobalt incorporated polyglycerol ester. And then, okay, maybe let me very quickly I share the screen so that to enable So enable me to give the explanation. Okay. Okay, so I'm not sure why it's not working. Okay, so maybe I just verbally explain, okay, whereby we have using this diffusion methods, whereby we show A is actually the vehicle control, whereby we use 1% of the DMSO, okay, and this 1% of DMSO, of course, is showing that like nothing happened, okay, and then B is the ampicillin. Ampicillin is the antibiotic, okay, so this antibiotic against the Staphylococcus aureus that we have used in the examples that I have shown. And there's another two. One is C, another one is D. So actually, D is the blank polyglycerol ester that have been used as blank polyglycerol ester that used to produce the coating material. So this coating material, okay, they are having produced by the polyglycerol ester, but without the cobalt. And the one that's showing the effectiveness is actually the C, whereby we use the cobalt incorporated polyglycerol ester to prepare the PU coating, the polyurethane coating, and it shows the inhibition in the Staphylococcus aureus. So did I answer your question?
Yeah, so the B is the plain ampicillin. B is plain ampicillin. C is ampicillin plus the cobalt coated ester.
No, actually there's no drug. Oh, okay, I get your question. All right. So actually, the control drug ampicillin is actually to serve as a control whereby we know that, okay, so this Staphylococcus is being inhibited, able to be inhibited by the ampicillin. So this shows that the experimental setup is valid. So it's actually to test on the validity, we make use of ampicillin. So in terms of what we're actually testing, we have two coating. One coating is the one that without cobalt incorporated, and then we produce it into a polyurethane coating. Another one is the cobalt incorporated polyglycerol ester as the PU coating. So actually the active over here in the pill coating is the cobalt incorporated polyglycerol ester. So the active is the one that we are using it, that one that will synthesize it.
I got the point. Thank you.
Thank you.
We would like you to please stay online. We want to have you here with the felicitation. Now I request our principal and the organizing secretary to please come on to the dais. I also request Dr. Vasudev Rao to please come on to the dais. I request Dr. Vasudev Rao to accept the felicitation on behalf of our speaker. I once again thank the speaker for your session here with us today on this conference. Thank you once again, ma'am. And I hope Dr. Vasudev Rao will bring the award safely to you.
Thank you to the organizer as well as the chairperson and also Dr. Vasu and everyone. Thank you so much.
Thank you, ma'am. Now I would request our principal and organizing secretary to please felicitate the chairpersons of this session. First, I request Dr. Abdul Rahman to please come forward and accept the felicitation. Now I request Jitendra sir to please come forward and accept the felicitation. Thank you. And now we break for lunch, and post-lunch session will start at 2 p.m. in the same venue. We'll break for lunch, and post-lunch session, that is plenary session number 10 on this conference, will start at 2 p.m. in the same venue. Thank you. Plenary session number 10 happening right now. And for this plenary session, I would like to introduce the chairperson and welcome Dr. Sachin R. Patil onto the dais. He is currently working as professor and head of department Pharmaceutics, Sarojini College of Pharmacy, Kolhapur. Sir has MPharm and PhD degree in pharmaceutics with a professional experience of 19 years. He has 16 papers presented in national and international conferences and 25 publications in several Scopus and PubMed Index journals. He has authored 2 books and published 2 patents to his name, and he is also the Assistant Controller of Examination for the last 14 years at KLE University, Belagavi. Sir, we welcome you, and I'm very happy that you are staying with us since all 2 days of this conference, and we are very happy to have you, and I request you to please introduce the speaker to the audience.
So good afternoon. This is, I think, plenary session 10. Before commencement of this session, I would like to congratulate Dr. Vidyadhar Rao, Principal, for his organizing impactful conference. So today in plenary session, we have a speaker, Dr. P. Sambashiva Rao, who is a founder and MD of Indie Global Labs, Hyderabad. Actually, sir is a seasoned clinical research professional and founder and managing director of Indie Global Labs and CDCSO approved contract research organization, CRO, based in Hyderabad. With over his 23 years of experience in clinical research industry, he has played a key role in advanced bioanalytical and clinical development services in India. He holds PhD in chemistry and master's degree in clinical research and regulatory affairs, supported by more than 15 years of experience in GLP-compliant bioanalytical laboratories. His expertise spans medical writing, clinical trial management, bioanalytical methods development and validation, pharmacogenetics and data management. Sir has also worked with leading pharmaceutical companies, CROs, and he's been involved in successful clearing more than over 20 national and international regulatory inspections. He has contributed to develop of more than 400 bioanalytical methods, participated in over 600 bioavailability and bioequivalence studies, and been involved in more than 10 clinical trials. Sir has specialized in setting up laboratories, developing quality assurance systems, and implementation of regulatory framework, ensuring compliance with the global standards and supporting successful drug development through Indie Global Lab. So with this brief introduction, I welcome our speaker and today's speaker, Dr. Sambashiva Rao, in the dais, and I would like to give this mic to him and to show his further slides.
Thank you, sir.
So I would like to welcome Sambashiva Rao, sir, Principal.
Thank you. Thank you. Please.
I'm sorry for this miscommunication. We have another chairperson chairing this session, and I would like to invite on stage Dr. P. Raghuveer Verma, who is currently the professor and principal of... I request him to please come on to the dais. I'm sorry for this miscommunication, sir. Sir is currently working as professor and principal at Chennupati Indo-American College of Pharmacy. He has a professional experience of 15 years and has 15 research publications presented by him at various conferences. He authored one book chapter and also published one patent. He received Science Academic Summership Fellow in the year 2018, AICTE PG Fellowship year for GATE qualification, and postdoc fellowship funded by DBT New Delhi. With this brief introduction, I once again welcome him and thank him for accepting our invitation for being the chairperson for this session. Thank you very much, sir. And now I request our speaker to please take on the session.
Thank you for the introduction, sir. And it's my privilege to be here today. And I thank CHIPS management and the organizers for giving me this opportunity. You know, gaining knowledge, keeping it for ourselves will not be a good. So, knowledge must be shared, particularly upcoming students like you all people. You are the future professionals in the industry or the education or maybe entrepreneurs. So if you have a knowledge, then it will be good to build your future. So today I am here to give you some added knowledge to your theoretical knowledge. Since I am from industry, I have hands-on experience conducting clinical research, starting from medical writing where the development of protocol, preparation of informed consent, taking informed consent from volunteers, followed by recruitment of volunteers, handling of investigational products, then the translations into vernacular languages so that volunteer can understand what they are giving the consent. And the method development, particularly when you are doing pharmacokinetic studies, it is very much required to measure the pharmacokinetics of a particular product so that the bioequivalence can be proved based on bioavailability of the generic drugs. Particularly the clinical research very much important for generic drugs because we need to serve society with economical medicines. In the society there are many people who may not afford to buy innovator products. So that is the reason generic products are very crucial for the society. Even that is the main reason for the government for opening generic medical shops. So even bioanalytical, when I say we use normally chromatographic methods, you might learn about paper chromatography, thin layer chromatography, high performance chromatography. So LC-MS is a liquid phase chromatography with mass spectrometer, which is the advanced technology where you can have a greater accuracy, precision with robustness. So I have a few slides where you can gain some knowledge about clinical research and innovation ideas or discovery or what are the stages, what are the regulatories are there, what guidelines you have to follow while conducting clinical research. Then as a pharmacy graduate or postgraduate, what is your role and where you can see your future. And I suggest you to observe my slides. There are proverbs. I will not repeat those proverbs because the proverbs may be very short, but the meaning is very depth. So if one will follow those proverbs, you have so much to understand. And I request everyone to note one thing that make a or display this word attitude and look at that word every day once for at least 15-20 days. That is your life. Once you understand what is attitude, then you will get everything what you are wishing for. So today's topic is also again discovery and innovative methods. So there are so many things that are happening nowadays in the world. People are not simply following traditional ways. There are advanced techniques. AI/ML is one thing that your life will be much easier. Say if I talk about 20 years back, if some pharmaceutical company wish to innovate a small synthetic drug, it takes normally 10 to 12 years by investing billions of rupees. Now, because of AI/ML, at least it got reduced to 6 years. So you see a lot of time is saved, so that the money also saved, so that the objective of supplying economical medicines to the society is fulfilled. So that technology has always helped humans to strengthen the knowledge or the skill, or to gain knowledge, or to speed up the processes. So whoever study pharmaceutical sciences, they become a pharmacist. You know the role of pharmacist. Pharmacist is a bridge between industry and the society. So you are the person who take care of the public for their health. Whoever suffering with any disease, they must approach a doctor followed by pharmacist. Doctor only can suggest what kind of medicines are the treatment, but you have to allocate or dispense the drug to the patients and guide them to take correct, accurate treatment so that the disease can be eased. So you have a vital role to play. So with the technology, particularly advanced technology as of today, the discovery is very much easier than old days. Once upon a time, to identify a lead, it takes lot of homework, lot of screenings. That takes almost 1.5 to 2 years. Hundreds of structures, hundreds of functional groups. We have to screen and select leads. Now, with the help of advanced tools like ChatGPT, it is very much easier because the ChatGPT has metadata. It can process the data in a faster manner. But it will not give you the conclusions. ChatGPT is a chatbot, is not a human. Human can think very logically than these advanced tools that we are talking. So don't rely completely on advanced technologies, but take support, take help, so that your work will get easier and you will see the speed in your work. So quality is always prime thing whenever you do any work. So you make sure that quality is there. Particularly when these chemically synthesized products are manufactured, during the process, lot of steps, lot of chemical reactions involved. The raw material, you have to pick raw material with a concern that it must be very much quality product, so that your process will not give you impurities. You know how much damage that an impurity can give it to the humans. So recently, if you come across the news, maybe the current thinking of the regulatory is not leachables and extractables. While process of manufacturing product or while preparing a tablet or capsule, the process should not give intermediate impurities. If there is an impurity, it must be less than the acceptable limits where it is not hazardous to humans. So you make sure that quality is always a concern, and you should not infuse adverse events while or along with the treatment. So that is a major motto. So because these are all the allopathy medicines normally based on chemically synthesized products. So chemically synthesized means reactions. Reactions of different mixtures, organics, inorganic, salts, buffers, like that. So you make sure that you follow quality practices so that you will not end up with a product which gives adverse events. And you may be knowing that there are cases where products are recalled. That is because of the adverse events only. You know, post-marketing surveillance. Regulators collect adverse events occurring to the patients who are taking the medicine. They collect the metadata and analyze regularly. If the risks associated with that product are weighing more than the advantages, then such product will not exist in the market. It will be recalled. And innovation. Why we need innovation? If you go back and review our 100 years history, there is always advancement. There are a lot of changes in humans. There are a lot of changes in environment. 100 years back, the compounds are very less. Diseases are also very less. Now, there are new diseases are coming. So we have to get new treatment for that. There are many viruses. You may be knowing 3 years back we had a coronavirus. So always there is a need. So always there is a need for innovation. Innovation to strengthen the treatment. Innovation to short the treatment period. Innovation to give comfort to the patient. Innovation not to repeat the doses repeatedly. I mean, the frequency must be reduced. Say, for example, paracetamol. Once in 6 hours you have to take to keep your fever controlled. If you have a thought where the release of this paracetamol can be increased to 12 hours, so the patient can take one tablet once in 12 hours. So that is one innovation. So then to get extended release, you should have advanced technologies like nanoparticle technology. So that is the upcoming stream where nanoparticles will be formulated as a tablet and given a smaller doses so that bioavailability will be increased without impacting by the gastric juices, so that the therapeutic efficacy will be observed, so that the disease will be in control. So for this chemically synthesized pharmaceuticals, always there is a regulatory. In India we have CDSCO. At the same time, every country has their own regulatory body who looks or who do the surveillance of the medicinal products. If the risks are more than the benefits, then such drugs will be recalled based on the post-marketing surveillance report that are submitted once in 6 months by the pharmaceutical companies to the regulatories. So why industry collaboration is much important? In a classroom, your lecturer maybe explain you about the process, the treatments, the class of drugs. But while listening, one can keep 60% of the whatever they taught you, 60% can be retained. Some may retain 80%, some may retain only 20%. But when you see with your own eyes and feel practically, then it will be remember for a longer time. And maybe you may not even forget. So that is the reason practical experience is very much important. So after studying your 3 or 4 years degree, or while studying, if you got a chance to explore yourself practically to learn something from your, say for example, how a clinical study is conducted, how a protocol is written, how a volunteer consent is taken, how a bioanalytical method is developed, how a LCMS looks like, what are the techniques. So if you get a chance to practically experience those things, then those skills are the advantage for you to go into the industry or to become an entrepreneur. So nowadays everyone is talking about entrepreneurship. So instead of going to someone to do a job, you start your own company, become an entrepreneur and give jobs to others. Anyone can do that. You need to have a attitude. Money always doesn't matter. Even governments are supporting students to, if you have an idea, you can go to government and they will fund you. They will help you to start your company. So don't think like a people who way back 20, 30 years, everyone used to think that I need to get a job. Now that is not the case. Now everyone must think that how to start a company. So you go for that, you get one idea, you have to work hard, think smart, use logics, then you will be survived. So as I mentioned, traditional is a manual method. It takes lot of time, lot of money, lot of people are required. Now modern research, there are advanced tools to strengthen your teamwork or shorten your durations, save money. So we have to use the advanced tools. It will be easy. Madam, that is not the one. There is a window, right? In the PowerPoint. But no, not that one. Not that one. No. The other one. That is the one, madam. Yeah. Sorry for that. So while doing discovery, so as I mentioned, while screening of molecules to identify leads. If you take advanced tools help, it will be easy. With a very short span of time, you can have a leads, so that those leads can be taken for further development, whether they really work to reduce any disease condition or not. So that is the clinical trials part. So before that, the toxicology must be checked. So now it is very much easy. 10 years back, everyone must go to office, work from office. Now it is different. You have a mobile, everyone has a mobile. You have an entire database. You have to just Google it. You will get information or the process, everything. Everything is in your hand. So 24 hours, 365 days, you have a privilege to learn or to work. I have one question, if anyone can answer. How many drugs are invented by Indian pharmaceutical companies? She has mentioned 0. So whatever the medicines that we are using are invented by foreigners. You can imagine how difficult to invent medicines. Maybe the first problem, we are not even thinking to do inventions. We are just following someone and thinking that someone will innovate, we just use it. You just go back and Google it. When the blockbuster medicines discovered, what was the outcome? How much that pharmaceutical company gained? You will be surprised to see those figures. If some 100 discoveries are there, maybe out of 100, 90 will go failure. That is because of lack of science, lack of planning, lack of finding a solution to the problem. Discovery, you should have always or continuous thinking, logics. Instead of doing 10 experiments, you have to do one after one. So you have to evaluate each experiment after its completion. Why you have done, what was the conclusion, what is your next step. So that you will go to your exit point. Otherwise it is a failure. So planning, evaluation is very much important for discovery. So once you have a leads, say for example you have screened lakhs of structures with different functional groups, and you have some confidence on 3-4 molecules where those can work for some disease treatment. Now you have to check whether they are really working or not. And the most important point is whether they are safe. So once you complete your laboratory work, so your next step is to see the product performance and the safety in living things. Since it is new to humans, we cannot administer directly to humans because human lives are very much important. So the other option is to test the same in animals like rats, mice, dogs, monkeys. So a lot of animal models are used to test the toxicological effect. During preclinical, entire process, how it is absorbing, where it is binding, how it is metabolizing, how it is excreting, how much drug is required to treat that disease, what is the frequency, what is the lethal dose, what is the lowest dose, what is the multiple dose effect, or what kind of accumulation it is happening, whether the drug entirely is eliminating from the body or not. These all things will be evaluated by using animal models. Once the toxicology data means that the trials are completed, then we will test in humans. Even during toxicology trials using animal models, we have to check each and every organ impact by that medicine, whether it is carcinogenic or not, how it is absorbing, or how it is interacting with your liver enzymes, or how it is impacting your kidney. So each and every organ is tested, and once the advantages are significantly higher than the risks associated with that medicine, then only further process will be continued. So if someone say that they got failure during their clinical research or preclinical research or at the in vitro stage. So this slide is about clinical research. So after starting clinical research, if your product is not showing good performance, it could be because of the lack of design. Particularly these clinical trials, you know, clinical trial phase 1, 2, 3, 4. There are different designs. So single blinded, double blinded, parallel study, crossover study, single center study, multiple center study, pediatric study, normal healthy volunteer study, or elder studies in elderly people, studies in females. So that is appropriate design will help you to get a success. So you should have a knowledge how to design those studies to fulfill your objectives. Even while preparing your protocols, it is very much important for what object you are developing that protocol. So you should ensure the correct design, the washout period if applicable. Then if it is a pharmacokinetic study, the blood samples where you are going to withdraw, how much you want to withdraw, what moiety you have to measure for the pharmacokinetics, and what kind of pharmacodynamic changes that you want to monitor. So each and everything is part of design. Design is nothing but your protocol. So protocol is entirely a design. Once your protocol is prepared scientifically, appropriately, and according to regulatory, then you will have a great percentage of success. So as I mentioned, quality is very much concerned in the regulated environment. If there is no quality, there is no success. So as I mentioned, there are two things. One is innovation. Whenever there is innovation, one should do in vitro studies, then followed by preclinical in animal models, then followed by clinical phase 1, 2, 3. Then your product will be in the market. Once it is in the market, we have to do post-marketing surveillance, that is phase 4. So generics are, on the other hand, those are different. Once the patent is completed for the innovator product, so other pharmaceutical companies can come up with their own solution. So that is a generic product, but we have to compare this generic product performance in respect of bioavailability by comparing with the innovator. So when these two products are matching in respect of bioavailability, those two are called bioequivalent. The regulators will accept only the generic products which are bioequivalent to the innovator products, because innovator products have an entire data that is generated, the preclinical, laboratory, then the phase trials. But generic product, you are not generating these all things. Only a study in healthy volunteers is sufficient for generic drugs. Then regulators will accept your product to be in the market. So whenever we do any process related works, say for example when you are developing a formulation and you have a multiple steps involved, then you have to ensure whether the process is validated or not. Whether the process is giving exact the final end object every time with the same accuracy and precision. So the process that you are using to make any product, we have to validate it. So that is the reason method validation or analytical method validation or COA generation. When there is any product, we have to generate a COA. For each batch that is manufactured, we will have a COA by using a validated method. It's not that simply they just checked the characters. No. The COA is generated based on a properly, scientifically, regulatory oriented developed method. Then you will have accurate results to present. Then conclusions, evaluations. Evaluations normally will be done in preclinical or clinical by using pharmacokinetic analysis and statistical analysis. So we have one subject in our pharmacy also for that. So there are different strategies for the evaluations of any clinical trial or any clinical study that we do. We have to check the results properly with science behind. It's not random conclusions. It will be based on a formulas appropriately designed according to the regulatory requirements. And bias. Bias is if someone is going based on bias, then you will not get what you are wishing for. So you should avoid bias at all stages. That is the reason if you see clinical trials, particularly clinical trial phase 3, when you are checking the effectiveness of the product in patients, so you have to go for a double-blind study. It means one arm will be the product, another arm will be the placebo. So since you are going to conclude your product effectiveness, it should not be a random. So you are very sure by using placebo controlled trial that yes, this medicine is going to work. So these are the regulatory pathways. So as I mentioned, every country will have their own regulatory. In India we have CDSCO. Likewise, America they have US FDA. In the same way, UK they have UK MHRA, Canada Health Canada, ANVISA, Brazil, ISP, Chile, China FDA, Japan. So every country will have their own regulatory. And if you want to market your product in any country, you must obtain their permission from the statutory body. ICH is a common platform. They develop guidelines and the world will use those. ICH is particularly for the harmonization. So there are 195 countries, each country will have a different guideline. If a manufacturer wants to sell their product in 195 countries, it is very much difficult to follow all the country guidelines, and every time we have to do a study. Then it will be multiple work for them. So to reduce that burden, ICH is coming up with a harmonized documents where countries adopt those guidelines so that one single clinical study will be sufficient to go for the all countries who adopted ICH guidelines. So compliance is very much required in regulatory environment. So it will be burden, but compliance will give you the final credibility. So if you have a good compliance, even though that is a burden, still you have to get the compliance entirely, not at the end point, entirely from zero to end. There must be compliance. Then you will have a good credibility. I give one example. If you go to, say for example, you want to purchase lock and key. Many may go and ask, give me Godrej. That is the credibility. So if you have such kind of quality compliance, then you will get that credibility. So as I mentioned, even if you attend an interview, they say attitude. If you have a proper attitude, even though you are not able to answer their 100% questions, still you will get a job because of your attitude. And ethics. Because we are into clinical research handling humans, we must have ethics. And learning is a continuous process. So you should ready to learn every day, every minute. So these are the career paths. You have a lot of opportunities there. You can go to hospitals, you can own your pharmacy, or you can own your manufacturing facility. There are so many requirements are there. Chemical preparations, impurities, or CROs like us, or you can have clinical trial CRO, or you can have a hospital, or you can have a pharmacy, or you can go to R&D, discovery, preclinical, or you can have a pharmacovigilance, or you can become a statistician. There are many opportunities for you. Every subject in your graduation will lead to one company. In company, say for example in CRO there are different roles. Say volunteer mobilizer, paramedic, doctor, clinical research associate. Then you have analyst, QA monitor, QC monitor, sponsor monitor, regulatory expert, or medical writer, or clinical investigator. There are so many roles in each company. So whenever you get a chance, try to go to industry and practically feel how they are working, what kind of instruments are there, how instruments are functioning, so that you will have an additional knowledge or the skill. It will help you in the future once you join somewhere as a professional or you want to become an entrepreneur. If you have a interest in one segment, go and visit those related industries so that you can have a practical experience. So practical experience is something different for your theoretical knowledge. Theoretical knowledge is just, you know the process, but practically how exactly that is functioning. So if you go and see practically, then it will be easy for you to understand and keep that in mind, so that it will give you edge in the future. And even I suggest, if you have a chance to interact with industry people, you try to discuss how they are doing and what kind of technology they use, what kind of instruments they use. And even every company in a regulated environment must use standard operating procedures. It's not that everyone will do or work according to their own wish. No. So there is a unique procedures. Unique procedures are developed based on the requirements. It's not that one person with their mindset has developed these standard operating procedures. No. According to the instrument, according to the requirement, according to the regulatory, those are standardized. So once it is standardized, everyone must follow those. So as I mentioned, you have a lot of opportunities to utilize. You have to just think, or you have to just go with an idea and your interest. So you check all your textbooks, all your topics. You evaluate yourself. What topic is most interested to you? Then go into that field. You will get success. So as I mentioned, practical. To get practical experience, you can go to industries as an intern, or you can do projects, and you can participate in research. So these are very much important. Regulators, they require data. So whenever you're doing discovery or clinical research or post-clinical, everywhere data is important. So you have to collect the data, evaluate the data, you have to process the data, then submit your conclusions to the regulators. So it is up to you that students, they have a fresh mindset, fresh knowledge, so you get more ideas. So you have to ask yourself, what is my idea? So if there is no innovation, there is no future. So innovation is required with quality, impact, ethics, then you will give impact to the society. Thank you so much. If you have any questions, I am very happy to answer.
Yeah, it's a very comprehensive and detailed session. Of course, Sir has put, because he is from industry, so he has put it all in bits and pieces, and I would like to keep this in a nutshell. And before going to that, if there are any questions, can you please, students, can you please ask any questions if you have? Okay, so let me put all of what has been told in a nutshell. So actually the first slide, drug discovery is not linear, yes, because the advent of AI and ML has, it brought in more molecules than the last few decades. So this decade we have a lot number of molecules in the drug discovery industry, and when you say drug discovery, so usually we in India, we start drug discovery programs, but we get only leads and we leave there. The problem is we have to convert these leads into which can answer questions or which can serve the public, which means that these leads have to be converted into drugs. So we do academic research more, but none of these academic research is going into the public use. So that's the topic actually, what I understand. Innovation to implementation to create impact, right? So that's what I understood. So these innovations, whatever we do at academia level, so they should be properly shaped so that they reach the public. So we stop only at ideation stage where we produce some new molecules, that's it. So if academia can be more, if it is more, what do you say, serious, we can have molecules. Because if you look at during COVID, if you look at some, because most of the innovations happened during COVID. Right? Yes or no? Do you agree with me or not? Right? So if you look at some of the innovations during COVID, Oxford vaccine, which was discovered by 2 PhD students in Oxford. So it has happened in academia. So that's what I wanted to say. So because there is a gap always, academy and industry has a gap. So this gap has to be bridged by, as Sir told, so it has to be bridged by proper exposure to the industry. And then Sir has also mentioned that you students should become, or you should establish enterprises or new companies, industries. And Sir has also tested on translational research, where that's what ideation to end product has to be done. And then I don't agree with Sir at one point, sorry to say this, because there are molecules from India, and when I was sitting there and I googled it, there is a molecule called as enmetazobactam, which is discovered by Orchid Pharma in 2008, and now it is in the market now as a combination, which is in the name of Exblifep, which is marketed for gram-negative microbial infections. So we have molecules. Of course, I agree with Sir. Indian market is full of herbal products. We are good at herbals, and so what we have to do is that we have to join hands to discover more molecules, and moreover pharmacist has a greater role, as the title itself says that the role of the pharmacist as a catalyst. So we have a greater role not only in the healthcare sector but also in discovering new molecules. So I really thank Dr. P. Samsharao Garu for this enlightening lecture, and I also thank my co-chairperson Dr. Sachin. Sir, thank you very much. And I also thank the organizers for giving me this wonderful opportunity. Thank you. Thank you very much.
Adding to Sir words, collaboration between colleges and industry is very less in India. If you see the foreign countries, USA or European countries, these pharmaceutical companies, they give grants to PhD students who are studying in the colleges. Then they start doing the research by their fresh knowledge. Then the leads will come. Once lead is generated, then this pharmaceutical company will take over. So that's how they manage this discovery. So such kind of scenarios are very much required in India as well. And thank you for pointing out one point. Maybe that is even knowledge for me now. Thank you so much. And once again, I thank CHIPS College management and the organizers for giving me this opportunity. Thank you so much.
So now I hand over the session to the organizers. Thank you.
Thank you so much, sir. Now I request our principal and organizing secretary to please come on to the dais. Thank you, sir. Now I request our speaker to please join the hands as we felicitate our chairpersons. First, I request Sachin sir to please come forward and accept the felicitation. Sir was with us today. We congratulate him, sir, because he was there with us all throughout the conference yesterday and today, and he was a very active participant, and we thank him so much for his participation. Raghuveer Verma to please come forward and accept the felicitation. Thank you so much, sir, for being here with us today. Thank you so much, sir. And with this, we completed plenary session number 10. And the only session that is remaining is the last plenary session of this 2 days international conference. And that plenary session is going to be very big and very special because we have the Vice Chancellor of Mangalyatan University who is going to deliver that plenary session. And please give us 2 minutes of time so that we can start that session immediately. Okay, and we are starting with the last plenary session of this 2-day international conference on emerging innovations in pharmaceutical sciences and drug discovery with the title Innovate Pharma 2026. And to do the chairpersons, I would like to call upon stage first Dr. D. Ravishankar Reddy, who is currently working as Assistant Professor at University College of Pharmaceutical Sciences, Acharya Nagarjuna University. I welcome him on the dais. Sir has master's degree in pharmacy and PhD. He has a total professional experience of 20 years and has 40 research publications to his name. He has authored 1 textbook and published a patent by himself. He achieved Young Scientist Award from Association of Biotechnology and Pharmacy and Best Academician Award from Scientific Laurels approved by Ministry of Corporate Affairs, Government of India. With this brief profile, I welcome you, sir, and thank you for accepting our invitation for being here today. Next, I would like to invite on stage another chairperson, Dr. P. Malleshwar Rao, who is currently working as Associate Professor at Department of Pharmaceutics and Pharmaceutical Analysis, Chebrolu Hanumaiah Institute of Pharmaceutical Sciences. Sir has a teaching experience of more than 20 years. He completed his master's in KL University, Belagavi, and then went all the way to New Zealand, where he did an extensive project on food sciences. And then he continued his career as an academician since the last 20 years. And I also welcome him for being the chairperson for today's session. Now I request the chairpersons to please introduce the speaker to our audience.
It gives me an immense pleasure to participate in Innovate Pharma 2026 as a chairperson, and I am happy to share the biodata of Professor K.R.S. Samsevarao Garu, Vice Chancellor, Mangalayatan University, Jabalpur, Madhya Pradesh. Professor K.R.S. Samsevarao is a distinguished academician, researcher, and visionary higher education administrator. Earlier, he served as the Vice Chancellor of Mizoram University, that is a central university, from 2017 to 2022. With more than 4 decades of experience in teaching, research, and academic leadership, Professor Rao has made remarkable contributions to Indian higher education. He possesses an exceptional and rare interdisciplinary academic background. He earned a Doctor of Science, DSc in Biotechnology, a PhD in Pharmaceutical Sciences, and a PhD in Zoology, along with postgraduate degree in Zoology, Physiology, Information Technology, Sociology, and an MBA in Big Data Analytics. He also holds a postgraduate certificate in Statistics and a postgraduate diploma in Computer Applications, enabling him to integrate science and technology and management in academic governance. Professor Rao began his academic career at Acharya Nagarjuna University, where he served for nearly 3 decades. He played a key role in establishing the Department of Biotechnology and launching innovative programs such as MSc Biotechnology, MTech Biotechnology, and MSc Nanobiotechnology. He later served as Rector and in-charge Vice Chancellor of the Acharya Nagarjuna University. A highly productive researcher, Professor Rao has published more than 277 research papers, authored and edited 22 books, hold 8 patents, and has submitted 6 gene bank entries. He has guided 26 PhD scholars and 21 MPhil students. He also served as visiting scientist and professor at several international universities in the USA, Germany, Malaysia, Panama, and the Czech Republic. As Vice Chancellor of Mizoram University, he transformed the institution by achieving NAC A grade, securing top 100 NIRF rankings, mobilizing over 575 crores for research and infrastructure. His leadership continues to inspire excellence in higher education. I think with these remarks, you can inspire by listening the lecture given by Professor K.R.S. Samsevarao Garu. I am inviting Professor K.R.S. Samsevarao Garu to the dais for delivering his lecture. Please come on to the dais, sir. Thank you, sir.
Good evening, everyone. I thank the chairpersons and principal of the institution, Professor Vidyadhara, and all the faculty members and guests invited. It was very, very difficult in the afternoon session. Even for me also, I am very much feeling tired, getting sleep. So I don't know whether I am going to give any kind of information or all going to sleep. I don't know about that. And today's topic is maybe a little difficult for you to understand, role of omics technologies in drug discovery and development. Drug discovery has got so much of transformation nowadays. the old traditional drug discovery, drug development has been changed. And now new technologies are coming up. What are these new technologies? That is, omics are the technologies which are actually been going around the various molecular pathways and biological pathways of the human beings. And they are actually been helpful to target the disease area and disease tissues, and it enables the pharmacists to develop the drugs which are actually useful to control of particular disease. Particularly in the last 2 decades, the various drugs which are actually being useful for the control of various diseases. The synthetic drugs which are actually being available now are going to develop, already developed resistance. For example, the penicillin which you are using for several purposes, since many days, now it is not been useful to control any pathological diseases. Similarly, several medicines actually has got the similar effect. And several drugs which are actually being useful for you to develop, to control the various diseases, they also developed resistance. And these drugs actually been very well not effective, and also the parasites or microbial organisms also developed resistance. And you might be seeing several presentations, antibiotic resistance, and even several diseases which we are now able to use medicines for various diseases have developed resistance. This kind of resistance, even though we are using, that will be not effective and also not useful for you to control the particular disease. For example, even we are using some tablets, every day in your house or my house or anywhere, if you get headache, immediately you will be using one headache tablet. If you continuously use the kind of tablets regularly for several years like that, that tablet will become ineffective because the headache also is not being able to control with that particular medication. So we need to seek the alternative mechanisms. And what are these alternative mechanisms? One best example for all of you is that this drug development takes, all of you might have been described in the past 2 days, will take 10 to 15 years. Because several things actually, just now we have listened to some people, and even in the morning also we have listened to some people. And phase 1, phase 2, phase 3 clinical trials, the previous speaker has been explaining. And finally, it is going to give a period of 10 to 15 years and several billions of rupees which actually been spent for the development of drugs. This is going to be costing so much. Now, for example, if you just see the recent COVID pandemic situation, though the process of drug development takes several years, even for cholera and other diseases, which has taken more than 10 years. But the COVID vaccine has been developed within a period of 6 months. How it is possible? This is possible only because they have not used the drugs which actually in the synthetic process, they have developed drugs which actually by the omics pathways, omics technologies, they have taken and they have developed this particular one, and they could able to do that, and within a period of 6 months. But even then they have to go for clinical trials, and with the basic steps of clinical trials, Government of India has permitted them to use for the treatment of COVID vaccine. But there is another possibility that we are talking about pharmacogenomics. That pharmacogenomics is the subject actually related to you and even molecular biologists. So it has got a particular targeted drug delivery for a group of individuals who has got a particular problem. That particular pharmacogenomics is one of the technique. They will understand the molecular makeup of the individuals, a group of individuals, and they will develop the drug for the particular group of individuals. This is called targeted delivery. Targeted drug delivery doesn't require the clinical trials. It is directly can be used for the individuals. and it can be tailored to a particular requirement. Now this has come in North America and Europe. The drugs are not required to be going for clinical trials. You can just directly use the pharmacogenomic drugs developed, you can use directly to the patient. This is also possible. How it is possible? It is through the omics technology. What omics actually? Genomics, proteomics, metabolomics, RNomics, and transcriptomics, and microbiome. These are all omics actually, which are going to play a crucial role in identifying the drug target. And this drug target actually being useful to develop the drugs of individual requirement. So this is the topic I am going to discuss. Maybe a little difficult for you because it is not your area, but it is a modern area. But little problems, anywhere you have problem, you can understand. I will try to be very simplified, and I will skip many slides are there. It takes around 1 to 1.5 hours. So I will make it half an hour, 40 minutes, and I will, because all of you might be very eager to go away. So I will just make it within 40 minutes. So omics technology is high throughput technology, scientific methods, and comprehensive study entire set of biological molecules like genes, RNA, proteins, metabolites. Within cells, tissues, organisms, providing a holistic view of biological system, unlike traditional methods of focusing on single molecules. And this is a revolutionizing medicine, personalized diagnostics, drug discovery, understanding complex diseases like cancer, agriculture, development, and environmental science by revealing intricate molecular interactions, enabling early disease detection, and accelerating innovation in health and food science through large-scale data analysis. Omics technologies insights into genetic variations, that is genetic variations in human beings, protein functions, because all of you know that gene is going to be transcribed into RNA, RNA is going to transcribe into protein, and proteins are mostly enzymes and the various metabolic compounds, actually biochemical compound, which plays a very important role in biological system. So this entire proteomics study, genomic study, and metabolomic studies will give you an understanding which drug is going to be effective, which enzyme is going to be effective, which gene is going to be effective, and helping to pinpoint relevant targets, design better drugs, and stratify patients for precision medicine. Just I'm giving only definition part very slowly. Other parts I will go very fast. What is genomics? Because there are several omics. First one is genomics. Studying the structure and organization of the genes of any cell or individual, it is called genome. That is genomics, structure and organization. Transcriptomics, analyzing the RNA molecules, which is also called transcriptome, to understand the gene expression. The central dogma will be like this. The gene will be transcribed to RNA, RNA will be transcribed to protein, proteins will be enzymes and metabolite compounds in biochemical compounds. So every part of that is called another omics technology. Proteomics, similarly to genomics, structure and organization of the proteins in a cell or in an organism, it is called proteomics. Metabolomics, measuring the full complement of small molecule metabolites and their biological reactions and functions, it is called metabolomics. Other omics, lipidomics, large scale study of lipids, fat, axes and steroids in a biological system, analyzing their structures, functions and metabolic pathways to understand health and disease at a molecular level. You have to understand several of your biological systems with the changes in lipid profile, you have so many diseases can be also forecasted. Even lipoprotein A is very high, it is going to lead to your heart attacks. And the low quality fatty acids are going to develop problems in your blockage of the various blood vascular system. So lipid profile is very, very important. Lipidomics is one of the important areas to study. Microbiomes. Microorganisms is the scientific study of microbes, bacteria, fungi, viruses, and their genes living within us, focusing on their collective structure functions. Microbiology is one of the area, microbiomics is one of the area where it is very easy to culture the microbes. It is very easy to transfer any gene into human gene into microbes and culture them in large scale. And our gene product can be cultured in a large scale. So microbiomics, study of microbiomics is one of the very important area. And the spatial omics, this is actually molecular location of the in tissues. Molecular location means what are the molecular components, DNA, RNA, various molecules, or where they are locating and where they are shifting. That is actually family technology that map the spatial locations of molecules like RNA, proteins, lipids, metabolites, where they locate. That study is called spatial omics. These are the various omics technologies which are going to play a very crucial role in drug development. Now this is the area, this is the regime of omics technologies for drug development. You are not going to depend on the traditional drug system. Whole world actually depending on omics only. And whole world is dependent on omics technologies for development of the new drugs. Now the traditional drug development process has been gone away. Whole world is depending on this. Please remember that. These are very, very important areas if you want to become a very good researcher, if you want to become something, a novel scientist. These are the very important areas. What is significance and applications of these omic technologies? Holistic understanding of the single molecules, understand complex biological networks, personalized medicine, I was talking to you, pharmacogenomics, identifying the genes, actually predispositions and molecular markers for tailored treatment, tailored drug treatment. Disease research unravels complex mechanisms of multifactorial diseases, cancer, diabetes, neurological disorders. Biomarker discovery finds reliable indicators for early diagnosis, prognosis, and treatment response, telomeres, and several biomarkers, prostaglandins, several biomarkers actually, their presence or their absence, and even several biomarkers are developed based on the various kits for the identification of the diseases. So this is one of the biomarkers are actually useful. Based on the biomarkers, you will identify particular gene is present or not, particular gene is absent or not. So, but that particular gene is expressed or not, that you can understand. Drug discovery accelerates the identification of new drug targets and understand the drug effects can be also studied. Nutrigenomics studies how diet affects the genes. Even diet, environment, and acquired characters also will be varying. That also will be known to you. Food science, analyze food quality, safety, shelf life, and authenticity. Systems biology, integrate the different omics data, all omics data integrating, and pharmacoinformatics, bioinformatics, cheminformatics, these are actually going to play a very crucial role. And that will give you an idea how this particular system is going to be responding for a particular disease. This also will be known to you. In a sense, omic technologies provide a panoramic view of life's molecular machinery, transforming biology from reductionist studies to comprehensive systems level insights crucial for modern science and healthcare. This is going to be a crucial thing for the entire drug discovery in future. What is different from the traditional drug discovery? Traditional drug discovery you all know, but I am taking you to a different world that is new and improved version. Drug discovery you have to forget after some years. It is not going to be valid after some years. Drug development process begins with identification of a disease and its root cause of the disease, rather than a treatment. If you go to a doctor, doctor will give you several medicines. He don't know what is the reason. Why you got the disease, doctor also not knowing. No doctor knows what is the root cause of the disease. But pharmacogenomics can give you what is the root cause. So knowing the root cause and giving treatment will not be causing any side effects to the human body, because doctor will be giving several medicines at a time, more than 10, 15 medicines. Trial and error basis, he will give first 5 medicines, and after 5 days he give another 5 medicines, and after 5 days he will give another 5 medicines, and he will not be knowing what is the root cause. That is why it is going on like this. The traditional medical practice is also not going to be very valid. Most of the human diseases in human beings are because of the genetic disorders. There are about 5,000 genetic diseases. Most of the human diseases, except because of any kind of environmental things or something, that is because of any cold weathers or something like that, you get most of the diseases, whatever you are getting, 80% of them are genetic disorders. Genetic disorders means if you are going to give some medicines which are not going to affect on the particular genetic disorder, how it is going to treat the particular disease. This is what actually happening in India or anywhere in the world so far. But now in North America and Europe, they change it totally. Now if a patient goes to a doctor, he will ask the doctor to go for the molecular analysis of your particular person. And after taking the molecular mapping of your genome, then he will identify what kind of genetic disorder is there. Because there is a change in any kind of molecular makeup. You all know, if you might have known about the DNA structure, there will be 4 nucleotide bases and AT-GC. If AT and GC will be pairing usually. Instead of AT and GC, if A and G are pairing, mismatching is taking place. There is a change in one single nucleotide. That is called single nucleotide polymorphism. Change in the nucleotide pairing is called single nucleotide polymorphism. One single nucleotide polymorphism may be a genetic defect and may lead to a disease. And most of the disease, most of the SNPs are going to be genetic disorders, but not all the SNPs. Kindly remember this statement. Not all the SNPs, single nucleotide polymorphism, will not be resulting in genetic disorders. Only some SNPs will result as genetic disorders. That identifying the genetic disorders is going to be the pharmacogenomics area, molecular of individuals. Then that particular group of SNPs can be used for developing a medicine. So drug development and discovery, how it is, modern drug development and discovery, pharmacogenomics, pharmacoinformatics is an important tool for identifying. Drug targets, usually proteins, binding of ligands, how proteins are binding to ligands, how rational drug design is useful, what are the benefits, saving time and rupees. So you have to spend minimum 1 billion rupees, $1 billion or $10 billion or so much of money for developing a drug. That is not required now. And 10 years time to 12 years time, 15 years time is not required. You can do within period of 3 months by identifying the molecular makeup of the individuals. So what is genomics and I just wanted to go from here very slow, very speed, because there are 67 slides. It will take, if I explain like this, it will take another 2 hours. So I go very speed. Just kindly excuse me for going speed. And also, if you have any doubt, you can stop me and ask anywhere. So genomics is one of the very important area. What is genomics and what is genome and genomics? Genetic content of a living cell is called genome. Total content of a genetic content of an organism or a cell is called genome. Genome is also defined as one haploid set of chromosomes total. In human beings, there are 23 pairs of chromosomes. That is haploid set of chromosomes. To study the whole genome of an organism is called genomics. Whole organization of genetic material content is called genome. The organization is called genomics. Now, we all know what is bacterial genomics, what is human genomics, what plant genomics, viral genomics, fungal genomics. Now, in 1987, when Human Genome Project has illustrated what are the functional genes in the human beings, there are 35 to 40,000 of functional genes identified in the human beings. And the other genes which are present, their function is not known to us, but they also may be useful in future because they will be also rearranging themselves and also getting into functional genes. So what is a gene? I'm going a little slow, only a few slides. Gene is a small piece of the genome, and roughly speaking, each gene is a set of instructions for making one specific protein. Set of instructions from a gene to become RNA and then to protein. That is actually called gene. Proteins are diverse group of large complex molecules that are crucial to every aspect of the body structure and function. So you can just see here chromosome. You will be finding a DNA, and a part of the DNA is gene, and that gene is going to transcribe into RNA. RNA is going to transcribe into proteins. That protein, most of the proteins are enzymes and hormones and various biochemical components. So now we know the genetic organization of many organisms. You may ask me, why you have to study the virus, bacteria, yeast, and several plants? Why you? Because human genes are not directly been cloned into human beings directly. Human genes are cloned into microorganisms. Human genes are cloned into bacteria. Human genes are cloned into cell systems. Human genes are cloned into plant systems, and they are being cultured large scale. So gene is going to transcribe a product of protein. So human genes directly we cannot clone into human beings. It is not possible. So because a lot of ethical issues are there. So we get microbes can be cultured easily. Plants can be cultured easily. Cell lines can be cultured easily. We clone the gene into human gene into particular organisms and we culture them large scale. For that we need the genomic organization of all the organisms which we plan. So that is the reason whole genome study of many organisms are being studied. Human genome contains 3,167 million chemical nucleotides, ATGC, 3,164.7 million genes, million nucleotides. The average gene consists of, each gene consists of 3,000 bases, ATGC pairing 3,000 will be there. But size varies greatly, with the largest human gene being dystrophin, which contains 2.4 million base pairs. The total number of known genes are estimated to be around 30 to 35,000 genes. Though it is maybe having more than 1 lakh genes, but we know only the functional significance of the genes. Functional value of the genes, we know only to 30 to 35,000 of the genes. And almost all nucleotides, 99.9% bases are exactly same in all people. 99.9% genes are same in all of you. Variation will be there only for 0.1%. Variation is there only for 0.1%. The functions are unknown for over 50% of the discovered genes. Less than 2% of the genome codes for proteins. Not all the genes will code for the protein, only less than 2%. Repeated sequence that do not code for the proteins, usually known as junk DNA, make up at least 50% of the human genome. Repetitive sequences are thought to have no direct function. These junk DNA will not have any direct function. But over the time, these repeats may reshape themselves, reorganize themselves, and they may be useful rearranging after that, and they may be useful genes after some time. During the past 50 million years, it has been observed that there is a dramatic decrease seems to have occurred in the rate of accumulation of repetitive gene sequences in human genome. So we cannot take it granted that the gene product which we have identified today may be same after 10 years, may be same after 5 years, because there is a possibility that repetitive sequence rearranging themselves. So we have to go for every time molecular analysis and to get the gene product. So just see here the primer replication stand gene and prepare, and you can see different nucleotides, and you will be getting several primers which actually replicate themselves, and you will be coding genes also you will be knowing, because it is been tagged with some kind of prime materials, and it will be going for agarose electrophoresis. Then you will be going for identifying the genes with various mechanisms. And this is the ATGC nucleotides. You can see cytosine, guanine, adenine, and thymine. These are the 4 nucleotides which will be repeatedly present. So study of genomics is classified basically as structural and functional genomics. Organization is called structural, functional genomics, which has got functional value, that is called functional. From here I am going to little slow, you can just little speed. Functional genomics deals with knowing how genes function, how they code for a particular RNA, particular protein. That is called functional genomics. And what is proteome? I am talking about genes, genomics, proteomics. Total protein content of living cell is called proteome. And structural proteomics and functional proteomics. Structure and organization is called structural proteomics. Functional, what kind of enzymes, what kind of hormones, what kind of metabolic products which are coming from that transcribing proteins, that is called functional proteomics. Knowledge of genomics and proteomics is now facilitating study of whole cell and its function, and it is possible to study the gene expression. So components of proteomics, how you have to study. Proteins, they are present in a cell system. We will go for the separation of the proteins by electrophoresis. And after separating proteins, identifying the protein, we go for the mass spectrophotometer, which identifies the various proteins. And then using bioinformatic tools, the various existing proteins, knowledge will be there. You will compare with the protein which has been isolated with the existing protein database. So you will identify the protein which is present in the system. So the basic proteomic analysis will be protein mixture will be there, separating using 2D gel electrophoresis and going for individual protein identification, cutting the interest of your protein, and then peptides will be isolated, and peptides will go for the mass spectroscopy MALDI-TOF analysis, and you will be identifying the proteins. This is all big procedure, you don't need, because sampling, if you just take the sample and give it to the particular analysis, they will do all this. So the same thing, proteins, how they have been isolated, and mass spectrophotometer, how it has been identified. Proteomic image will be coming in the last figures, and you will get the comparison of proteins, what kind of protein you have. So the central dogma of molecular biology, which I have been explaining to you, genome, the cell, genetic organization, and then you will be getting genes from that, and genes will be coding to proteins. The lower ones are proteins, different enzymes. Proteins act alone or complexes to perform many cellular functions. All cellular functions, almost all your enzymes are proteins. So proteins will be coded by, and genes will be coding to the RNA, RNA will be coding to the proteins. So there is a paradigm shift in molecular biology. Earlier, when we are talking about molecular biology, is the pre-genomic area, studying individual pathways in an organism, and their properties are only dealt. Now it is called modular cell biology. It is not organism, it is a modular cell biology. Meaningful study of organized and multi-component pathways in a cell, studies in particular about a cell at a particular instance, comparative study of healthy and diseased cells. So what is that you identify the proteins or genes? You will be having a particular database of the proteins, database of the genes. You have to compare the unknown sample with the known sample, and you will identify the gene, which actually causing a particular disease. So post-genomics biology, it is functional genomics. Major areas study of functional genomics, which play a crucial role in study of genomics. Transcriptomics, study of RNA. RNome, it is also same with transcriptomics. Proteome, that is about the proteins. Metabolome, that is metabolic activities, metabolic enzymes, metabolic contents which are taking place, metabolome. And glycome, that is the glucose and other biological activities going on in the human body. So just a small abbreviation, the total content of transcribed RNA, it is called, from the DNA is called transcriptome. And the study of it is also called transcriptomes. RNome, transcriptome is also referred as RNome. Proteome, the total protein content is of the genome. Metabolome, the total metabolites that constitute the metabolic pathway system of the cell. Glycome, the total carbohydrate content of the total glycoprotein present in a cell. All these are important for you to develop a drug. You understand? These are important now in the modern drug discovery. Again, the same all we have explained, we have just discussed genome to transcriptome. Genes are coding to RNA. that is called transcriptome. And RNA is coding to protein, that is called proteome. And that is being coding to the enzymes, metabolic enzymes and metabolic organs. That is metabolome. So relational database, store, compare and cluster. All databases are available in the website. NCBI database you can have. So you can compare your database of genes, proteins, metabolomics, RNA, everything you can compare with existing database. And which gene is present, which protein is present, which RNA is present. So you can find out with that. And correlation of genes and gene products, identification of gene function, gene annotation, identification of various pathways is possible. Biological knowledge, plant improvement, genetic and metabolic engineering is possible. So DNA sequencing, if it is known, you can make genetic alterations, genetic engineering also. So functional genomics and pharmacogenomics, this is your area. I was already explained to you, genetic difference between individuals, single nucleotide polymorphism, can cause differential mode of drug influence. Stratification of patients into genotypes may increase the probability of testing of drug efficacy. Therapeutic window, mode of action and limitations of its actions will be known. Can help to develop the personalized drugs for individuals. We don't need to wait for years together. We don't need to wait for the testing of the various drugs. Now it is easy. Individual drugs can be developed. Drug metabolic enzymes, drug transporting and drug receptors, everything will be coming like this. So pharmacogenomics, the branch of science, wherever the genome information has been made to exploit for the design of the new drugs. Pharmacogenomics is the branch of science in which you are using the particular information of the molecular data for developing the new drugs. This information is going to help you to develop the individual drugs for the individual patients. The concept of personalized customized drug design has been one of the most emphasized area of pharmacogenomics. So what you are doing here is human genome project after it has been elucidated. The genetic makeup of the individuals has been elucidated. 30 to 35,000 genes are known to us now. Remaining genes are not known to us. This particular known genes has got known functional significance also known to us. This known functional significance will be knowing what gene is causing cancer, which gene is causing particular Alzheimer's disease, which gene is causing Parkinson's disease, which gene is causing sickle cell anemia. We all know about that. This known information about the genes which are causing the various diseases will be identified. And what kind of defective gene is present in your body. Defective gene can be replaced now by gene therapy. And there are other techniques also. Defective gene can be removed with the functional gene. So this is functional genomics. So single nucleotide polymorphism, you can see here, the top one is in yellow color, the second one is red color. There is a change in base pairing. AT-GC pairing is not there. So the DNA sequence AT-GC pairing should be there. That is not there. That is called single nucleotide polymorphism. You have to remember, all single nucleotide polymorphisms are not going to develop a disease. But most of the genetic disorders are caused by single nucleotide polymorphism. So you can also see here, which is actually, this is not working, so I cannot show you. You can see here, on the top it is a functional gene, it is a change, it is a change, it is a change here. Nucleotide pairing is having a change. And that pairing, that is called single nucleotide polymorphism. So pharmacogenomic study combines the traditional knowledge of pharmaceutical sciences with an understanding of common DNA variations in the human genome. And single nucleotide polymorphisms are going to be taken as a coding for a particular drug development. It is estimated that in a human body, there are 11 million SNPs are present in the human genome, 11 million. But with an average of one for every 1,300 base pairs. I already mentioned to 3,167 million base pairs are present. With an average of 1,300 base pairs, single SNP molecule polymorphism is present. An individual response to drug is often linked to these common DNA variations. Individual drug response. You have to remember, the drug is not going to commonly working for everyone. But the present drug system is being developed in such a way that we are developing a drug, we are selling a drug. For everyone they will give, if you go to pharmacy shop, they will give a drug for everyone, same drug. But in reality, it is not going to work. For every individual, drug response will be different. Drug metabolism is different. Drug reaction will be different. This is the modern concept. But all, I cannot comment here, but all the present system and approach is not really good, really true. Pharmacogenomics is one of the area you have to understand. Pharmacogenomics focused on gene encoding either metabolic enzymes that can alter drugs activity or defective structural proteins that result in increased susceptibility to disease. So wherever you want me to stop, you ask me to stop or each other. Because time is, I don't know, because a lot of slides are there. So I can stop anywhere, I can finish within 5 minutes. 4 o'clock, okay. So this is one example for you, sir. Sir? I want to ask a question. Sir, you can ask.
As per your information,
yes, sir.
For all the people, the same drug will not be working in the same way.
Yes, sir.
That is also the principle of the homeopathy.
Yes, sir.
You mean to say that allopathy and homeopathy are nearing each other?
Sir, actually, pediatric pharmacy, adult pharmacy, geriatric pharmacy, these are the 3 areas now being developed in pharmaceutical education. The same drug which is actually being used for the adult people will not be working same in the children.
Yeah.
And it is not working same for the old people.
Yeah.
So the drug formulation has to be changed, drug quantity has to be changed, drug approach has to be tested. That is one point, basic point about the present drugs. But actually, basic problem is, because you are not there in the beginning, most of the diseases which are there in your human beings are genetic disorders.
Yes.
5,000 genetic disorders are there. You can sit there. Yeah. You can sit and ask, no problem. So 5,000 genetic disorders actually will be really giving disease because of the reason for the genetic defective genes. And the genes actually will be particular drug which has been used now will be, this slide will tell you, you have a group of people here, one is short, one is long, and one is black, and one is red. The same drug which you are developing, these all people will not have the same effect.
That is called pharmacokinetic variability, sir.
It is actually, I am not talking about pharmacy, sir. I am talking about pharmacogenomics. Pharmacokinetics and genomics cannot be compared here. Genomics total approach different.
They are different.
So genomics is understanding the molecular content of the human beings and tailoring of a drug of individuals basing on the particular drug response to the individuals. This is the modern concept of the drug. So I am not going into area of pharmacy. I am not a competent fellow to go into area of pharmacy. I am a competent person only to go into the pharmacogenomics area. So the drug response will be different from one individual to another individual. When the same drug is given to many people, it will not properly function for all the individual. It may be little properly function, it may reduce its function, or it may not function also. That is the reason why the tailoring of drugs for individual based molecular analysis is required. That is now coming up in the Europe and North America. In India, it has not come.
Yeah.
Still, we depend on the traditional medicine, traditional treatment, sir.
So, no, my point is, homeopathy people, they say they don't give the same medicine to the same causes in different individuals.
Homeopathy medicine, they vary the content.
They give that way.
They vary the formulation, they vary the mix the drug, and drug quantity also will be varied.
Yeah.
And they will reduce the quantity for children, they will reduce the quantity for adult people, they will increase the reduce the quantity again to geriatric people.
Yeah.
So, I am not knowing the mechanism of homeopathy, but the expectation that it is possible.
Yeah. Now, what you say is it is coming nearer to the homeopathy, or homeopathy is near.
No way comparison to homeopathy or no way comparison to allopathy, sir. It is a pharmacogenomics medicine. It is totally different.
Yeah.
So it is actually basing on the molecular analysis of the individuals.
But the medicines to be supplied and to be given to different individuals, they are different to different individuals. That's what you say.
Actually, sir.
Yeah. Then are we competent at the present in India to administer many medicines like that?
Yes, sir, it will come, sir. Because no, any development in the world, it is coming slowly. So it is the true fact, it will come after some years.
Not many people know the pharmacy also.
No, sir. Pharmacy is a different area. Bulk production of the drugs. Same drug, bulk production is required. Pharmacy people are required. But concept of development of drug discovery only I am talking.
Okay.
Concept drug discovery is different from pharmacy formulation and pharmacy large scale production. Okay. I am not going into the area of pharmacy, otherwise they will beat me. All of them will beat me, sir. So I am not going into the area of pharmacy. I am going only into the development of drug discovery, sir.
Okay, thank you.
The same drug will not be functioning for all individuals same way. It has to be different. Drugs that work, we don't know. Drugs that are safe, we don't know. Drugs that are appropriate for an individual, we don't know. That is possible only pharmacogenomics. So, one of the example, basing on the pharmacogenomics, Abcavir, this is one of the drug developed, which is actually been useful for developed for the treatment of HIV with HLA B57 gene defect. So it is been developed with 2000 samples can be treated with this particular kit which has been developed. So it is useful for the treatment of HIV disease with the defective gene of HLA B57. So this is how the group of people who are having a defective gene of HLA B57 will be controlled by this particular Abcavir disease. So I was talking about the databases of the proteins. There are several databases are like this available. So unknown disease database, diseased gene we are taking, diseased protein we are taking, diseased enzyme which we are taking from different sources, they will be compared with regular known genes, known functions, known proteins. What are the techniques used for this particular study of genomics? DNA and protein microarrays, DNA and protein chips. In that chips, known genes are loaded into the chip. When unknown sample is been treated with the known, compared with the known chip gene, you will be having a perfect matching of the certain genes for that gene chip or protein chip. That perfect matching is an indication that the particular gene which is responsible for cancer or HIV or Parkinson, it will be known. This is what actually the mechanism involved in that. Two-dimensional electrophoresis, that is separating the proteins based on the molecular weight and isoelectric pH. Mass spectrophotometry, separating the proteins. Computational biology, you will take the sampling, comparing various databases available, and you compare with the pharmacoinformatics and bioinformatics. Because I have been given with only another 5 minutes, I will try to finish. Gene knockout, the functional gene will be replaced with the defective gene. That is called gene knockout technology. RNA interference, the gene which cannot be removed from the individuals or plants, that gene will be stopped in functioning by developing another RNA interference compound, and they will introduce into the particular system, and that RNA will not be coding for the proteins. The protein expression will be stopped. That is called RNA interference technique. So DNA protein, because I am not going to study anything, because all this very long, that is synonymous of microarray technology. It can also be called biochip technology, DNA chip technology, gene array technology, gene chip technology, oligonucleotide technology, gene spotter technology. And what do microarray? I am going to skip these slides. Just only this slide, normal, without drug treatment will be taken, normal tissue, diseased tissue, and you will be comparing with the gene chip, with the diseased tissue or normal tissue, and you will find matching of the genes. If one gene is matching, that gene is responsible for a particular disease. This is the gene expression technique they follow. This is similar to that. This is production of brain tumor chip. Brain tumor is present, which gene is responsible for that? You have to go for the comparison of normal genes of a particular person and also the defected area, and you compare and you will find out which gene is responsible for that. So this is what actually microchip, actually DNA chip looks here. This is a micro ampli chip CYP450 array gene chip tech, how it looks. Applications in pharmacy, discovery of therapeutic drugs, and it is customized drug selection is possible, and there is a possibility for ineffectiveness of drug over the period of usage of a particular drug. I was mentioning you, penicillin now is not being useful. Penicillin has to be changed. Its tertiary structure has to be modified, or its secondary structure has to be modified. Then only it is going to be effective. 2D gel electroporation, this is not required now. So this is isoelectric focusing, basing on separating the protein, basing on the pH, isoelectric pH. So how protein using mass spectrophotometry will be isolated? So you just see here. I don't want to discuss because I'm given another 5 minutes only. This is mass spectrophotometry analysis. Proteins finally will be isolated basing on the last mass spectrophotometry graph. You will identify the proteins basing on the fixed databases. Computational biology, comparing with the regular database, and also analyzing all omics technologies into one particular analysis. So here, computational biology predicts the three-dimensional structure of proteins. So normal protein structure, structure with the desired gene expression, structure displaying gene suppression, structure with enlarged gene suppression, enlarged view of gene expression, mutant cancer protein with the additional amino acid sequence. So normal chemical structure, modified new chemical structure. This is only idea for you. So 3D chemical structure and prepared for docking of the particular molecule. So gene knockout, I already mentioned, particular defective gene will be replaced with functional gene. RNA interference, the expression of a particular gene will be blocked by another RNA. So then, for example, tomato, artificial tomato, preservation of tomato. Tomato normally if you keep outside it will be spoiled within 3 days. But this RNA interference, tomato can survive for 6 months now. This has been proved true in tomato. Gene expression. So you are studying the gene expression. How many genes are from the diseased organism you are taking out, and they are comparing with the known genes. So you can see here, it is a gene actually, gene 91, gene 92, gene 93, gene 94 has been analyzed. So gene 91 is not functioning. It is off. And gene 94 is also off. Only 92 and 93 and 95 are on. So you can say that gene 91 and 94 are not functioning for the particular disease. So like this they will identify. Even obesity also, there are 2 patients has been compared and they have identified that some of the genes are not functioning in one individual, even though same age people, they have obesity and the other person is not having obesity. That is because of the gene which is responsible for obesity has been expressed in a person that is not expressed in another person. So protein microarrays, I will just skip here, and all thousands of proteins at one time. You can see here protein-protein interaction, protein-protein interaction, how they will be, protein-ligand interactions and radioactive materials, and small molecule arrays, protein arrays, that is protein chip technology, and antigen and antibody chips, and 2 major types of protein functional proteomic arrays and the antigen and antibody arrays, and protein ligand docking. You can remove one ligand and another ligand can also be added. So protein ligand will be taking place like this. Many commercial antibody arrays are available now. Advantage of protein chips, high throughput analysis can be done, and what proteins are functioning or what proteins are not functioning you can identify. Drug development today and tomorrow. Today it is taking 20 years, billions of rupees. But tomorrow, future, we are going to develop drug for every individual, drug tailoring of individual drugs is possible. Pharmacogenomics is defined, tailoring a specific drug to the biological state of the patient. This is what definition you have to identify. You have to remember, pharmacy is not just only the industrial pharmacy. Now pharmacy has got a big scope, lot of opportunities for the pharmacy people. Don't look only for industry jobs. This is what my message. So opportunity to deliver the promise for individualized medicine is now US has been covering very well, and pharmaceutical companies now will be able to create drugs basing on pharmacogenomic analysis. They can develop drugs, any kind of drugs they can develop now. They don't need to depend on FDA, or they don't need to depend on any approvals. Advanced screening for diseases and more accurate methods for determining appropriate drug diseases, better vaccines, improvements in drug discovery and approval is process. Vision for medicine, actually you are going to develop pharmacogenomics for any kind of disease you can develop very easily. So challenge, the take home message, it has to be still streamlined. Genome analysis for all individuals is not that possible, not that fast. Rapid automated methods must be developed to efficiently identify SNPs. Studying the biology of genes involved in disease and drug reactions is also has to be studied. It can take decades of study to gene product function. SNP analysis and expression profiling are their infancy, and very few success stories are existing. Complex diseases are very complex. Really, disease and drug response can involve hundreds of genes. Environmental factors such as age, nutrition, and lifestyle can influence disease and drug response. Thank you. Now, if organizers permit, any questions can be asked, no problem, but it depends on.
Thank you, sir. Thank you for such a wonderful presentation about the omics technology. When sir has started this topic, I was also just wondering, because omics technology is very complex to understand, but sir has started from, intermediate level, and he took us to the PG level. Like, he started with simple DNA thing, RNA thing, and all the things. This technology, what sir talking is omics technology. I think it involves so many things, starting from the genomics, then followed by transcriptomics, proteomics, metabolomics, and there are several things which needs to be completely understood to develop this technology. And presently the COVID vaccine, I think, which is being developed is also based on omics technology, if I am correctly said, mRNA based technology, the COVID vaccine. And one more thing is, the sir what mainly talking is about the pharmacogenomics, which is developing the medicines based on the genetic makeup of the individual person. So every person, as sir rightly told, will behave differently. Their body behave differently to the same drug. So now in future, maybe not in near future, maybe it may take more than a decade, when you go to the hospital, the first thing what they used to do is they used to study your genomic structure and genomic things, and followed by prescribing the medicines what is most suitable for you, so that the disease can be cured without any side effects. That is what all sir is talking about. So this is also called personalized medicine, because every person is going to get this medicine. But you may see in a couple of Hollywood movies where people are just going and scanning and they will be prescribing the drugs. But that will be the near future with the omics technology. I think sir has given a wonderful session, and now the session is open for questions. And one more thing I want to point out to sir is, maybe you must have not heard, sir has done his he has done double PhD. Doing one PhD is different, but sir has done 2 PhDs. I always wonder, sir, how you have done the double PhD. So he has a vast knowledge, and sir is the powerhouse speaker. So he starts, he keep on going, but because of the time limit, sir may not able to complete his topic. But if you have any doubts, you can ask him now, or you can interact with him later. Sir is very friendly to students. So thank you, sir, for such a nice presentation, and we are honored to have you here, and we'll be having you here again in December for the conference. Okay, sir. Thank you, sir. Thank you very much.
Now I request our speaker, our principal, to please felicitate the chairperson for this session, Ravi Shankar Reddy sir. We also request Malleswara sir to please take the felicitation. We also request Malleswara sir to please take the felicitation for acting as a chairperson. So with this, we successfully completed all the plenary session. So with this, we successfully completed all the plenary sessions of this two-day international conference. And we are very shortly starting the valedictory event, less than 10 minutes. So I request all the audience and delegates to please be seated. Please give us 5 minutes to arrange the stage and we will be starting the valedictory. All the guests will be here itself. So I request everybody to please be seated so that we can start the valedictory as soon as possible. Thank you.
Who is he?
ఇలాంటి విషయాల్లో ఆడపిల్లకు ఒకే ఒక దారి.
ఇది బాగా ప్లాన్ చేసి కేర్ఫుల్ గా ఎగ్జిక్యూట్ చేసిన ఒక కోల్డ్ బ్లడెడ్ మర్డర్. We are dealing
dreamer, is it?
A very good evening to one and all. I wholeheartedly welcome you all to the valedictory function of the two-day international conference on emerging innovations in pharmaceutical sciences and drug discovery with the title Innovate Pharma 2026, organized by Chebrolu Hanumaiah Institute of Pharmaceutical Sciences in association with Indian Pharmaceutical Association. We are happy to say that we successfully completed the two-day international conference, and it's the time to celebrate those 2 days with the amount of presentations that happened, the amount of research that is discussed, and the amount of exchange of knowledge that has happened. So, without any delay, I would like to start this valedictory event and call upon the distinguished personalities onto the dais. To welcome them at the outset, I invite on stage our beloved principal, Dr. S. Vidyadhara, onto the dais. Next, I invite on stage Dr. G. Mangaya, who is the Secretary and Correspondent of RVRR College of Education, onto the dais. Thank you, sir. Thank you for being here with us today on this occasion. Next, I would like to invite on stage Dr. P. Gopijan, who is the management member of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences. Next, I would like to invite on stage the organizing secretary of this conference, Dr. V. Venkatrao, onto the dais. Next, I would like to invite on stage the distinguished guest for today's function, Dr. T.V. Narayana, onto the dais. Dr. T.V. Narayana, President of AP Pharmacy Colleges Private Colleges Association, onto the dais. Next, I would like to invite on stage the chief guest for today's function and also the person who gave us the last plenary session, Dr. K.R.S. Samsarao, the Vice Chancellor of Mangalyatan University, Jabalpur, onto the dais. Next, I would like to invite on stage the spectacular trio of this conference, Mohammad Zulkifli, Dr. Chin Swee Yi, and Dr. Vasudev Rao, onto the dais. They traveled all the way from Malaysia to be here with us for these 2 days, and I cordially welcome them. Next, I would like to invite on stage another guest, Dr. Venkat Ikurti, the founder and CEO of Chanakya Education Hub, onto the dais. Next, I would like to invite on stage Dr. M.A.M. Prasad, the principal of M.A.M. College of Pharmacy and also the president of IPA AP State Branch, onto the dais. Next, I would like to invite on stage Dr. G. Sumalata, who is the IPA Education Division Chairman, onto the dais. Now the stage is all set for the valedictory to begin, and we would like to begin with a prayer song.
Mahaganapatim गणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि वसिष्ठवामदेवादिवन्दितमहागणपतिं महादेवसुतं महादेवसुतं गुरुगुहनुतं महादेवसुतं गुरुगुहनुतं मारकोटि प्रकाशं शान्तं मारकोटि प्रकाशं शान्तं महाकाव्यनाटकादिप्रियम् महाकाव्यनाटकादिप्रियं मूषिकवाहनमोदकप्रियं महाकाव्यनाटकादिप्रियं मूषिकवाहनमोदकप्रियं महागणपतिं मनसा स्मरामि वसिष्ठवामदेवादिवन्दितमहागणपतिं मनसा स्मरामि. Thank you.
Now I request the organizing secretary of this conference, Dr. V. Venkatrao, to give the conference report.
Good evening to one and all. As we came to the conclusion of Innovate Pharma 2026, a two-day international conference on emerging innovations in pharmaceutical sciences and drug discovery, on behalf of the organizing committee of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences, I consider it a great honor and privilege to present the closing remarks. Over the past 2 days, this conference has truly emerged as a vibrant platform for knowledge exchange, innovation and collaboration, with the enthusiastic participation of 1,500 delegates, so 1,100 online and 400 offline. 1,100 offline and 400 online. Innovate Pharma 2026 witnessed a remarkable convergence of academicians, researchers, industry professionals and students from across the country and abroad. We received an overwhelming response in the form of 650 scientific abstracts reflecting the strong research culture and innovative spirit of the participants. I am pleased to highlight that all accepted abstracts have been published with an ISBN number, ensuring global visibility and academic recognition. So furthermore, eligible and selected articles will be provided the opportunity for publication in the Malaysian Journal of Medical and Health Sciences, which is a Scopus indexed journal, thereby adding significant scholarly value to this conference. The oral and poster scientific presentations showcased emerging research trends and fostered meaningful scientific discussions, particularly among young researchers and students. The plenary sessions delivered by our distinguished speakers offered valuable insights into drug discovery, artificial intelligence, advanced drug delivery systems, pharmaceutical biotechnology, clinical research, and translational sciences. Our special appreciation to all speakers from IMU University, Malaysia for their collaboration and support extended to us and for making the event grand success. We would also like to express my sincere gratitude to our chief guests, session chairs, scientific session adjudicators, and advisory committee members for generously sharing their expertise and valuable time. We would also like to extend our sincere gratitude to our event sponsors, namely KC Overseas, Abroad Aspirants, Leo Overseas, Experts Advise Overseas, IDP Overseas Education, and Saptagiri Printers and Pharma Press Book Syndicate. Last but not least, the entire success credit goes to the whole team of chiefs worked for this event under the esteemed leadership of our visionary principal, sir. So for the first time, we are proud to announce travel grant to Dr. Vasudev Rao Avupati for international collaboration with IMU University, Malaysia, alongside with our management for their continued support in extending their helpful and grateful hands to provide all the needful support to achieve this milestone. Once again, thank you all.
Thank you, sir. And a very big round of applause to the organizing secretary. Now I request our principal to give his closing remarks.
Respected Mangai Garu, Secretary and Correspondent, RVRR College of Education. Respected Gopichand Garu, Member, Chiefs and NES. Distinguished guests for this valedictory function, Professor K.R.S. Samsura Garu, Vice Chancellor, Mangalayatan University, Jabalpur. Another distinguished guest, my mentor, brother, Dr. T.V. Narayan Garu, President, AP Private Pharmacy College Management Association, past President, Indian Pharmaceutical Association, Secretary, CR Farm Forum, and another distinguished guest today, Dr. Venkat Ikurti Garu, three dynamic leaders from IMU, Dr. Julfik Ali, Dr. Vasudev, and Madam, and another friends on the dais, Dr. M. Prasad Rao, IPA AP State Branch President, and our Secretary for this two-day conference, Dr. Venkat Rao, and IPA Education Division Chairman, the young and dynamic lady, Dr. Sumlata, dear students, participants, chairpersons for various sessions, speakers, invitees, warm greetings to all of you. First of all, on behalf of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences, I profoundly thank all the participants for making this event a grand success. Way back in 2007, in the same place, in the same area, when this college was just 2.5 years old, we have conducted a very big conference, that is first annual convention of Association of Biotechnology and Pharmacy in the same ground. And after that, the conference, we have conducted several conferences, but this is the very biggest conference after 2007 we have organized with more than, as per the statistics given by our secretary, it is 1,500. Out of 1,500, it is around 1,100 are offline. So we have to thank all the participants and all the college principals and faculty members and students who have taken active role in participating this. And this conference has definitely gave you some inputs about the novel areas of pharmaceutical sciences. We are all talking about artificial intelligence. We are all talking about personalized medication. We are all talking about the pharmacogenomics. And there is a controversy. What is the difference between pharmacogenomics and pharmacokinetic variabilities? And several novel thirst areas have been discussed by 10 eminent speakers. All these speakers have given insights about the novel pharmaceutical frontiers. Hope that all these topics will be definitely helpful to you for your future prospective research purposes. And I can definitely say that this pharmacy course is an evergreen course where no such impact on global economic crisis or whatever it is. So you might be thinking that artificial intelligence, if it emerges, several placements may be decreased. But in pharmaceutical sciences, it will never be like that. So we are going to use this artificial intelligence for upgradation of our platforms so as to enable the more refined form of the products can be developed by using this artificial intelligence. And also we can use this artificial intelligence to achieve the platform for data management and achieve the valuable data that is required for developing any research or whatever it is. So, artificial intelligence and several new areas of drug development will definitely boost us. Our course is highly research oriented, and we, the pharmacy professionals, particularly the students of pharmacy, should focus on higher education and research. But when it comes to our Andhra Pradesh state, it is highly pitiable situation that students after B.Pharm are so, they are simply going for small jobs and they are settling in that particular one, and after few years or few days, they are coming out with frustration saying that pharmacy is having no value, no professional outcome or prospective careers. So you have to focus on research, and if you succeed in the research, definitely you will be the one of the entrepreneur, you will be the one of the startup entrepreneur in pharmacy. And startups in pharmacy is very easy, and you can think in such a way that those lines will definitely help us and boost us in settling our career in pharmaceutical sciences. So with these few remarks, I once again thank all the participants for actively participating, and my special thanks and appreciation to all the students. This is very first time I am observing 70% of the auditorium is always filled with young and dynamic students, particularly girl students, and they are very silent, they are very keen, and they are very keenly noting the talks that are being delivered by several eminent personalities. We have to thank all the students particularly for maintaining such an utmost discipline and making this particular program more successful. Thank you one and all.
Thank you, sir. And again, one big round of applause to our principal for making sure that this two-day conference is happening. Next, I request Dr. P. Gopichan, sir, to please speak a few words on this occasion.
సభకు నమస్కారం. Chebrolu Hanumaiah Pharmaceutical Sciences, College of Pharmaceutical Sciences 2 రోజులు అంతర్జాతీయ సమావేశాన్ని జరపటం, దానిలో మలేషియా నుండి కొలాబరేషన్తో ఈ కాన్ఫరెన్స్ జరపటం జరిగింది. నిన్న ప్రారంభోపన్యాసంలో అతిథులందరూ కూడా ఈ కాన్ఫరెన్స్ ఉద్దేశాన్ని చెప్పటం జరిగింది. ఒక వాల్యూమ్ కూడా ఈ సమావేశంలో వచ్చినటువంటి వాటన్నిటినీ కూడా సంక్షిప్తం చేసి ఒక సావనీర్లాగా నిన్న విడుదల చేయడం జరిగింది. అది చాలా ఉపయోగం. దాంతోపాటు నేను ప్రిన్సిపాల్ గారికి ఒక చిన్న సూచన, 15 మంది ఈ కాన్ఫరెన్స్కి 1500 మంది స్కాలర్స్ రిజిస్టర్ చేసుకోవటం, చెప్పారు కదా సంఖ్య 1100, అలాగే 400 మంది ఆన్లైన్లోనూ వచ్చారని. మన ఎంఫార్మసీ కనీసం ఫార్మా డి, బి ఫార్మసీ విద్యార్థులు, ఫైనల్ ఇయర్ విద్యార్థుల్ని ఇనార్గరల్ సెషన్కు ఉంటే నిన్న కొన్ని వైస్ ఛాన్సలర్ గారు గంగాధర్ గారు, మధుమూర్తి గారు కొన్ని విషయాలని చెప్పారు. వీటికి సంబంధించినటువంటి సమావేశ ఉద్దేశాన్ని ఉద్దేశించి వాళ్ళు చెప్పారు. అవి కూడా వాళ్ళు వింటే ఈ ఇంటర్నేషనల్ కాన్ఫరెన్స్లో ఎలా జరుగుతుంది ఏంటి అనేటువంటిది వాళ్ళకి అనుభవం. మన కళాశాల హోస్ట్ చేయటం వల్ల మన స్టాఫ్కి అట్లాగే విద్యార్థులు అందరికీ కూడా ఇది ఉపయోగం. మారుతున్నటువంటి సమాజంలో వస్తున్నటువంటి మార్పుల్ని మీరు అందరూ గమనించవచ్చు. అవి మీకు ఉపయోగపడతాయి. అలాగే చాలా క్రమశిక్షణతో నిన్నటి నుండి విద్యార్థులే కాదు, మన అధ్యాపక బృందం అలాగే అధ్యాపకేతర సిబ్బంది కూడా ఇంత పెద్ద కాన్ఫరెన్స్ని సక్సెస్ చేయటం అంత ఈజీ ఏం కాదు. వాళ్ళందరూ కూడా క్రమశిక్షణతో పనిచేశారు కనుకనే ఇది సక్సెస్ అయింది. అలాగే వచ్చినటువంటి వాళ్ళందరికీ కూడా, బయట నుండి వచ్చినటువంటి వాళ్ళందరూ కూడా ఇక్కడ ఏర్పాటు చేసినటువంటి సమావేశ మందిరాలు కానివ్వండి, ఇక్కడ ఉన్నటువంటి 2 రోజులు స్టే వాళ్ళు చాలా కంఫర్ట్గా ఉన్నారని చెప్పి నేను అనుకుంటా ఉన్నాను. ఇట్లాంటి సమావేశాలు ఏర్పాటు చేయటం వలన అటు రీసెర్చ్ స్కాలర్లకి, అట్లాగే న్యూ ట్రెండ్స్, నూతన పోకడలను తెలుసుకోవడానికి, అట్లాగే విద్యార్థులకు కూడా ఉపయోగపడుతుందని చెప్పి నేను భావిస్తున్నాను. ఇటువంటివి నిర్వహించటం అనేటువంటిది పాలకవర్గం నుండి ఎప్పుడూ ప్రిన్సిపాల్కి వారి బృందానికి సపోర్ట్గానే ఉంటుంది. ఇంత మంచి కాన్ఫరెన్స్కి మరి చాలామంది బయట నుండి వచ్చినటువంటి వాళ్ళు ఇందాక కేఆర్ సాంబశివరావు గారు వైస్ ఛాన్సలర్ గారి ప్రసంగం విన్నాం. ఆయన చాలా వాస్ట్ సబ్జెక్టు ఉన్నటువంటి ఆయన, మల్టీఫెసెట్ పర్సనాలిటీ. ఆయన ఇక్కడ నాగార్జున యూనివర్సిటీలో అన్ని పోస్టులు చేశారు. ప్రస్తుతం మంగళయాన్ యూనివర్సిటీలో వైస్ ఛాన్సలర్గా ఉన్నారు. అంతకుముందు కేంద్ర విశ్వవిద్యాలయంలో కూడా వైస్ ఛాన్సలర్గా 5 ఏళ్ళు పనిచేశారు. మన మేనేజ్మెంట్లో అటు ఇంజనీరింగ్ కాలేజ్కి, జేకేసీ కాలేజ్కి కూడా ఆయన అకాడమిక్ కౌన్సిల్ సభ్యుడిగా ఉన్నారు. వాళ్ళందరూ కూడా మన కళాశాలని సందర్శించం. ఈ సమావేశాన్ని దిగ్విజయం చేసినందుకు వాళ్ళందరికీ కూడా పేరు పేరున కృతజ్ఞతలు. సెలవు.
Thank you, sir. Now, we would really love to hear from the trio of this conference, Dr. Mohammad Zulkifli, Dr. Chinswi, and Dr. Vasu. We would love to know how do you feel, what was your experience on now? So, would you please take a minute and come onto the dais and talk for a minute?
Good afternoon to all distinguished guests. Okay. First of all, I would like to say thank you very much, first and foremost, to the Institute Chebrolu Hanumaiah Institute of Pharmaceutical Sciences led by the principal, Professor Dr. Vidyadhara Garu, and also the organizing committee, not to forget the senior management of the university for organizing the conference and invited us to this beautiful country and wonderful place. And special for me that this is my first time in my life visiting India, as my wish almost 10 years ago. Honestly, I feel so amazing actually from the first day I touched down in Chennai airport and then moved to Deyawada, okay, airport, and maybe a little bit private story, okay. First thing that we will look for when we touch down here was to look for authentic biryani. Actually, I'm a very fan of biryani. So all my life in Malaysia, I went to so many restaurants offering biryani. But now I can enjoy the most authentic biryani in India. Okay. My impression, especially with regard to this conference, I fully agree that the conference was organized very successful. As mentioned by Mr. Principal, I'm very impressed to see the attention paid by the students, okay, in every talk that been delivered. Okay. And I also acknowledge the level of questions thrown to the speakers. Those questions really portraying the level of knowledge that the institutes have given to the students here. So you should be proud of yourself, okay? And as mentioned, I believe mentioned by the a few distinguished guests just now, that pharmaceutical sciences area or profession, there are much more things to be explored, especially with the capacity building that you have in this country. Each of you have a potential to grow up not only as a leader but also as a distinguished person in this pharmaceutical science area. Okay. And one day, when you reach that level, please remember me. So I think my last word is, I wish the institute, the students, the management of this institute to keep the excellent work, okay? And then I wish to the greatest success of this institute and to the students. Thank you so much.
Good afternoon, everyone, especially to all the distinguished guests. I'm very grateful for given this opportunity to attend this international conference. And actually, this is not my first time in India. I've been here for, I think, the third time. Actually, I spent around, 3 months during my PhD time in IIT Kharagpur. So I'm actually quite familiar with all the Indian culture. So, and I also very impressed with the students' performance yesterday during the cultural show. And I really appreciate all the effort that you have poured in for the show and for organizing this event. And we truly agree that what your principal has said, the active participation by all the students is really, really engaging, and it makes us feel very heartwarming, right? So it's really a good gesture from you that you actually take your time to attend this conference, right? So without your participation, this conference wouldn't be so successful, right? And the next thing is, I would also like to thank CHIPS Institute of Pharmaceutical Sciences for this opportunity, especially providing us a travel grant that we are able to travel here. And we truly looking forward for future collaboration, and in case if there's any chance for us to cross path our future, especially in student mobility program, and if you would like to come to Malaysia, do visit us in IMU University in Bukit Jalil, Malaysia, which is in Kuala Lumpur. Right? There is also a very lovely country, same as India, full of all the lovely people around, and same as Indian, okay? Because I do have many Indian friends from Karpur, but unfortunately, many of them, they are no longer in India because they have already explored in other countries due to their career advancement. Right. I think I would end my session here. Before I end my session, I would like to wish you all the best in your future undertakings, and hoping to see more international conference organized by CHIPS Institute of Pharmaceutical Sciences. Thank you very much.
A very good evening to all the distinguished dignitaries on the dais and also off the dais. And also a very good evening to all the participants, dynamic young researchers, faculty members of pharmaceutical sciences. First and foremost, I would like to start with giving some short background about this great event. As you all may not be knowing, what would be the groundwork for bringing Malaysian citizens and academic leaders and researchers to this event all the way from Malaysia to on-site participation in this great event. So this inception is from Professor Dr. S. Vidyadhara. Give him a great. When I was first communicated by the organizing secretary, Professor Dr. Venkata, the first word he mentioned, Vasu, we would like to organize a big event. We are really looking forward to have a great dynamic leadership personalities and good researchers who are really can enhance our students and motivate our students, and who can deliver a very good enhancing eminent speakers we want. Can you do that? Then I immediately, without any hesitation, I mentioned, I can do that. So, thank you. So that is how the initial discussion started. When I explained about them, so what is the expectations you have? The very first beginning, the Professor Venkata, he mentioned, we really don't have any expectations. We want the great section plenary talks and scientifically standard and eminent personalities. When I was thinking about the personalities, the first person came into my mind is our former Dean School of Pharmacy and Quality Director, International Medical University, formerly now IMU University, Professor Muhammad Julkifli. So, since he is a foreign citizen, you might not be knowing much very well about it. You can just Google about him. You will understand the complete profile. He is also the member of the Malaysian Pharmacy Board, same like our PCI He is the chief member and also the Malaysia Qualification Agency Scientific Committee member as well, and so many other regulatory bodies. He is one of the distinguished person in national level in Malaysia as well, and collaborated in several scientific communities across the globe. And he successfully completed his PhD, postdoc, B Pharmacy, everything from Japan. And he did postdoc fellowship in USA. So he has a very great meritorious record. I found he is the right person to motivate our Indian students. So that's the how, just would like to thank for accepting our invitation. The moment I mentioned Chebrolu Hanumaiah Institute of Pharmaceutical Sciences, without any second thought, honestly, in front of him I am saying, without second thought, I am ready. I never been to India, Vasu, can you bring me to India? I want to interact with students. So, such a great personality. Then next, not less than almost equal personality. You can also, just now she mentioned, she is actually an intern student in IIT Kharagpur. None other than Dr. Chinsui, who is also the MSc in Analytical and Pharmaceutical Chemistry program in Malaysia, program director. She is responsible for the postgraduate studies in our IMU University School of Pharmacy. So she is also actually, to be honest, because this kind of things will really motivate. Honesty reduces 90% of problems, I feel. So when I asked about why don't you participate, you are very young and dynamic and students love to interact with you, then she said, because 3 children, so very busy person. Also, all postgraduate programs are started and currently running, even though she managed to succeed in joining us on site and trusted our security, everything, and participated all the way. So, thank you. I extend behalf of you all, an Indian community, pharmacy, professional community, I would like to thank these 2 Malaysian citizens for being with us. So this is one part of the story. And when it comes to the event organization, Professor Venkata and Professor Dr. Our principal, Vidyadhara sir, you can bring as many as people, I will give you the travel grant for you. This is what I received. As many as people. They never restricted me, you bring only 2 or you 3, only 1 person. They never mentioned like that. So that is the motivation and that is the enthusiasm they have towards the enhancement of the student competencies, further to provide some additional opportunities to the students, sharing sections from the foreign dignitaries so that the networking will be there and you further associate. Yes, I just know Professor Jule Kifli already sent email to me. I can show to you if you want. Someone asked for the student mobility program. Immediately he put me in the CCs and we will keep in touch and further explore. So that's the message. So this is the main objective. This is the main objective, to encourage, to motivate, and finally to support students. So totally for the sake of students. Totally for sake of students. Okay, so once again, give them these great dignitaries on the dais and off the dais. A big round of applause. So they are all dynamic leaders, educational experts. I have no that much level to speak in front of them. Just only I have some enthusiasm to learn and work together, collaboratively grow together. So that's all. What I feel, this is like a lifetime opportunity. So most memorable event. I never participated in such a big event before, to be honest. I participated more than 50 conferences, but never seen this much crowd gathered and sit exactly listening to every single presentation very, very keenly. So that is the interest you all have. So surely, you all will reach to greater heights. As Professor Muhammad Julkifli mentioned, you all will receive, I wait to receive some good messages from you one or the other day. Okay. So once again, it's my immense pleasure to be part of this great team. And once again, thank you, Chebrolu Hanumaiah Institute of Pharmaceutical Sciences Senior Management, and also all the School of Pharmacy administrators, and also to all the participants and all the team members, press and media who work directly or indirectly for the sake of grand success of this event. Thank you once again, all.
Thank you so much, sir. Once again, on behalf of our institute, we thank all these three people for traveling all the way from Malaysia to be here with us on these 2 days. Thank you once again, and to say terima kasih, if I'm not wrong. And now, I request our principal, our organizing secretary, and also the dignitaries on the dais to please come forward to present the certificate of travel grant to Dr. Vasudevarao Avapati, and I also request the other two speakers to please join. So our institute is presenting the travel grant and also the logo design to Dr. Vasudevarao Avapati. As they mentioned, we made sure that they travel all the way here and we provided them the travel grant, and we would like to do this on this valedictory stage. Now I request our chief guest, Professor K.R.S. Samsarao Garu, to address the gathering.
Good evening, everyone. Respected guests on the dais, Professor Vidyadhara, principal of the institution, and our good friend Professor T.V. Narayan Garu, Organizing Secretary Venkatrao Garu, and friends from Malaysia, because I don't know the names. Friends, I call them as friends. And Gopichand Garu, very good friend of mine. Prasad Rao Garu, Venkat Kurti, Sumalata. And in front of the dais, Mangai Garu, a very towering personality, very senior academician in Guntur. He was professor in Delhi Venkateswara College almost more than 50 years ago. So he did chemistry and he worked as a chemistry professor in Venkateswara College almost 50 years back. And he left that job and came to Guntur, and he has started business on his own. And every conference he interacts with everyone. Even today with me also interacted for some time. And very critical questions he used to ask in any of the conferences. Though he is basically a chemical engineer, he shows interest for everything. So thank you very much, sir, for your gracious presence. And guests in front of the dais, faculty members and student friends, the most important part is my observation. The last lecture of this conference, I have seen a big gathering here. Normally, I see up to the last row of the auditorium how students are responding to my lecture. Because if students are not responding, I will stop my lecture there. But I have been keenly observing every student from here, and every student has been showing a lot of interest in listening to me. I really surprised because in many conferences I have given lectures, but actually students will be some kind of murmuring, some kind of talking, side talkings, and and chit-chat with friends. This kind of things will be there. I have not seen a single person chit-chatting. This is greatness of the students. I have to appreciate these college students. Wonderful performance and wonderful academic interest. It is actually I have not seen anywhere this kind of pin drop silence. And I could see some of the girl students sitting in the second, third row. When I am talking about the subject which is not known to pharmacy students, they are waving their head like this. I have seen some students waving the heads like this. The topic which I have given is not known to many people, even they have not heard, but they are waving heads means they are listening very keenly. And I hope that you might have understood that is the wonderful new innovative topic for the students, which actually you have to change your ambitions. Don't be like industrial pharmacist going for looking for a job in industry of pharmacy. Pharmacist has got wonderful opportunities in many areas. You have many opportunities already existing, but you are not showing interest to go there. You are only showing interest to go to pharmacy industry, going Hyderabad and working in a pharmacy industry. That is not the way. You have to think about various avenues like hospital pharmacy, community pharmacy, chain pharmacies, industry pharmacies, and clinical research pharmacies. Many things are there. And all this actually, which I added 2 or 3 things in the afternoon lecture about pharmacogenomics and omics technologies and even drug discovery development. Actually, you have to have a collaborative network with many people actually in changing your interest, changing your attitude. And there is no bar that you cannot enter into any of the area. Internationally, now education has become in such a way that there was no bar. You can study anything. You can do anything. I'm a zoologist. I did PhD pharmacy. I did psychology. I did computer science. I did MBA. So there is no bar for your study. Anything you can study, whatever the interest you have. So even up to age 65 years, I've been doing studying. Even last 2 years back, I did MBA. And 2 years back, I did MSc Psychology. This is no bar for your study. You have to learn. But the students who actually been listening today, I am very much confident that you are having a bright future. Not because you are studying only in chips. I am not talking, it is an added point. But actually, the way students are listening today, I have been seeing everyone. There was no chit-chat in that last gents also. I have been observing the gents sitting there, but they are also not talking. Ladies here, they are listening very keenly, but gents also not talking. I have observed very clearly. I congratulate and really well impressed by your behavior. So, please remember, don't stop your academic zeal, academic thirstiness. And you have a zeal, nothing is impossible. If you want to become Nobel laureate at your age, you take a decision, you will become Nobel laureate. I cannot take a decision that I want to become a Nobel laureate. I cannot become, my age has really been grown. Now I cannot take a decision to become Nobel laureate. You take a decision now, 20 years child or girl, take a decision, I want to become Nobel laureate. You will definitely become Nobel laureate. I took a decision to become Vice Chancellor long back when I joined into university. Now I became Vice Chancellor for 3 universities. So this is what actually one need to have I don't have a Nobel laureate zeal that time. I'm telling now that you take a decision to become something Nobel laureate, you become. Good pharmacist, good entrepreneur, to establish a company. So practical training is very important. Just listening to the theory to your professors is not possible to become something. Innovation is required, creativity is required, and the incubation is also very much essential. So you need to be motivated, you need to get proper training, you need to go for industry training every year. Pharmacy first year student go every year in a pharmacy industry. Try to get more knowledge as much as possible. Utilize the opportunities. Your principal is well connected. So you can go to any industry, every year one industry. Get yourself trained well. Get your innovative knowledge and become as one of the best pharmacists or best researcher, best Nobel Prize winner. Thank you very much and good luck for everyone.
Now I request Dr. P. Vijeta to introduce our distinguished guest, Dr. T.V. Narayana.
distinguished guests on the days, off the days, esteemed delegates and honored faculty members, a very good evening to one and all. And I really feel it as a privilege to introduce our distinguished guest, Dr. T.V. Narayana sir, President, AP Pride Pharmacy College Management Association, and sir is Director, Vikas Institute of Pharmaceutical Sciences, Rajahmundry. And sir has almost 30 plus years of experience in this academic and entire pharma professional, and he is the man behind many associations of our pharmacy background. And sir played a pivotal role in shaping pharmacy education and professional practice in India, serving the pharmacy fraternity through various prestigious organizations. Notably, he served as president for the prestigious Indian Pharmaceutical Association, providing visionary leadership and strengthening professional standards across the country. And he is a prolific academic mentor for many of the postgraduates and published several articles. And among his achievements, the major achievement, he is one of the having the international recognition such as FAPA Ishidate Award 2020 under the Pharma Education category. With this brief introduction, I'd like to welcome today's distinguished guest, Dr. T.V. Narayana sir, to give his address. Thank you, sir.
Thank you, madam, for the nice introduction. Respected chief guest of today's valedictory function, Sri K.R. Samsura garu, and veteran member of RVR College of Education, Sri Mangai garu, another member Gopichand garu, my professional friends from Malaysia, IMU University, Mohammad Julfikil, Dr. Chin Sri E, and Vasudev, and Venkat Kurti, and President of Indian Pharmaceutical Association, Andhra Pradesh State Branch, Dr. M. Prasad Rao, and Chairman of IP Education Division, Dr. G. Sumlata, and Organizing Secretary of this Innovate Pharma, Dr. Venkat Rao, and Principal, my brother, Dr. Vidyadhara, of IPA, past Chairman of Education Division, and members, principals of various colleges and pharmacy fraternity, and my dear students, good evening. Good evening. Don't worry, I will not take much of your time. I will leave as early as possible. First of all, my sincere thanks to our IMU professional colleagues for accepting the invitation of the local organizing committee and coming here and sharing their thoughts and knowledge with all our Indian students from particularly from the state of Andhra Pradesh and other areas. My sincere thanks to all three of you, and my congratulations to Dr. Vidyadhara, Venkatrao and team, the local organizing committee, for making commendable arrangements for organizing this Innovate Pharma, that is the two-day international conference. And also the students, that's what first time in any valedictory function, I am seeing big number of students or delegates, because even in IPC it is very hard to find more than 500. Initially it will be 10,000. At the time of valedictory hardly it will be 200 or 100 plus. Here that shows the interest of the students. As Samsaragar rightly pointed out, pharmacy students, patience is the main. You have that patience, definitely you will be the successful professional pharmacist in your future. That's what I can say proudly. And coming to the topic, really it is impressive, the need of the day. Innovate Pharma, emerging innovations in the pharmaceutical sciences and drug discovery. I think the experts might have briefed you about what are the emerging technologies like artificial intelligence, machine learning, how we have to adapt, 3D printing, and also gene therapy, personalized medicines. So many things are going to come. That is what predicted by the future experts from industry. To the market may not come to the market. That's why our people are not taking interest or risk in the research. With our reverse engineering skills, with our thorough knowledge in chemistry, we are able to achieve the number 3 position. That's why what world is calling India is the pharmacy of the world. But vaccines, we can proudly say, we have proved, because latest COVID vaccine, if you see, that was from Hyderabad, Krishnayalla. He has come out with the vaccine. Vaccine definitely we proved we are having that expertise. Rota vaccine, again from the Krishnayalla, that is thanks to the hard work done by Varaprasad Reddy, Santa Biotech, and Krishnayalla Bharat Biotech. How that vaccine, why I am telling you what is the importance of drug discovery and research, it no need, మీరు పెద్ద పెద్ద ల్యాబ్లు పెట్టుకొని పెద్ద కోట్ల ఇన్స్ట్రుమెంట్స్ అవసరం లేదు. What Krishnayalla did, generally vaccine, same thing Serum Institute, Covishield, that is the chimpanzee vaccine. They injected the COVID virus and taken out the antibodies and prepared the vaccine. That was the routine practice. But the idea what Krishnayalla got is, he has prepared the structure of the vaccine with the help of Institute of Virology from Pune. Luckily, 100% structure they are able to generate and given to Krishnayalla. He has just broken down one chain link. That's what he killed the vaccine. That is simple. That's why he offered, he can supply the vaccine for a less than a distilled water bottle. That the research done at his own lab, and more than 10 pharmacy students worked there. I know all of them. I visited those industry, and this was stopped by the experts because world has not taken Covaxin in the beginning. They said they will not approve the Covaxin. They approved the Covishield, Moderna, all those vaccines, because he has used artificial intelligence for the post-market surveillance study. That are not able to accept. Afterward, they have accepted. That's what research. Actual research starts at university level or institution level. It is not at big R&D centers. If you take any drug has come from the small laboratory. So you have to concentrate on research. Any spark from you, it creates wonders in our life. Because now I am happy to know they have a MOU with IMU University. Collaborative works, you can go ahead. What are the latest development taking place in the world? What is the technology they are adapting for drug discovery? You can go ahead, because 2027, $50 billion worth of drugs are going to be patented. It is a very big business. If any students come out with your reverse engineering skill, definitely each molecule will get more than $100 million. That is the maximum research going on at various institutions. That's what I suggest you people. Manam cheyalem ani em ledu. Guarantee one spark will give the result. So concentrate on research, take the help of the teachers, take the help of the experts from other universities and other countries. That's what I want to suggest to you people. And this emerging innovations, majority of the work is going to be rooted through artificial intelligence and machine learning. Because it was not included in our curriculum. We have to add on programs, we have to undertake training, and we have to implement this in our research level, at our institution level. And pharmaceutical sciences is entering transformation era from the year 2025 with cutting edge technologies and innovative procedures for drug discovery. It is going to reshape industry from personalized medicine to artificial intelligence. These are some of the areas, because no one drug fits all. That is the future says. Earlier COVID-19 time everybody used to take Dolo 650. But your body may not require 650. Your body may require 500. That's what going to come in future. That's what people call personalized medicines. 3D printing. They are going to customize the tablets as per your body needs. And future, our genome map is going to come, like our MRI scan and X-ray. Even your body gene map is going to come. And pharmacists are going to prescribe how much quantity of drug is required for your body on basis of gene therapy. That is what going to come in future. These are some of the areas you have to update yourself. This was not included in curriculum. You have to attend the seminars and conferences organized for this specific purpose, where you can interact with the experts, where you can update yourself with the latest development taking place. That's what I want to advise you people. Dear friends, dear pharmacists, future pharmacists of our nation, it is a time to prove or perish, because the entire scenario of pharma industry is going to change in future. No more reverse engineering going to take place or survive in future. It is time to prove or perish with your research, with your intellectual property rights. And this is the time to implement or make use of the technology available. Here I will quote the great words told by His Excellency Dr. APJ Abdul Kalam in 2004 IPC at Chennai. Technology is important tool to give fast health care. Technology is important tool to give fast health care. You have to use it. That time nobody knows what is the AI, what is ML. But when we approached him to invite him for the IPC conference inauguration as a chief guest, he told, what is the role of your pharmacist in future? And he told, why you are not adapting the technology to meet the future needs? And we really, pharmacists, we were shocked. And he asked us, he come out with a Pharma Vision 2030 that made the Pharmacy Council and IPA prepared the Pharma Vision 2030 and came out with that. That's how the experts predict. That's what I want to advise you people. Make use of the technology available and be at least one Krishna Yalla or one Kiran Majumdar I want to see from the audience in future. Kiran Majumdar, now Bayakan everybody is telling, she is the dynamic lady of pharma industry. Then when I met her in 30 years back, she is doing research on papaya. ఇదెవర్రా బొబ్బాస్కాయలు తీసుకొని రీసెర్చ్ చేస్తుందని నవ్వుకున్నాం మేమందరం. Afterwards we were not knowing the intermediates has created crores money. That was the success of her. That's what I am telling you. Their spark will come if you concentrate seriously on research. I wish concentrate on research also. In addition to the academic curriculum, concentrate on research. Definitely we are going to prove to the rest of the world that we have an excellent brain when compared with other any national. Our brain we are using for other activities. Multi-talented. ఎందుకంటే బైక్ తొక్కుతూ కూడా మొబైల్ మాట్లాడగలిగేది మనమే. బయట ఏ కంట్రీలోనూ మాట్లాడలేరు. ఇమ్మీడియట్గా దే విల్ స్టాప్ అండ్ దే విల్ స్పీక్. ఒక 10 మంది వస్తే ఫైట్ చేయగలిగేది, అరవగలిగేది అందరితో మనమే. బయట అరుపున పడితే పారిపోతారు. సో దిస్ మల్టీ టాస్క్ టాలెంటెడ్ individuals, you are diverting unnecessarily to the other areas. Please concentrate. That's what I want to advise you. I want to see some of you to succeed in your research and bring laurels to our Indian pharma industry. With these few words, I thank the organizers for giving me the opportunity. I wish you all success in future. Thank you.
Thank you, sir. And before we proceed on to the Best Poster Awards and Best Research Awards, we have a very short address by one of our guests, Dr. Venkati Kurti, and I request him to please address the gathering.
Thanks for giving this opportunity. Just now, Shar mentioned about the Indian pharma industry. So everybody across the globe is looking for India because highest English literate people, highest 80% of people knows about computers. Intellectuals are here, but the problem is Indian students are very, very, very intelligent. But I am sorry to say they are not honest. Because I am in US, I am US citizen. For the last 30 years I am in US, working with so many universities in US also. Their GRE exams, they are not taking the examination. TOEFL, no. Somebody is taking the examination. Don't do any unethical works. Because of in Trump era, everybody is thinking about problem. I am telling the word is, if you have skill, nobody is going to stop you. If you don't have skill, nobody is going to help you. So better to sharpen your skills. AI is the game changer. For next morning onwards, I am keep on talking the same thing. Game changer for it. Better to sharpen your skills first. New pharmacy syllabus. There are 6 papers related to artificial intelligence, Python, blah, blah, blah is there. That means government of India also recognized, Pharmacy Council also recognized to add those courses. So better, probably it going to implement from next year onwards. Better to sharpen your skills with AI and everything. And also like, finally I can say one thing, select right guru first. KRS Garu mentioned about right guru. Select right guru first or right institute first. If you are at right guru, you never did anything wrong. If you are not at right guru, whatever you are doing is wrong. If you are at right institute, no, you never did wrong. So Tom, think about right institute, right guru. That's the most important. And finally, success and excuses never talk together. If you want to get a success, don't give any excuses. If you are giving excuses, you never get a success. Thank you.
Thank you, sir. And now we have the prize and the award distribution. And to do so, I request on stage Dr. P. Malleswarao and also Dr. R. Sasidhar onto the dais. Sorry, my mistake. Before we go to the award presentation and prize distribution, we would like to felicitate the chief guest and distinguished guest who made their time and present with us during this valedictory function. So I request our principal, the distinguished guest on the dais, to please come forward as we felicitate our chief guest and distinguished guest. First, I request Dr. First, I request Professor K.R.S. Samsarao Garu to please come forward and accept the felicitation. Next, I request Dr. T.V. Narayana Garu to please come forward and accept the felicitation. Now I also request the dignitaries on dais to please provide a token of memento to Dr. M. Prasad Rao and also to Dr. G. Sumalata. First, I request M. Prasad Rao sir to come forward and accept the token of memento. Now I request Sumalata ma'am to please come forward and accept the token of memento. I also request the dignitaries on the dais to please provide a token of memento for the following people, and I request them to please come on the dais and receive the token of memento. First, I request on stage KC Overseas Education to please come forward and accept the token of memento. Next, I would like to invite on stage abroad aspirants. Next, I would like to invite on stage Leo Global Overseas. Next, I would like to invite on stage Experts Advised. Next, I would like to invite on stage IDP Overseas Education. We thank all these 5 organizations for their active sponsorship for this conference, for setting up the stalls, and also made sure that the delegates had good time at their stalls with exciting activities, trying to make them some playing carom board, giving them some targets, giving them some chocolates, and we thank them for actively taking part in this today conference, and thank you so much for your contribution. And now I invite Mallesrao sir and Sesi sir to please do the proceedings.
Good evening to one and all. Normally every conference creates knowledge, but only few moments that creates memories during the conference. So this is one such moment at Innovate Pharma 2026, where we recognize the young talents and young minds for their valuable contribution that truly reflects the innovation in pharmaceutical sciences. So during this conference, 650 e-posters, oral presentations, online and offline were presented, and it was a big task for the scientific committee to decide the winners. So without further delay, now I request my colleague Dr. Shashidhar to announce the most awaiting moment of this valedictory function, that is the prize winners for the e-posters and the oral presentations. Thank you.
So good evening all. Now the time for announcement of winners of this scientific oral bar e-poster presentations. So we are giving 10 first prizes for the winners. For these winners, we are giving a 2,000 rupees of cash prize, a memento, and a textbook along with the appreciation certificate. Okay. Now we are inviting the winners college-wise, and I also request the dignitaries on the dais to award the prizes for the winners. First, JVN Shravya Devi from A.M. Reddy Memorial College of Pharmacy. She has secured the second best prize for her presentation. JVN Shravya Devi from A.M. Reddy Memorial College of Pharmacy. And next, B. Sukanya from ANU College of Pharmaceutical Sciences. She has secured the second prize for her presentation. A memento, a textbook, and appreciation certificate will be awarded to her. B. Sukanya from ANU College of Pharmaceutical Sciences. So any representative from ANU College of Pharmaceutical Sciences can come on to dais and take the prize. V. Renuka from ANU College of Pharmaceutical Sciences. V. Renuka from ANU College of Pharmaceutical Sciences. She has secured the third best prize for her presentation. Okay, now from ASN Pharmacy College. So the first prize goes to the Esther Balaguri. She has secured the first prize for her presentation. A cash prize of 2,000 will be awarded to her along with the memento, textbook, and appreciation certificate. Esther B from ASN Pharmacy College. And K. Roshini from ASN Pharmacy College has secured the second best prize for her presentation. K. Roshini from ASN College of Pharmacy. And B. Veeranjali from ASN Pharmacy College also secured the best consolation prize for her presentation. A textbook and appreciation certificate will be awarded to her. B. Veeranjali. And next, the prize is from Bapatla College of Pharmacy, M. Sukanya. M. Sukanya from Bapatla College of Pharmacy. She has secured the second best prize for her presentation. Anybody from Bapatla College of Pharmacy? Or any representative or faculty member? Okay, next from Chalapati Institute of Pharmaceutical Sciences, D. Dakshayani. D. Dakshayani from Chalapati Institute of Pharmaceutical Sciences has secured the third best prize for her presentation. Next from CHIPS, that is our Chebrolu Hanumaiah Institute of Pharmaceutical Sciences, M. Kirti. M. Kirti has secured the first best prize for her presentation. She has been awarded with a 2,000 rupees of cash prize, memento book and an appreciation certificate. Next, B. Rishika from CHIPS. She also secured the first best prize for her presentation. Next, A. Sri Lakshmi from CHIPS has secured the second best prize for her presentation. She will be awarded with a memento book and appreciation certificate. Next, K. Pujita Manikanteswari from PharmD of CHIPS. She has secured the second best prize for her presentation. And D. Hemalata from PharmD. D. Hemalata from PharmD has secured the third best prize for her presentation. D. Hemalata or anybody on behalf of her can take the prize. Next, Tamizullah Khan from PharmD has secured the third best prize for his presentation. Tamizullah Khan. Next from NRK and KSR Gupta College of Pharmacy, B. Pravachana has secured the second best prize. Or anybody on behalf of her can take the prize. Next, T. Arun Kumar from NRK KSR Gupta College of Pharmacy has secured the third best prize. T. Arun Kumar from NRK and KSR Gupta College of Pharmacy. Next, the prize for the IMU College from Malaysia. So, Jing Liang has secured the second best prize for his presentation. Jing Liang. Sik Jing Liang.
Sik Jing Liang from Biomedical Sciences University School of Health Sciences, IMU University.
Now I request Vasudevara sir to accept this prize on behalf of the student. Next, from Jagan Institute of Pharmaceutical Sciences, K. Prabhu Kumar. K. Prabhu Kumar has secured the first prize with a cash award of 2,000 memento, book and appreciation certificate. K. Prabhu Kumar from Jagan Institute of Pharmaceutical Sciences. And D. Soumya from Jagan Institute of Pharmaceutical Sciences. D. Soumya or any representative of Jagan College can get the certificate. Yeah. Next, from KL College of Pharmacy, KL University, K. Reshma Gayatri has secured the second best prize for her presentation. K. Reshma Gayatri. T. Arun Kumar. Gupta College hai kada? Jagan School, sorry. D. Soumya. Sir, Suresh sir, Dr. Suresh sir, so please come on to dais and take the award, sir. Next, from KL University, K. Reshma Gayatri and M. Mahima Swarupa. KL College of Pharmacy, K. Reshma Gayatri has secured the second best prize, and M. Mahima Swarupa has secured the third best prize for their presentations. Next from KVSR Siddhartha Institute of College of Pharmaceutical Sciences, A. Rishika has secured the first best prize for her presentation. A. Rishika. K. Renuka Chaudhary from KVSR Siddhartha College of Pharmaceutical Sciences has secured the second best prize for her presentation. And P. Durga Malleshwari of KVSR Siddhartha College of Pharmaceutical Sciences. Anybody from KVSR? M. Rishika from KVSR, K. Renuka Chaudhary and P. Durga Malleshwari. KVSR. Next, from MAM College of Pharmacy, R. Soundarya has secured the second best prize for her presentation. R. Soundarya from MAM College of Pharmacy. Next from Manipal College of Pharmaceutical Sciences, Mahe, Tanvi Panigar has secured the first prize for his oral presentation. Actually, it is an online presentation. So we will forward the cash prize of 2,000 and memento to the college shortly. Next from Nirmala College of Pharmacy, C.H. Lahari from Nirmala College of Pharmacy has secured the second best prize for her presentation. And K. Bhavya from Nirmala College of Pharmacy. K. Bhavya from Nirmala College of Pharmacy has secured the third best prize for her presentation. Next from NRI College of Pharmacy, J. Lulika Kumari has secured the first prize for her presentation. She will be awarded with a cash prize of 2,000 memento and book. J. Lulika Kumari from NRI College of Pharmacy. And J. Durgambika from NRI College of Pharmacy has secured the second best prize. Okay, next we go to the Santiram College of Pharmacy. N. Venkateshwar Reddy. N. Venkateshwar Reddy from Santiram College of Pharmacy. Next, from St. John's College of Pharmaceutical Sciences, K. Shainaz Banu has secured the second best prize. K. Shainaz Banu from St. John's College of Pharmaceutical Sciences. Okay. Next, from Vignan Pharmacy College, Guntur, T. Saimani has secured the first best prize for her presentation, and she will be awarded with the cash prize of 2,000 memento and a textbook along with the appreciation certificate. P. Sivakrishna from Vignan Pharmacy College has secured the second best prize. T. Saimani has secured the first prize, and P. Sivakrishna from Vignan College of Pharmacy secured the second prize. And Shubham Kumar from Vignan University, I think, Shubham Kumar has secured the third best prize for the presentation. Next, M. Lakshmi Banusri from Vignan Institute of Pharmaceutical Sciences, Vizag. Next, from Vignan Institute of Pharmaceutical Technology, Vizag, M. Lakshmi Banusri has secured the first prize for the presentation. Any representative from Vignan Vizag? She will be awarded with the cash prize of 2,000 memento and appreciation certificate. Next, S. Alekia from Vignan Institute of Technology, Vizag has secured the second best prize. And K. Lakshmi Monica from Vignan Institute of Pharmaceutical Technology, Vizag, has secured the third best prize. And S. Nilima Maheshwari from Vignan Institute of Technology has secured the third best prize. Next, from Vijaya Institute of Pharmaceutical Sciences, M. Asmita M. has secured the second best prize for her presentation. M. Asmita. And J. Leena Jessica has secured the third best prize for her presentation. M. Asmita from Vijaya Institute of Pharmaceutical Sciences. She will be awarded with a memento, a textbook, and appreciation certificate. And J. Leena Jessica from Vijaya Institute of Pharmaceutical Sciences. Next, from Vishnu Institute of Pharmaceutical Education and Research, Hyderabad, M. Swati has secured the first best prize for her presentation, and she will be awarded with a cash prize of 2,000, memento, book, and an appreciation certificate. Leena Jessica, yeah. Vijaya Institute of Pharmaceutical Sciences. Vijaya Institute. Next, M. Swati. Next, M. Swati from Vishnu Institute of Pharmaceutical Education and Research. Next, from VJ College of Pharmacy, B. Divya Sri Lakshmi. From VJ College of Pharmacy, B. Divya Sri Lakshmi has secured the second best prize. Any representative from VJ College? Okay, now we move on to the prizes for the cultural events. So as a part of this two-day international conference, Innovate Pharma, we have organized the cultural dance programs. And the winners of this cultural dance program, the first prize has been combinedly awarded to Chebrolu Hanumaiah Institute of Pharmaceutical Sciences and KVSR Siddhartha College of Pharmaceutical Sciences with a cash prize of 4,000, appreciation certificate and medals. First I call upon the team of CHIPS to take the prize, memento, medals and certificates. The team of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences for the cultural award. Next, I call upon the team of KVSR Siddhartha College of Pharmaceutical Sciences. They will also be awarded with a cash prize of 4,000 along with the memento, appreciation certificates and medals. Team KVSR has secured the first prize with a cash prize of 4,000 and mementos. Next, the runners are the team Vignan Pharmacy College. They have been awarded with a cash prize of 2,000, memento, certificates and medals. I call upon the team Vignan Pharmacy College for their prize in the cultural group dance competitions. So team Vignan College. Sir, one more, sir. So, D. Soumya from Jagan College of Pharmacy. She was missed when we announced. Now she is with us, sir. Okay, sir. Thank you.
So, just attention please, sorry to disturb. This is our student Sik Jing Liang from School of Biomedical Sciences, School of Health Sciences, and he is the graduate of BSc Biomedical Sciences Honors in IMU University. So, he secured the best prize. So, once again, I would like to congratulate our student Sik Jing Liang. Thank you for all the support.
Thank you, everyone. Thank you, everyone. My sincere request to all the dignitaries on the dais, I request you to please sit down for 8 minutes. Just for 8 minutes. My principal asked me to conclude this vote of thanks in less than 5 minutes. But I strongly believe in the point that appreciation is required for anybody to make sure that their work is appreciated and they work hard for the next thing. So this event happened for 2 days. That is what we saw. But the groundwork for this event started 2 months ago. Just imagine the work that is being done for 2 months for accomplishing the task for just 2 days. ఉండటానికి 18 అధ్యాయాలు 700 శ్లోకాలు ఉన్నా సరే భగవద్గీత అంటే చాలా మందికి గుర్తొచ్చే ఒక శ్లోకం ఏంటంటే కర్మణ్యే వాధికారస్తే మా ఫలేషు కదాచన మా కర్మఫల హేతుర్భుః మాతే కర్మోత్సవ సంగమణి. తప్పు చెప్తే ఏమన్నా సారీ, బట్ ఆ శ్లోకం అర్థం ఏంటి అంటే మన కర్మ మనం చేయటానికి మనకంటూ ప్రత్యేకమైన కారణం అంటూ ఏమీ ఉండకూడదు. మనం కర్మ ఎందుకు చేయాలి అంటే అది మన బాధ్యత కాబట్టి. ఇంత పెద్ద and I request the dignitaries, I may be losing my mother tongue as I do this vote of thanks, please excuse me. So to do this 2-day conference, there will be a lot of questions in your mind, whether this will be successful, whether will it reach the mark that we are expecting to, will the delegates come, if they come will they listen, if they listen will they implement. Too many doubts, but there needs to be a motivating factor for everybody of us to cross these doubts, to come across these hurdles and work for that cause. And I think every staff member, every non-teaching member and a volunteer of this conference did that. And I perfectly believe that they made justice to Shloka by saying that we did our work based on our karma, not because we will be appreciated, not because this will be a super success, because we don't know what is the outcome is. We never know what the outcome is, but there has to be a reason for working, and that working is our responsibility. So one big round of applause for all the people who worked there. And I'll quickly read out the names, tell you what work they did. First comes the registration committee. The registration committee includes Ramu sir, Vijeta ma'am, Sandeep sir, Ravichandra sir and Shravani ma'am. Before you clap, just know what work they did before you clap. It is not easy registering 1100 plus participants. It is not easy writing certificates. It is not easy segregating all these works. It is not easy to make sure that everybody is registered and everybody is processed. So one big round of applause to all these people. Ramu sir, Vijeta ma'am, Sandeep sir, Ravichandra sir and also Shravani ma'am. Ramu sir is actually fighting with fever. I hope he's well. I don't know where he is. Next comes the scientific committee. According to me, this is the most difficult committee because they will be receiving abstracts from different parts of the world in different fonts, in different font lengths, in different abstracts. Something will be organized, something will not be organized, something will be coming in different font style, something will be covering in different style, but they have to make sure that every abstract is synthesized and extract so that they have to prepare the souvenir. And the people who did that hard work are Sasidhar, Mallesrao sir, Haritha Pavani madam and Prachet sir. A very big round of applause to all these people. I'll come back to you later. Last time I compared Haritha Pavani madam with Arundhati. This time I want to compare with a small character in Shankar Dada MBBS. అందులో ఒక ముసలాయన ఉంటారు. క్యారమ్స్ ఆడుతుంటే నిద్రలో ఉన్న ఆయన కూడా లేచి వచ్చేస్తారు. Actually, madam had a very big bad fracture to her hand. Whenever we said there is a conference, she slowly walked into the college, sat in her room. Just as the way that she did it. One big round of applause to her. And the newest addition to this is Prachet sir. Sir, సరదాగా అంటున్నాను, ఏం అనుకోమాకండి. ఏదో సినిమా డైలాగ్ ఉంటుంది, సెంటీమీటర్ చనువిస్తే కిలోమీటర్ దూసుకెళ్ళిపోతారని చెప్పి. సార్కి సెంటీమీటర్ పనిస్తే కిలోమీటర్ పని చేసేస్తారు. సార్, థ్యాంక్ యూ సో మచ్ సార్. Next comes our important transportation committee. Because we don't know when the conference will end, the plenary sessions may drag on. Sometimes the programs may get delayed and we have to make sure that all our works will be done. So for that, these people made sure that they provide transportation to every people, making sure that even at late night they were transported to their respective places. One big round of applause to Vishwanath sir, Yasdani sir, Sulochana ma'am, Rihana ma'am, Teja ma'am, Mallikarjuna sir, Deepak sir and Shafia ma'am. And next comes the accommodation committee. We received more than 70 requests asking for requests. So these people, Kotaya sir, Saujini Lakshmi ma'am, Jansi ma'am and Sivaprasad sir, all these people made sure that all the guests who visited our place were given with proper accommodation, and one big round of applause to all of them. Next, the hospitality committee. Hospitality committee is not just about serving food. It's more than serving food. I'll tell you a very small example. During a conference, everybody will have something or the headache. Registration committee నుంచి కోపంగా ఒకళ్ళు వస్తారు. నిన్నగాక మొన్న ఇప్పుడే వచ్చాడు, నా సర్టిఫికేట్ ఏది అంటాడు. వాళ్ళ ఫ్రస్ట్రేషన్తో అన్నం తినడానికి వస్తారు. సైంటిఫిక్ కమిటీ వాళ్ళ ఫ్రస్ట్రేషన్తో వస్తారు. ఇవాల్యుయేటర్ ఇంకా రాలేదు, ప్రెజెంటర్స్ రెడీగా ఉన్నారు, ఎప్పుడు చేయాలి, వాళ్ళకు ఫ్రస్ట్రేషన్తో అన్నం తినడానికి వస్తారు. ట్రాన్స్పోర్టేషన్ కమిటీ వాడికి ఇంకో భయం, ఎంత లేట్ అవుతుంది, ఎన్ని బస్సులు పెట్టాలి, వాళ్ళకో భయం. ఇలా తినడానికి వచ్చినప్పుడు అంత స్ట్రెస్లో ఉంటే పాపం తింది అరగదు. ఇక్కడ ఉన్న వాళ్ళు ఏం చేస్తారంటే, అవన్నీ మనకి ఎందుకు సార్, రండి తినండి అని చెప్పి అక్కడ ఉన్న వాలంటీర్స్ ఎవరో ఉంటే, అమ్మాయి మీ సార్ వచ్చారు, మంచి ఫుడ్ పెట్టు అని చెప్పి అంటారు. ఐ థింక్ ఆ ఒక్క చిన్న మాట చాలు, వాళ్ళు ప్రశాంతంగా వాళ్ళ స్ట్రెస్ అంతా మర్చిపోయి ప్రశాంతంగా భోజనం చేసి మళ్ళీ ఆ స్ట్రెస్లోకి వెళ్ళిపోతారు. అది తప్పదు. But at least ఆ 5 నిమిషాలకైనా they made sure that they had that plenty meal. And one big round of applause to hospitality committee, Ramesh sir, Saidulu sir, Meeraj Sultana ma'am, Ashok sir, needless to say our PT sir. And please వెళ్ళిపోయేటప్పుడు PT సార్తో ఒకసారి మాట్లాడండి, ఆయన గొంతు చాలా బాగుంది ఇవాళ. And finally, the most important committee is the protocol committee, because stage మీద ఏం జరిగినా సరే protocol committee దే రెస్పాన్సిబిలిటీ అంటారు. సో there is a wonderful combination. అంటే శంకర్ దాదా సినిమాలో శంకర్ దాదాకి ATM ఎలాగో కొత్తగా వచ్చిన రవి సార్ సుబ్బారావు సార్కి అలా తోడారు. ఇప్పుడు దాకా సుబ్బారావు సార్కి ఒక చిన్న ఫ్రస్ట్రేషన్ ఉండేది. నేను చెప్పినట్టు ఎవరు పని చేయట్లేదు, నేను మాట ఎవరు వినట్లేదు. He used to have a lot of frustration since all these days. I think the addition of Ravi sir is a very good thing to him. అంటే వాళ్ళిద్దరు కాంబినేషన్ ఎంత బాగుందో నేను ఇంకంతకన్నా చెప్పాలంటే చాలా చెప్పొచ్చు. వానకి మెరుపులాగా, వరదాల్లో వర్షాల్లాగా అలా చెప్పుకుంటూ వెళ్ళిపోవచ్చు. I think in the coming years that's going to be a deadly combination in this college. And I'm excited and afraid also for that combination. Needless to mention about Sindhu Vaishnavi ma'am and Reshma ma'am. They made sure that they are standing always there, making sure that everybody were given the proper felicitation for their efforts, for their contribution. One big round of applause to both of them also. And also, actually Chakri sir పేరు సైంటిఫిక్ కమిటీలో ఉంది. బట్ సైంటిఫిక్ కమిటీలో ఉంటే ఆయన సైంటిఫిక్ కమిటీ, ఇక్కడేమో ప్రస్తుతానికి ప్రోటోకాల్ కమిటీ. భోజనాలు స్టార్ట్ అయిందంటే హాస్పిటాలిటీ కమిటీ. బస్సు లేట్ అయింది అంటే ట్రాన్స్పోర్టేషన్ కమిటీ. సో అన్ని కమిటీల్లో ఉన్న చక్రి సార్ కూడా వన్ బిగ్ రౌండ్ ఆఫ్ అప్లాస్. And another big round of applause to all our non-teaching faculty, because they are the ones who made sure that everything were arranged, everything were made sure that ante staff members are really busy. వాళ్ళు ఏంటంటే ఇది ఇక్కడ పెట్టండి, అది అక్కడ చేయండి, ఇది క్లీన్ చేయండి అని చెప్తారు. బట్ దే మేడ్ షూర్ దట్ దే అంటే మేము చెప్పిన ప్రతి మాట వాళ్ళు చేసినట్టు మాకు చాలా ఆనందంగా ఉంది. సో వన్ వెరీ వెరీ బిగ్ రౌండ్ ఆఫ్ అప్లాస్ టు ఆల్ ద నాన్ టీచింగ్ మెంబర్స్, టు ఆల్ ద సపోర్టింగ్ స్టాఫ్, టు ఆల్ ద IAs and to all the watchmen who made sure that all the works that were given were successfully handled by them. And last but not the least, all the student volunteers. That's the most difficult job because they are on the receiving end. Receiving end ఎందుకన్నాను అంటే ఫస్ట్ ప్రిన్సిపల్, నెక్స్ట్ ఆర్గనైజింగ్ సెక్రటరీ, దెన్ ఇండివిజువల్ కమిటీ, ఫైనల్ గా స్టూడెంట్ వాలంటీర్. అక్కడి నుంచి వచ్చింది పాపం వాళ్ళకి ఇంక్రీజ్డ్ ఇంటెన్సిటీలో వస్తుంది. అంటే ప్రిన్సిపల్ ఒక ఇంటెన్సిటీతో ఆర్గనైజింగ్ సెక్రటరీకి చెప్తారు, ఆర్గనైజింగ్ సెక్రటరీ ఇంకొక ఇంటెన్సిటీతో చెప్తారు. పాపం వీళ్ళకి మాత్రం మంచి ఇంటెన్సిటీతో వస్తుంది. పాపం ఆ ఇంటెన్సిటీ అంతా వాళ్ళు అబ్జర్వ్ చేసుకుని ఈ 2 రోజులే కదా కానీ చేద్దాం, ఈ 2 రోజులు పని చేస్తే చాలు, సంవత్సరం మొత్తం మనం గుర్తుపెట్టుకుంటారు వీళ్ళు అని చెప్పి దే నో దేర్ లాజిక్స్, దే నో దేర్ టాక్టిక్స్. నీడ్లెస్ టు సే స్టూడెంట్స్ దీస్ డేస్ ఆర్ వెరీ వెరీ ఇంటెలిజెంట్. ఈ 2 రోజులు మంచి మార్కులు కొట్టేస్తే చాలు అని చెప్పి దే మేడ్ షూర్, అండ్ ఎవ్రీబడి ఇంక్లూడింగ్ ద ఎంటర్టైన్మెంట్, డాన్స్ పీపుల్, ఎవ్రీబడి యు నో యువర్ థింగ్స్ అండ్ యూ డిడ్ వెరీ వెల్. థ్యాంక్ యూ ఫర్ ఎంకరేజింగ్, థ్యాంక్ యూ ఫర్ సపోర్టింగ్ అస్. Thank you for making sure that you are all actively involved. And finally, last but not the least, ఇద్దరిని మర్చిపోయాను నేను. ఎవరు? Our principal and our organizing secretary. ఒకే ఒక మాటలో ఫినిష్ చేసేస్తాను. 2024 ఫార్మెయిర్ లోకల్ సెమినార్ హాల్, 2025 ఫార్మా నెస్ట్ సెమినార్ హాల్ LED స్క్రీన్, 2026 ఇన్నోవేట్ ఫార్మా అవుట్ సైడ్ LED స్క్రీన్, 2027 ఒక నిమిషం ఫార్మేర్ లోకల్, ఫార్మా నెస్ట్ నేషనల్, ఇన్నోవేట్ ఫార్మా ఇంటర్నేషనల్. 2027 మరి ఏలియన్స్ తీసుకొస్తారో లేకపోతే ఎక్కడి నుంచి తీసుకొస్తారో సార్ కొంచెం ముందే చెప్తే విల్ మేక్ ఆల్ ద రిజర్వేషన్స్. అండ్ వన్ బిగ్ రౌండ్ ఆఫ్ అప్లాస్ టు ద convener of our conference, our beloved principal sir, and the organizing secretary Venkatrao sir. I think we can say that that is the hat-trick combination: Pharmair 2025, Pharma Nest 2020. Order much pen sir. Pharmair, Pharma Nest, Innovate Pharma. They completed the hat-trick. I think this is the successful Balaya Boya combination of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences. అయితే వాళ్ళు 4వ సినిమా తీస్తారని నాకు తెలియదు, బట్ వీళ్ళైతే గ్యారంటీకి నెక్స్ట్ ఇయర్ 4వ దాంతో రెడీగా ఉంటారు. అండ్ ఐ విష్ దెమ్ ఆల్ ద బెస్ట్ అండ్ థ్యాంక్ యూ ఆల్. వన్ బిగ్ రౌండ్ ఆఫ్ అప్లాస్ టు ఎవ్రీబడి ఆఫ్ అస్ అండ్ విల్ కంక్లూడ్ విత్ జనగణమన. అండ్ థ్యాంక్ యూ ఫర్ లిసనింగ్ టు మీ.
Snacks and tea are served in the food court. Snacks and tea are available in the food court.